Hi everyone, I'm Dr. Preeti Sharma and welcome to the YouTube sessions for the Microbiology crash course. This is particularly for the FMG students who have their exam coming up. However, all the NEETPG and the next aspirants can definitely benefit with this rapid review of the course that we are going to do. So we will be covering in a sequential order all the different chapters and important topics of microbiome, starting from general, going on to fungus, bacteriology, virology and parasitology. Alongside, we'll be practicing all the questions that are important for the FMG students, especially in terms of their PYQs. We'll be also talking about a lot of images, how to identify those images in the exams, and alongside, we'll be completing our theory. So all in all, the most high-yielding topics and important points from your previous year will be discussed. So what do we start off? First and foremost, we need to know about the historical aspects. Especially in the FMG exam in the past years, there have been questions from scientists. From the history point of view, there have been a couple of questions, and that is exactly what we are going to cater to in the first part of the session. So let's begin. The number one scientist who is one of the most important that we have is Sir Louis Pasteur. Well, you also have an image in front of you for Sir Louis Pasteur, and he's definitely contributed immensely to the subject. So what all has he told us about? You can see from the surname itself. When we talk about Pasteur, obviously what has he given us? He's given us pasteurization of milk, which we'll be talking about again in general microbiology. There's also an institute set up on his name that is referred to as the Pasteur Institute in Paris, and that is all that you have from his name, that is Pasteur out here. Whereas what do you have from his first name, that is Louis, L for Louis and L for liquid. Sir also gave us about the liquid medium. Now we are going to read a lot of media today, and you will actually realize that all the media and culture media that I talk about are solid culture medias, like the blood agar, the chocolate agar, they are all solid culture media. But Sir Louis Pasteur in the primitive earlier days gave us the first liquid media with which further advances were made. So what all have we learnt up till now? We know about Sir Louis Pasteur giving us pasteurization of milk, naming a Pasteur Institute of Paris, and L for L, Louis Pasteur for liquid media. What else? Guys, he's also known as the father of microbiology. He's referred to as the father. And when we say father, we can take every alphabet out of father and come to a particular answer. So when we say father, the F will tell us about the fermentation principle. He's given us the principle of fermentation. When we talk about AT, he's given us AT autoclave. Autoclave was described by Sir Louis Pasteur. And when we talk about THER, he's given us one theory, and THER for theory. And which theory has he given to us? He's given us the germ cell theory, or the germ theory has been given to us, and he said that germ theory is something that you have to accept, and abiogenesis theory is something that you have to reject. So one theory for acceptance, one theory for rejection. Germ theory for acceptance and abiogenesis theory for rejection. What else? So let's repeat, because we called him father, F for fermentation, AT for autoclave, THER for theories, and finally Sir has given us the CAR vaccines. What are the CAR vaccines? C for cholera, number one, A for anthrax, and R for rabies. Repeating, cholera, anthrax and rabies, their vaccines, not the organisms. I'm repeating, the organisms were not identified by him, but the vaccines for these diseases were definitely started off. So these are all the different kind of contributions that Sir Louis Pasteur has, and we can go on to the next one. So we'll finish all the scientists and then we'll take up the MCQs, right? So the second one that we have, that is Robert Koch. So Robert Koch is said to be the father of modern microbiology. Repeating, Sir Louis Pasteur was father of microbiology. Sir Robert Koch is the father of modern microbiology. And look at Sir's name, surname, because a lot of things are named after it. So when we talk about K for Koch, he's given us something called the Koch postulates. He's also identified the Koch bacillus. What is the Koch bacillus? The Koch bacillus is nothing but TB, tuberculosis, tubercle bacillus. Then when we go to the next alphabet, that is C, he's given us C, C, C. Three things with C is what he's given us. Number one, he's given us the cholera bacilli, the organism cholera, Vibrio cholera has been given by him, which you all have to learn that cholera vaccine, again repeating, cholera vaccine was given by Sir Louis Pasteur, the CAR vaccines, but cholera organism has been given by Sir Robert Koch. What else with C? Finally, he gave us the solid culture media. Again, recap. We have Louis Pasteur for liquid culture media, Louis Pasteur for liquid culture media, and Sir Robert Koch for solid culture media. What else with C? He's also given us a color or a dye, and that is known as the aniline dye. The color or the dye is referred to as the aniline dye. Coming to the last alphabet, H. H for hanging drop motility. Hanging drop motility testing was also given by Sir. Repeating, first we'll take the Koch alphabet, Koch postulates, Koch bacilli. Then we'll take the C, three things with C, that is cholera, culture media, color, and one thing with H, that is the hanging drop motility. Let's move on to the other scientists. Let's come to Sir Paul Ehrlich. Sir Paul Ehrlich, after his surname, there's an organism that has been identified that is known as the Ehrlichia organism. The Ehrlichia organism is named after him. Other than that, please remember, if you look at the last two alphabets, CH tells you he is the father of chemotherapy. So firstly, Ehrlich will tell you Ehrlichia organism. CH will tell you it's the father of chemotherapy. And PA, what is PA? P for prize, A for acid fast. P for prize. What prize? He's actually a Nobel Prize winner. He's a Nobel Prize winner, and what Nobel Prize has he won? For telling us that toxins and antitoxins work against each other. How he standardized that a toxin can be counteracted upon by an antitoxin. So he's standardized the toxin antitoxin concentration, for which he also received a Nobel Prize. So P for prize, and what is A? A for acid fast stain. Nowadays you know acid fast stain with some other name. Nowadays you know acid fast stain as the Zn stain, guys, right? So that is what you have to know, that originally the acid fast stain was given by Paul Ehrlich. Later on, it was modified by Z and N, Zeal and Nelson. Zeal and Nelson modified it, and today we know it as the Zn stain. But originally, who gave us the acid fast stain? It was Sir Paul Ehrlich. Moving on to the next scientist, it is Joseph Lister, and if you read the name carefully, Joseph Lister is how we call it, because he's the father of antiseptic surgery. He's the first one who told us that during surgery you must use antiseptic, or you must use asepsis in the operation theatre. So he's the father of antiseptic surgery. Joseph Antisep is how we learn it. Coming to the fifth one, which you've been reading ever since you were preparing for your undergrad NEET UG days, Anton von Leeuwenhoek. Anton von Leeuwenhoek is said to be the father of microscopy. And which microscopy are we talking about over here? The usual light microscope. So if light microscope was by Leeuwenhoek, L for L, if light microscope was by Leeuwenhoek, electron microscope is by whom? Electron microscope is by Ernst Ruska. Let's do a recap. When we are talking about light microscope, L for L, it is by Leeuwenhoek. And when we are talking about electron microscope, it is E for E, it was given by Ernst Ruska. Then there's another E, Edward Jenner. This also you read, I remember we used to read it in our undergrad days when we were preparing for NEET UG. Edward Jenner. Edward, you'll say, ma'am, this is also E. How will I not confuse it with Ernst Ruska and electron microscope? Edward Jenner was awarded because he gave us the vaccine. The first vaccine, I hope you all remember, the first vaccine was for smallpox. So Edward gave us the vaccine for smallpox, the first ever vaccine that we had. Coming to the last scientist, three names, or three, first name, middle name, last name. Carrie B. Mullis has given us something which has three alphabets again. So the scientist with the three alphabets has given us a technique with the three alphabets, and that is PCR, polymerase chain reaction. Well, that is a wrap up of the most important scientists that we have. And why don't we practice a few questions in order to know whether we've been able to recall this well. Okay, cholera vaccine. Repeating again, there are two kind of questions. Cholera vaccine has been discovered by, or cholera bacillus has been discovered. Over here, vaccine. If you all remember, we had something called the CAR vaccines. CAR vaccines means cholera vaccines. Number two, anthrax vaccine, and number three, rabies vaccine. And all these rabies vaccine, anthrax, cholera, they were given by Sir Louis Pasteur. On the other hand, if I would have asked you, cholera bacillus, that is the organism has been given by what? By whom? Then cholera bacillus has been given by Sir Robert Koch. Coming to question number 2, who is the father of antiseptic surgery? The father of antiseptic, how did we learn it? Antisep, Joseph. So the father of antiseptic surgery is Joseph Lister. Coming to question number 3. Electron microscopy was discovered by. There was a mnemonic for this also. And as you're watching the video, my request to you all, whenever you get a question, try to pause the video, try to give an answer on your own before I start discussing, so that you know what you were thinking and then you know whether you got it right or not. Okay, electron microscope, E for E. So there are two E's over here, Ernst Ruska and Edward Jenner. I hope you remember the answer is Ernst Ruska. Edward Jenner has given us what? Edward Jenner has given us the vaccine, smallpox vaccine. Coming to the next question. Acid fast stain was discovered by Joseph Lister, Paul Ehrlich, Louis Pasteur or Robert Koch. Acid fast stain. So if you all remember, correct answer, Paul Ehrlich. Do you remember what all Paul Ehrlich sir had given us? Let's do a recap. Firstly, Ehrlich tells me that Ehrlichia organism was given. Then CH tells me chemotherapy. He is the father of chemotherapy. P tells me he got a prize. What did he get a prize for? Toxin, antitoxin. And A tells me that he's given us the acid fast stain. So Paul Ehrlich happens to be the final answer. Coming to the next PYQ guys. PCR was discovered by. Polymerase chain reaction was discovered by 1, 2, 3 needs to have a scientist with 3 names, 3 alphabets. Carrie B. Mullis gave us polymerase chain reaction. Well, that is a wrap up of all the scientific aspects. I hope all the scientists are clear to everyone and we can move on to the next topic. Now, the next topic is something you probably see in every microbiology and every pathology lab. However, this is probably also something which you never like to switch on because you many a times don't understand what is seen under it. But the examiner is really fond of all the parts that you see in a microscope. So one by one, firstly, what kind of a microscope is it? Guys, this happens to be a light microscope. This is a light microscope and the first question that we'll ask you is, what kind of light are we talking about over here? We have different kinds of light. We have transmitted light, we have reflected light. So what kind of light is being spoken about over here? You'll say, ma'am, look at this. This is the bulb. This is the bulb, this is the light. I will switch on the bulb, I'll connect it to an electrical source. I'll switch on the plug, I'll switch on this bulb and the light will just get transmitted like this, like regular light from any other bulb in the room comes, right? So what kind of a light am I talking about over here? In a light microscope, we are talking about transmitted light. Like any other bulb or tube light or light source, light is getting transmitted. So first and foremost, you understood the first thing and that is known as a light source. Okay, from the light source, the light starts coming. Then what else do we have? You'll say, ma'am, these are all the light rays. Now a bulb will obviously give light in all directions, right? Bulb is going to throw light in every direction, but I want it to be focused. I want it to go towards one area. So what am I going to do? I am going to put out a condenser over here. Can you all see this tiny little structure at the bottom? We label it, this tiny little structure is referred to as a condenser and there are two things inside it. Remember that tiny little structure which I am putting up, this round black thing, this is a condenser. And do you know what is fitted inside a condenser? Inside a condenser we have something called iris. Just like you have the iris in the eyes to see, what does the iris in the eye do? It regulates how much of light is going in or not. Same way, there is a condenser which has an iris inside it which will make sure how much of light is going through it. Where is the light going? Let's highlight the next thing. The light is going here. What is this square over here? This square is referred to as the stage. We call this the stage of a microscope. Stage is where the hero will be sitting, right? The hero and all the main lead actors will be on the stage. What's the main lead actor? Your glass slide. Whenever you want to see a glass slide, you will be putting it over here. All those glass slides that you get, which you conveniently ignore in your path and microbiome labs, all those glass slides are kept over here. You'll say that, ma'am, everyone will want this at a different level. Stage might be wanted a little up, a little down, because my power of the eyes and your power of the eyes are different, right? So we have these two knobs that they've given to us, the big knob and the small knob. The big knob is said to be the coarse adjustment. The big knob is said to be coarse adjustment. The small knob is said to be fine adjustment. Repeating, the big knob is coarse adjustment and the small knob is the fine adjustment. What else? We have something over here. This is where you are sitting. This is where your eyes will be there. So there's a lens over here which is known as the eyepiece lens. There is an eyepiece lens which is present over here, and this has a magnification of 10 times. It has a magnification of 10 times. Versus, do you see a lot of lenses over here also? That is where the examiner asks you maximum questions. What are these sets? See, over here there was eye, your eye is here, this is where you are sitting and seeing. So this is the eyepiece lens. What are these set of lenses known as? These are said to be objective lenses, and that is exactly what I want to teach you next. If you've learned all the names, I'll teach you the details now. See, let's do a repeat. What you see at the bottom, this is a light source. After that, what you see where the light is going, that's a condenser with an iris. Then the light is going on to the stage. The stage can be moved up and down by the coarse and the fine adjustment. Thereafter, you have a set of lenses over here called objective lens, and then you have a lens over here that is known as the eyepiece lens. Now, what are the other details that you have to know in terms of magnification? So listen to me carefully. You will say, ma'am, one set of lenses that you've told me are the objective lenses, and one set of lenses that you've told me are the eyepiece lenses. And you've told me eyepiece lens is only one and it has a magnification of 10 times. But objective lens is of 4 times. Now that is what requires attention. One lens will only help you scan the slide. Like you scan a page on a printer, like your human eye, a little bit magnified, 4 times magnified, only 4x. That is a scanner lens. Okay, next you have something you'll say, ma'am, I want to increase from scanner. I'll go to low power magnification. Then I'm going to go to a higher power magnification, and finally I'm going to look at the slide with oil. I'll put oil on the slide and I'm going to look at it with oil. So I will then use the oil immersion lens. What is the difference? Scanner was what magnification? 4 times. Low power is 10 times. High power is 40 times, and oil immersion is 100 times. Simple to learn. What is the protocol? 4, 10, 40, 100. Repeating, we have 4, 10, 40 and 100. Scanner, low power, high power, oil. 4, 10, 40 and 100. So what do you think? Now if I ask you, what is the total, or what is the, how do you calculate the total magnification? You will have to add both of these. You will have to add. So for example, you say, ma'am, this is your microscope. You are looking at the microscope from here, your eye. So 1 is fixed. 10 times will always happen. Then depends which one are you using from here. Are you using the 4x? Are you using the 10x? Are you using the 40x, or you're using the 100x? So for example, I'm using the 4x. So what is the total magnification? 4 into 10, 40. So let's do from here. If I want to calculate total, you'll say if you're using the scanner, then your total becomes 40 times. If you're using low power, then your total becomes 100 times. If you're using high power, then your total becomes 400 times. And if you're using oil immersion, 100 into 10, your total becomes 1000 times. So if I ask you, what is the maximum magnification that you can obtain in a microscope? Think logically. What's the maximum magnification? You will say oil immersion will give me 1000 times, and the eyepiece is going to give me a 10 times. So what is the maximum magnification that we can receive? It is a 1000 times. 100 into 10, 1000. That's one of the most favorite questions that you have in the previous year papers. So that's it about the light microscope. Let's move on to some other microscopes. Let's take up a dark field microscope. So what do you understand by dark field microscope, guys? Please remember, when you're dealing with dark field, you can see the background is very, very dark. Why is the background dark? Because you are dealing with dark as R, so you're dealing with reflected light. Now you know that why I was emphasizing so much on the type of the light, because when you're dealing with a light microscope, light has a transmission to it, transmitted light. When I'm dealing with a dark field microscope, I'm talking about a reflected light. The background will be dark, and anything that is very, very thin. What are you going to use it for? Anything that is very, very thin, like a spirochetes. Or for that matter, guys, even flagella of the bacteria is very, very thin. Anything that is very thin can easily be picked up on a dark field microscope. Coming to the next one, everyone I'm sure has seen this microscope earlier. This microscope is the very famous fluorescent microscope. What is the light source that we have for fluorescent microscope? The light source is the UV light. It is the ultraviolet light. So let us analyze a few things. You will say that, ma'am, fine, you've told us that fluorescent microscope is going to do with ultraviolet light, but that ultraviolet light, ultraviolet light you've seen at so many places. Forget this microscope, but ultraviolet light you've seen at so many places, right? Even in day-to-day life, you might get to see in some instrument, in some machine, ultraviolet light is used. But not every ultraviolet light is going to give me this kind of a green color, because when you're dealing with a fluorescent microscope, the light is ultraviolet light, but what you're also using is a shiny color. You're using a fluorescent dye. You'll say, ma'am, which dye are you using? For example, this is a slide. You say that, ma'am, there's a patient in our country. This patient has come up with cough with sputum. So cough with sputum in our country, the first thing that I think logically is it could be a case of tuberculosis. If I'm suspecting tuberculosis, this is the sputum sample that is there on the slide. What is the dye that I will add to it? If you're suspecting TB, auramine and rhodamine are very famous fluorescent dyes. So please remember, auramine and rhodamine are the fluorescent dyes. Coming to next, if you're suspecting fever with chills, malaria, what is the fluorescent dye? Acridine orange is the fluorescent dye. And number 3, if you're suspecting a fungus, anyway, fungus has become, once upon a time in the second wave, was quite a hero, second wave of COVID. So fungus, fluorescent stain, remember fungus, fluorescent is calco flower white. Fungus, fluorescent is calco flower white. Repeating, for TB, it is auramine rhodamine. For malaria, it is acridine orange. For fungus, it is flower white. And what will then happen? So when this UV light comes in contact with any of these dyes, the next question that they will ask you in the paper is what wavelength changes to what. So please remember, shorter wavelength changes to longer wavelength. Initially, the ultraviolet light had a shorter wavelength. When it will come in contact with the fluorescent dye, it will change into a longer wavelength. So shorter to longer is what you know for fluorescent microscopy, and that's the final outcome. That's the final outcome that you get. You finally get to see all these fluorescent color organisms, and this was actually a case of TB only. You can see the bacilli. So now if I ask you if this is actually a case of TB, what fluorescent dye must have been used? The fluorescent dye that must have been used must have been either auramine or it would have been. Okay, so this one's also done. Let's move on to the next microscope, the very, very famous electron microscope. So first, the picture is quite characteristic. If you look very carefully, if we look at this very carefully, we will see that it looks somewhat like a spaceship. It looks like a space shuttle because it's closed from everywhere. Why is it closed from everywhere? For example, if I keep it here, this is the electron microscope. If I want to peep inside it and ask you what is the source, you'll say, ma'am, for everything you were telling me a light. Let's do a recap. For example, for light microscope, I told you the source was transmitted light. For dark field microscope, I told you the source was reflected light. For fluorescent microscope, I told you the source was UV light. But for electron microscope, there is no light. The source is a beam of electrons. The source is a beam of electrons, and electrons always have to be kept in vacuum. You can't keep electrons out in the open. You have to keep it in a vacuum, and that is why they've created this kind of a vacuum out here. That is why this looks like a closed space shuttle kind of thing. What is the next difference? When you take the electrons and they are in a vacuum, finally you'll say, ma'am, at the end of the day, you have a slide, you have a specimen on top of it. The difference is that in light microscope, when you're given a slide during your practical exams, is that slide made up of glass? Is it a glass slide? Yes, because in light microscope we use light. Light and glass are, compatible. But when I'm dealing with electron microscopes, I can't put electrons on a glass. With electron microscope, I will have to use something called a metal slide, and that metal slide is usually made up of copper. That metal slide is usually made up of copper. So remember, this slide that we are using over here is going to be a metal slide, and finally you will be able to see the picture on an electron microscope. There's one more difference that we have. Actually, that's a very famous pathology question, but even in microbiology, they tend to ask you that what is the difference in the fixatives? Like, for example, let's take an example of, say, a kidney biopsy. A surgeon has performed, a nephrologist has performed a kidney biopsy, and all of us know that in kidney we have to do light microscopy also, and in kidney we have to do electron microscopy also, right? Okay, so when both of these are decided anyway, then I would say that fine, this kidney, there are two, from here you've taken one tissue, from this area you've taken another tissue, you have two tissues. I'll say send one for light microscope, send one for electron microscope. So you will put them in what solutions? What containers will you drop first biopsy in? What container will you drop the second biopsy in? What solution? So the one that you're sending for light microscope, you will drop it in a container of 10% neutral buffered formalin. 10% neutral buffered formalin, and in a container which you want to send for electron microscope, you are going to use 2 to 2.5% butylaldehyde. You are going to use 2 to 2.5% butylaldehyde. So repeating, for light microscope it is 10% NBF, for electron microscope it is 2 to 2.5% butylaldehyde. Very, very important questions. Now that we've done the microscope, just a quick one or two questions to assess our knowledge. PYQ. Maximum magnification that you have of a light microscope. Repeating, maximum magnification that you have of a light microscope, 10x, 100x, 40x, 1000x. So they've asked you the maximum magnification, and I think everyone knows, we've just done this, that you have to multiply the maximum of the objective lenses with 10. So 100 into 10, you get 1000x. That's the maximum, right? That solves your question perfectly. Let's move on to the next question. Which of the following fluorescent stains is correctly matched? Which of the following fluorescent stains are correctly matched? Let's go one by one. Acridine orange for TB. Does not sound right. If I remember correctly, acridine orange is for malaria. Acridine orange is for malaria, and TB के लिए we have auramine rhodamine. For TB we have auramine rhodamine. So this is not correctly matched. Auramine and rhodamine for malaria, again a mismatch. Auramine and rhodamine are for TB, and for malaria we have acridine orange. Coming to the third one, calcoflower white for fungus. That is sounding right. Calcoflower white for fungus. Up till now, I think this is a right statement. Coming to the fourth one, acridine orange for fungus, not at all. For fungus, it is calcoflower white. For acridine orange, that is basically used for a case of malaria. This is also incorrect. So what is the final correctly matched statement? The correctly matched statement, calcoflower white for fungus. Okay, these were the two quick questions that we had, and I think now we can move on to the third topic. I hope you guys are not finding microbiology up till now to be a very tough or an extremely lengthy, and it is definitely cramming up, I wouldn't deny it. But yes, a few mnemonics and concepts here and there will help us sail our boat through, right? Okay, let's go on to the next topic of general microbiology, the bread and butter of micro, the different staining techniques. And I think while I've said bread and butter, I'll start with genuinely the bread and butter, that is gram staining. No microbiology lab can be ever made without starting a gram stain. So what do you have? What all questions can you get? Either they'll give you the names of these chemicals and they'll say identify the stain, or they'll give you these colors, pictorial representation, Or they'll simply tell you that, okay, I've given you these 4 chemicals, arrange them in the right order. So how do you identify and how do you deal with a gram stain? The mnemonic is come in and stain. If you want to learn it, simple, that's the protocol. If you want to learn gram stain, then you just have to come in and stain. So what is this? Come in and stain. CIAS. That is how we learn it. We call it come in and stain. So what is the first thing that you add? The first thing that you add is crystal violet. You can see a violet color. So please make sure when we add crystal violet, the gram positive organisms and the gram negative organisms, both of them are going to become what in color? Violet, purple in color. So whether it is a gram positive or it is a gram negative, on adding crystal violet, both of them will become purple in color. After crystal violet, I for iodine. Iodine is said to be the mordant. What is a mordant? Mordant is like a hammer. Like if you put a nail and then you hammer it so that the nail goes inside the wall. Same way, if you put a stain, you want it should go to all the bacteria. So you put a hammer on top of it. Iodine is that hammer which will make sure that crystal violet reaches all the bacteria. So that hammer is known as a mordant. Next, you use alcohol. Either you can use alcohol or acetone. Have you seen the color has vanished? Because alcohol or acetone is a decolorizer. Means it's going to pull all the color out. So now telling you, initially the gram positive and the gram negative both had got a purple color. On adding alcohol, gram positive doesn't show any change. Gram positive still remains purple because positive and purple will always go together. The gram negative ones are going to become colorless. What is your mnemonic? Positive people, positivity stays till the end. Once positive, always positive. So remember, positive gram bacteria, the gram positive bacteria which had become purple once, they will still remain purple always till the end. But gram negative, negativity makes you lose everything in life, correct? Exam going students, remember, negativity will make you lose everything in life. So negative bacteria will lose the color, they will become colorless. You say, fine ma'am, now add a color to them also. So there you add the last red color thing that you're putting over here. Safranin. Safranin is the last stain which now you'll say, okay, positive ones from the beginning to the end have remained purple. The negative ones had lost the color in between. So now the negative ones have also become red because what have I got? I'm finally having safranin. Finally having safranin. So this is your final interpretation of a gram stain. So let's repeat. When you look at a gram stain, can you see all these things which are purple in color? All these organisms which are purple in color, they are going to be gram positive. And all these organisms which are reddish in color, reddish pinkish in color, those organisms are definitely going to be gram negative. I hope the entire protocol is clear. Come in and stain. Yes. Coming to the next one, that's another one which you have in terms of the bread and butter of microbiology, and that is the Zn stain. Zn stain, also known as the acid fast stain. Zn stain, also referred to as the acid fast stain. What do you have to know in this, guys? Let's proceed. So there's a mnemonic for this also. So once a student had asked for a mnemonic in class, I was just teaching it generally. The student said, no ma'am, we need a mnemonic. So then we made an impromptu mnemonic, and that was class has asked mnemonic. For Zn stain, that is how we learn it. Class has asked mnemonic. So what is this CHAM? Let's start. What do you add with C? With C, we add something called carbonyl fusion. The first thing that you're adding over here is carbonyl fusion. With H, you can see that there is heating going on. First, you put carbonyl fusion. After that, you're going to put heat. You're going to heat the slide. After that comes A, you put acid. Most of the times you put sulfuric acid. Most of the times. Most commonly added is A for acid, sulfuric acid. And finally, CHAM. In the end, you have a second stain. Can you see something bluish in color? They've added something called methylene blue. They've added something called methylene blue. So repeating, CHAM, carbonyl fusion, heating, acid, and methylene blue. Sometimes, okay, now if you look at the colors, when you added carbonyl fusion, what was the organism becoming? Everything was becoming red. So please remember, carbonyl fusion gives a red color. And when in the end you're adding methylene blue, you'll say, ma'am, the background things are becoming blue. So what are the two colors that you have in acid fast state? You'll say, ma'am, we primarily have red and blue. And now if we see this particular picture, I think you will be able to guess this very, very well. And what do you have over here? Have a slide, and that slide is classically showing you that the organism, this is a case of TB guys, the organism has come out what in color? So let's ask you, when we ask you in terms of the result, the organism has come out to be red in color, and if they ask you what is the color of the background, you can very well see that the background has come out to be blue in color. So, you know, the examiner is very smart. He won't give you the photo. He'll ask you as a fill in the blanks that in the Zn stain or in the acid fast stain, organism is what in color, background is what in color. So, can we see that the organism is nicely yellow in color, the organism that I've marked over here is nicely red in color, and the background that you're having is blue in color. And this is actually a case of TB. So, the next question coming up in front of you, you used carbonyl fusion, then you used heat, then you used acid alcohol, then you used methylene blue. Let's write that down once again and see some variations. Repeating, carbonyl fusion, heat, acid, methylene blue. If I say in the first, particular variation that heating has not been done, I have removed this heating step. You will say, ma'am, if you're not heating, mainly you're doing carbonyl fusion, then you're doing alcohol, acid alcohol, then you're doing methylene blue. You're not heating. So what kind of Zn stain will you call it? You will now call it the cold Zn stain. We will now call this the cold Zn stain because you're not heating it, right? And this is known as the Quignon or the Gabbett stain. This variation is said to be the Quignon or the Gabbett stain. Repeating, the cold Zn stain is known as the Quignon, KK sound. The cold Zn stain is known as the Quignon or the Gabbett stain. Okay, now having said that, moving on, let's go on to another variation. Firstly, okay, if I ask you, like I think this question must be coming in your mind, that ma'am, why have you removed the heating process? Why have you made it cold? For what purpose? What is the use of the Quignon or the Gabbett stain? Remember, there's a parasitic family, many of you might have heard of, known by the name of coccidian parasites. Coccidian parasites, again a K sound coming. They don't need the heating process. Even without the heat, they can get stained. So remember CC or KK sound. Cold Zn stain is known as Quignon stain and it is used for the coccidian parasites. Let's do one more variation. Again, we'll write it down again. So what do we have? The Zn stain says carbonyl fusion, then heat, then acid, then methylene. Now they ask you what concentrations of acid are you using. So please remember, what are the different concentrations of acid that we are using? Anything where the word TB comes, TB like tuberculosis or any atypical mycobacteria, anywhere the word TB comes, we are talking about 20% H2SO4. We are dealing with 20% H2SO4, T20, right? T20 is how we learn it. So anything to do with TB, we say T20. Next, anything to do with leprosy. What is leprosy? Lepra bacillus. Anything to do with leprosy or lepra bacillus. How many alphabets does lepra have? 5. That means we are going to use 5% H2SO4. 5% H2SO4. Then for things like nocardia and legionella, we use 1% H2SO4. For nocardia and legionella, 1% H2SO4. And finally, if you want to stain the spores of a bacteria, you go in for the least concentration, that is 0.25 to 0.5% H2SO4. So basically I've written it in a decreasing order, right? For TB, you can very well see that for TB and atypical TB, T20 you will consider. For lepra, you will count the alphabets, 5 alphabets, 5% H2SO4. For nocardia and legionella, it is 1% H2SO4. And for spores, it is 0.25 to 0.5% H2SO4. Last question pertinent to this table is, out of all of these, which of them is known as the phytophoraco stain? Very, very important question. Which of these is known as the phytophoraco stain? So what does phyt stand for? Or what does it sound like? You will say, ma'am, phyt sounds like 5. So which of them was using 5% H2SO4? The leprobacillus was using 5% H2SO4. So can I say the same Zn stain in which I'm using 5% H2SO4, basically I'm using it for the lepra, that has been given a separate name called as fight for ACO. So easiest mnemonic, fight sounds like 5. So the 5% H2SO4 variety is known as the fight for ACO stain for the 5 alphabet leprobacillus. Okay, so having said that, now the next thing that you would want to ask me that, ma'am, right now you told us TB, leprosy, nocardia, are these the only things that are positive for Zn stain? No, now comes the next thing. What are all the things that you have in microbiology that are Zn positive? Or in other words, you can say that is acid fast positive. So please remember there's a mnemonic for that. And that mnemonic is my nose is cold and hot. How did I make this mnemonic? Because I got to know that Zn stain can happen with heating also. Zn stain can happen without heating also. Did I just tell you that? Zn stain can happen with heating also, the routine wala, and Zn stain can happen without heating also, the cold one, right? So then we made a mnemonic, my nose is cold and hot. So what are the things which are Zn stain positive? My stands for everything with mycobacteria. My stands for everything with anything of mycobacteria, mycobacteria TB, mycobacteria leprosy, or any atypical mycobacteria. As long as the word mycobacteria is coming, you should be happy that yes, mycobacteria for my. Coming to next, no. No for nocardia and legionella we put together. Nocardia and legionella we are going to put together. Then we come to is for isospora, is for isospora, C for cyclospora, C for cyclospora, and one more C, that is cryptosporidium. Isospora, cyclospora, and cryptosporidium. What do they belong to? For those who have not read parasitology, nothing to worry. Let me tell you, Isospora, cyclospora, and cryptosporidium are nothing but members of which family? They are the members of the coccidian parasites. And have I told you that for coccidian parasites, will you use the regular Zn stain or will you be using the cold Zn stain? The Kinyon variety is what you will be using over here. Moving on to the next, my nose is cold and hot. So HO, the HO that you have over here are hooklets of hydated. Please remember HO for hooklets of hydated are acid fast, and T, the T is left. The T is eggs of Taenia saginata. Taenia saginata eggs are also going to show you an acid fast appearance. So repeating, my stands for mycobacteria, nose for nocardia legionella, is for isospora, C for cyclo and cryptosporidium, HO for hooklets of hydated, and T for Taenia saginata egg. These are all the things that are Zn stain positive. One more stain that I wish to discuss at the outset is this one. You will say, ma'am, this is looking a little different because the background has got a color. In everything when I was showing you, I used to say, look, bacteria has a color. In the next photo that I showed you, again for Zn stain, I said, look, the bacteria has a color. But now I'm showing you something different. I'm saying the background has a color. So the background is going to take up the stain. The organism is going to be unstained. The background is going to pick up the stain and the main organism is going to be unstained. So what do you think? Is this a routine one or is this going to be something like a negative stain? So please remember, this staining protocol is a little different. Like you had negatives in a film, negatives in the Polaroid and the other photos that we used to take long back. Now obviously very few cameras are used, but we used to click and we used to get a film, a negative film from which we could develop more pictures. So what is a negative stain? It looks something like a negative film. It looks very black in the background. Negative stain is particularly used for organisms that have a capsule, because you can't put color to the capsule. In many of the organisms you can't put color to the capsule. So what do? You put a color to the background. You will give a color to the background so the capsule will shine out. The capsule will get highlighted that. Okay, so please remember this is very commonly used for cryptococcus. Cryptococcus, which we'll be studying in the next session, which comes basically under fungus or mycology. Cryptococcal meningitis we will study. So whenever you want to pick up cryptococcus, you do negative stain. Any examples of a negative stain that you heard of? Remember, there's something called an India ink stain. There's India ink stain. There's something called a nigrosin stain. India ink and nigrosin stains are negative stains that we have. Okay, well, the most essential stains that you need for the exam are somewhere done, and I can probably ask you this question now, which has come as a PYQ in the FMG exam. Gram staining is what? Is it a simple stain, a differential stain? Is it a dry heat sterilization, or none of the above? So this much I think I know, that it is not any sterilization technique, it is definitely a stain. So tell me, is it a simple one color stain, or is it helping me differentiate? As far as my thought process goes, gram stain differentiates gram positive organisms versus gram negative organisms, right? It differentiates gram positive versus gram negative, which basically means that this is a kind of a differential stain. This is a kind of a differential stain. Let's move on to the next question. What is the correct order of this gram staining? Think of your mnemonic and then select the answer. I give you 5 to 10 seconds. Select your answer, gram staining. If you remember the mnemonic, come in and stain. Come in and stain. This means the purple color one and the red color one. The purple color one was crystal violet. I was for iodine. A was for alcohol or acetone, and S was for safranin. Come in and stain. That's the protocol that we have for gram staining. Crystal violet will give a purple color, as all of us know, and safranin will give a red color. Coming to the next question. What is the stain used in the preparation of the AFB? Acid-fast bacillus. The stain used in the preparation of acid-fast bacillus. Jimsa, Loeffler's, Zeal-Nelson, or India ink. I think we've understood. Acid-fast means we are dealing with the Zeal-Nelson stain, none of the others. And right now, one more question that you can get. India ink. India ink is what kind of a stain? India ink is a negative stain, means whenever you want to stain something like a cryptococcus, or you want to stain something that has a capsule, negative staining is what you use. Coming to the next question. In Zn staining, what is the secondary stain? In Zn staining, what is the secondary stain that you have? Crystal violet, safranin, methylene blue, or alcohol? So think of the mnemonic for Zn stain. Class has asked mnemonic. What is a secondary stain? First, let's write down. Class has asked mnemonic means I first add carbonyl fusion. That's the first red color that I add, primary stain. After that, I do heating. After that, I put an acid, which everyone knows, sulfuric acid, H2SO4. And then after that, we add something called methylene blue. So this becomes the next color. This becomes the secondary color. The stain, the color that you add first will be primary. The color that you add second or last will be secondary. Over here, the secondary stain was asked. So the secondary stain definitely becomes methylene. Let's practice another question. Metachromatic stain. Now this is something I'm giving you as homework, because this stain I will be teaching you under the bacteriology section. So in the next section, obviously I will be teaching you bacteriology, and that time I'm going to give you the answer. So this is going to be a little bit of a homework, and if you know the answer to this in this session or in this video only, you can always type out and let me know what are metachromatic granules, which of the following are the stains which are going to stain them, which bacteria am I talking about. So which bacteria and which stain? If you know the answer, put it in the comments. If you don't know the answer, the next session of bacteriology, this is going to be very, very, very important. Okay guys, staining methodology questions are done. I think we can move on to the next one, and the next one is actually very interesting. I'll first make you attempt the question. The next topic is going to take care of this. Mesosomes are formed by the invagination of what? Mesosomes are formed by the invagination of cell membrane. Invagination more prominent in gram-positive bacteria. Both the statements are true. both the statements are false. This is not a PYQ, this is a new question that we brought up, but is something which is definitely expected. So this is about the anatomy of a bacteria. What all does a bacteria have? So what we'll do, first let's discuss the entire anatomy and then let's come back to the question. The answer to this is both the statements are true. So let's talk about the bacterial anatomy. I've tried to draw one hand-drawn diagram before we go on to something very big. Let's do something basic that we have over here. Okay, so the first thing that you see over here, you'll say, ma'am, let's start from the outermost. This outer pink color thing that you have, that is either a capsule or a slime layer. So obviously, common sense tells me capsule will be very, very rigid. If the organism has a capsule, it's going to be very, very rigid. And slime layer is going to be very, very loose. So the organism will have either it will have a tough, rigid capsule or it will have a slime layer. So all of you can see that the outside is made up of capsule. Now there's a question on the capsule that you get, guys. Please note, what is a capsule made up of? Remember, capsules are made up of polysaccharides. Capsules are made up of polysaccharides. If I said that, this means there will be some exception. And yes, there is an exception which I'll be teaching you under bacteria, that is Bacillus anthracis or Bacillus anthrax. Remember, Bacillus anthracis does not have a polysaccharide capsule, it has a polypeptide capsule. That's going to be a very important question that we'll be doing under bacteriology. Repeating, all the capsules that you have are polysaccharides, but the one that you have in Bacillus anthracis, that is going to be a polypeptide capsule. Coming back, what do you have after capsule? Look at the inner ones. Look at the black and the green color layers. They are the cell walls. They are both part of cell wall. So this black color one also and the green color one also, they are all cell wall, and that is where 10 star most important questions are going to come. Why? Because cell wall of gram positive and gram negative will vary. I'll show you another picture. Have a look at these photos. I'm sure you've seen them in your textbooks. This is the cell wall of a gram positive and this is the cell wall of a gram negative. So ma'am, how did you identify just within 2 seconds? What made you identify gram positive, gram negative within 2 seconds? Let's see. Let's try to, you remember in sometimes in our free time when we get those things or links on Instagram or probably on Facebook where they tell us to select the differences between two very similar looking photos. So these are two very similar looking photos and I've given them to you to find out the difference. You'll say, now first I'll tell you the similarities. This also has something called NAG and NAM, and this also has something called NAG and NAM. Means N-acetyl glucosamine, N-acetyl glucosamine and N-acetyl muramic acid is over here. N-acetyl glucosamine and N-acetyl muramic acid is over here. So both of them are present in a gram positive and a gram negative cell wall. But then I see something else also. I realize that there is something extra that is present. There is something extra. The yellow color ones are what? The yellow color ones are NAM. So these yellow color ones which are NAM, I am seeing that they are getting connected by 1, 2, 3, 4, 5 molecules. I've also tried to draw it out. See, you've got NAM and NAG, NAM and NAG, NAM and NAG. But in a gram positive organism, you will say that ma'am, NAM is connected. NAM is connected, whereas in a gram negative organism, I don't see any connection anywhere. So what is the point? There is a pentapeptide bridge between the NAM molecules. There is a pentapeptide bridge in a gram positive organism and there is no bridge in a gram negative organism. It's the easiest mnemonic that you can learn. P for P. What do I mean by that? That in a gram positive organism, a pentapeptide bridge is present, and in a gram negative organism, a pentapeptide bridge is not present. Have a look at this photo again. You'll say, yes ma'am, I can see the 5 pentapeptide here, but I don't see the pentapeptide here. So you are right, I can finally spot the difference. Pentapeptide bridge between the NAM molecules, P for P, positive organism, not seen in a negative organism. What other differences do you have? What other differences do we have between gram positive and gram negative? Another P coming up. Peptidoglycan layer. So which of them will have more peptidoglycan? A thicker peptidoglycan. You will say, ma'am, P for positive, P for peptidoglycan. So gram positive organisms have a very, very thick peptidoglycan and gram negative organisms have a very, very thin peptidoglycan. So you will say that, ma'am, okay, over here in gram positive, majority of it is P for P. It has a lot of peptidoglycan on it. But in gram negative, when the peptidoglycan is very, very thin, what other thing makes up for it? What makes the cell wall then? I'll say it is lipopolysaccharides. Lipid. So if I ask you, out of gram positive and gram negative, lipid content is where more in amount, where you say lipopolysaccharides are more in the gram negative ones. So lipid content will be more in the gram negative ones. Repeating, guys, now finally the take home message for you guys is that when you are told to differentiate between a gram positive and a gram negative, remember P for positive, which means P and P is what you are going to write. Which of them has a thicker peptidoglycan? Which of them has a thicker peptidoglycan? It is gram positive. Which of them has the pentapeptide bridge? The pentapeptide bridge is also present in the positive. So positive peptidoglycan and pentapeptide. That's the cell wall, main component, cell wall. Finally, after cell wall, you say, ma'am, this brown color thing is the cell membrane. And can you see? So I'll label it. This brown color thing that you have is the cell membrane. Can you all see the cell membrane has dipped inside over here? And the cell membrane has dipped inside over here. What is this dipping inside? This dipping inside is a mesosome, and that was the question that I had asked you. The cell membrane dips like this and the cell membrane dips like this. So can I say it is formed by the invagination of the cell membrane? Yes. Does it happen both in gram positive and in gram negative? Yes, but it is more in gram positive organisms. Mesosomes are prominent, again P for P. Mesosomes are prominent where? Mesosomes are prominent in the gram positive organisms. But what are mesosomes? होते क्या हैं? What is the purpose of these mesosomes? So please remember everything about mesosomes that you need to know. Remember, mesosomes are also known as chondroids. That's another name that has been given to them. Mesosomes are also known as chondroids. Number one question, they are the cell membrane invagination. They are the invagination of cell membrane. Number two, where are they seen more? They are prominent in positive than in gram negative. And last, what is their function? They function for the respiration. This is a respiratory organ of the bacteria. Respiratory function or respiratory organ of the bacteria is a mesosome. Other than this, there is no fourth question that can come. Cell membrane invagination, prominent in positives, and respiratory function, and hence the answer over here was both the statements are correct. What else am I left with in that diagram? I'm left with these three projections coming out called pili. Remember, pili are also known as fimbria, and their function is attachment. Whenever two bacteria want to attach, whenever there are two bacteria and those two bacteria want to attach, they can attach like this by the pili. So pili or fimbria are basically for attachment. Versus, what is this? Flagella. The next one is flagella. And when we say flagella, these wavy things coming out, I think everyone knows that flagella is used for locomotion. When the organism wants to move around, lot of movement, motility we'll be talking about in bacteriology. When the organism wants to move around, definitely locomotion is something which will be carried out by flagella. Okay, having said that, what is the next question that we have? Here you go. Mesophilic organism grows at what? So mesophilic organism is going to grow at what particular temperature? There are three main temperatures that we have. Want to learn those? We'll come back to the answer after that. Requirements for a bacteria. For a bacteria to grow, please remember, the temperature can either be a medium range temperature or it can be in minus. So they are like psychic temperatures, psychrophiles, psychic temperatures, minus temperatures, or they can be higher temperatures. So when I'm talking about medium range temperatures, 20 to 40, anything between 20 to 40, the medium range, the bacteria that grow in this medium range temperatures are mesophils. Meso will remind you of medium range temperatures. The organisms that grow at minus temperatures, they are psychic temperatures, psychrophiles. Psychrophiles grow at psychic temperatures of minus, and the organisms that grow at high temperatures, 55 to 80. For high temperature, we've used the word thermo. They are thermophiles. So easy to learn. Mesophile, medium temperature, 20 to 40. Psychrophile, psychic temperatures minus. And thermo means more temperature, so higher temperature, 55 to 80. So the PYQ that came was mesophilic organisms, medium. So it cannot be under the minus, it cannot be low temperatures, it cannot be high temperatures, it has to be somewhere medium between a 20 to 40. So that's the best answer that you have over here. Okay, having said and done with this question, let's move on to the next. The next question that we have, sporulation occurs in this particular phase of the bacterial growth curve. Repeating, which phase of the bacterial growth curve is going to show you sporulation? So I hope some of you definitely know the answer. For those who don't, you will know the answer in exactly 5 minutes, because what we are going to discuss is the bacterial growth curve. So what I'll do, I'll first draw the bacterial growth curve. What exactly are you trying to analyze in a bacterial growth curve? It has a flat line, then the line goes up, then again a flat line, and then the line goes down. So basically, when you're talking about the bacterial growth curve, this axis over here is going to basically tell you the number of the bacteria. This is going to tell you the number of the bacteria. This obviously will tell you the time and the duration. So first you will say that, ma'am, there's a flat line, means this is said to be a lag. This is said to be the lag phase. After that you will say rise in the number. So rise in the number means we are in a log phase. Log always means exponential, increase in number. Bacteria are multiplying. Then comes this, you'll say, ma'am, we've reached a plateau. This is a stationary phase. This line over here is the stationary phase, and finally dropping down is what phase? It is referred to as the decline phase. This is referred to as the decline phase. So repeating, we have something called the lag phase and the log phase and the stationary phase and the decline phase. So now let's do the characteristics of all. Let's start with the first one, this particular phase where everything is standing still, the lag phase. This is basically all of us in the lockdown. When all of us were in the lockdown, what were we doing? We were sitting, we were eating, we were just sitting at one place, no activity, no moving around. That's it. Just sit and eat and become fat. That is what we did in the lockdown. That is what the bacteria are also doing. Although we didn't actually, because most of us were on duties and working. But let's consider it as what most of the people did was sitting and eating and becoming fat at that time. Bacteria in the lag phase, in the lag is just like a lockdown bacteria. It's just going to increase in size. It will gather all its raw materials. It will eat a lot of food. It will make a lot of enzymes. It will make a lot of metabolites. It is going to become very fat. It's going to increase in size. Why is it doing so? I can say it's the silence before the storm. Because after this it knows I have to work a lot. After this it knows that I will have to work a lot, multiply a lot, so let me gather all my energy. Let me gather all my energy, let me adapt and gather enzymes, metabolites, so that I'm ready, all my raw material is ready. Then in the next phase I anyway have to work a lot. So if I ask you a question, that maximum size is at the end of a bacteria. Maximum size of a bacteria is at the end of which phase? Will know that okay, at this phase, at this junction, at the end of the lag phase, there is going to be maximum bacterial size that I will encounter. That is the lag. After lag, what do we have next? After lag, the next goes on to this. That is, there's going to be log. There's going to be the log phase. Means exponential. Bacteria has started dividing. There is maximum activity. All the bacteria are dividing. Means all of them are living. All the bacteria are dividing means all of them are living. All of them will take the same color. When all of them are living, all of them will take, like for example, you say, ma'am, I do gram stain. I'll say if all of them are positive, gram positive, all of them will take the color because they are all living. If they are gram negative, all of them will take the color because they are all living. So log basically tells you number increase. There's an increase in the number of the bacteria till a time there comes something known as stationary phase. Can you see? We've entered into a station out here. Why is this a station? Because now some cells have started dying. But we are not going down. If the cells would have been dying, we would have going down. I'm saying we've reached a plateau because right now the live cells and the dead cells are equal in number. That is why we are at a standstill, we are at a plateau, because the live cells and the dead cells are equal in number. That is why can I say I have now some living cells, some dead cells, so the kind of staining will be non-uniform. If you remember prior to this, in the log phase, all of them were living, so the staining was very, very uniform. Now I've got some living and some dead, equal in number, which means non-uniform staining will be there. What else? Please remember we write it as stationary phase. We write it as stationary because it's going to tell me that three things happen in this phase. What are the three things? Sporulation. Spore formation happens here. So S for spore formation in stationary phase. Toxin production by the bacteria happens here. T for toxin production. And if, and even bacteria can produce antibiotics. If antibiotic production has to happen, that will also happen in this particular phase. Repeating guys, if spore formation has to happen, toxin production has to happen, antibiotic production has to happen, all of it happens in the stationary phase, where the live and the dead cells are the equal in number. And finally, you will say, ma'am, the cell is dying. When all the cells have died, when all the bacterial cells have died, bacteria have died, when the dead cells are moved, the number will go down. So simple. Now if I get you to the question which I asked you, sporulation occurs in this phase of bacterial growth curve. S for sporulation, what's your answer? Stationary. Because if you remember, sporulation, toxin, antibiotic. Three things happen in the stationary phase. Let's do a last recap. Remember what happens in the lag phase. Lag phase is all about becoming fat. It's all about maximum size. Log phase, finally, the number will increase. Stationary phase, we've already done spore, toxin, antibiotic. Decline phase means everything has basically died. It is all the dead bacteria. That's a bacterial growth curve that they ask you in the exam, which is very, very important. Well, so I can say roughly half of the microbiology is done for us, and in terms of general microbiology. And now in the next session, we'll be taking up the second part of general microbiology, which will include two very important topics. Number one, sterilization and disinfection, and number two, there's going to be bacterial genetics. So two very important ones which I'll be covering up in the second part of the microbiome crash course. Thank you for joining in, guys. Hi everyone and welcome back to the YouTube session. Well, I'm Dr. Preeti Sharma and today we'll be discussing the second part of the microbiology crash course. This will be important for all the FMG as well as the NEETPG aspirants. Well, if you remember well and if you've not seen, I would request you to see the previous session also. In the previous session, we've taken the first part of general microbiology, under which we've covered some very important topics like scientists, microscopes, stains, bacterial anatomy, bacterial physiology. So all of that was the first part, and this comes across as the second part of the general microbiology. There are two very important topics that we'll be covering under this particular lesson. Number one, we'll be dealing with bacterial genetics, and the second one that we'll be talking about is sterilization and disinfection. So I think indeed both are very important topics for the upcoming exam. Well, let's start right away. Let's begin with bacterial genetics. When we talk about bacterial genetics, we have three things that come in our mind. I'll show you these three terms that we have to know: transduction, transformation, and conjugation. And the most important thing that you need to know is that this is a genetic transfer. So basically this is happening from one bacteria to another bacteria. So all that we need to know in these three situations is how the DNA or the genetics is transferred from one to another. So the very first out of these that we'll be considering is transduction. So who transfers material from this bacteria to this bacteria, and this is by the help of something known as a bacteriophage. I'm sure everyone has seen a bacteriophage which looks something like a tadpole. If you've seen a bacteriophage earlier in the textbooks, it looks something like a tadpole shaped structure. But what exactly is a bacteriophage? When we look at a bacteriophage, although it has the name bacterio under it, but guys, bacteriophage primarily refers to, it's a DNA virus. It belongs to a DNA virus. So can I indirectly say that ultimately it's a virus which is transferring material from one bacteria to another? It's a virus which is doing that. So let us see a particular situation. Let us see, this is something known as generalized transduction. What do I mean by that? So you can all see that this is a bacteria, right? And this is the DNA of the bacteria. I want all of you to note over here that bacterial DNA will always be circular like this. Bacterial DNA always is circular. So you can see the bacteria. I am calling it the donor cell. This is the donor bacteria, because from here something is going to go onto this bacteria, onto this bacterial cell. So that is known as the recipient bacteria. So we have a donor bacteria, we have a recipient bacteria, and you want that this DNA from here should somehow reach this DNA over here. You want the transfer to occur, and who's going to come to our rescue? That tadpole shaped bacteriophage. So you can see that the bacteriophage enters, first attaches, first it is going to attach to the donor cell. It's going to actually penetrate. Can you see this DNA entering? So now you'll say, ma'am, in this particular bacteria, you've got two types of DNA. One, you've got the DNA of the bacteria itself. So one, you've got the host DNA in this particular bacteria. And the second DNA that you've got is the DNA of a bacteriophage. Is the DNA of a bacteriophage. So these are the two DNAs that are present, and now more bacteriophages will form. Why so? Because I just told you bacteriophage is a kind of a DNA virus. So that is how a virus replicates. It first enters into a cell, then from the different kind of DNAs that are present, can you see more bacteriophages have formed? There are more DNA that has formed. Some of the bacteriophages have the blue color DNA, some of them have the red color DNA. Means some of the bacteriophages have their own DNA. Some of the bacteriophages have the DNA of the bacteria, the red color one. Have a look at this. This bacteriophage has taken up the DNA of the donor bacteria. It has taken up the DNA of the donor bacteria, and can you see it is now giving it to the other bacteria, to the recipient bacteria. So no one's going to make you write this down when it comes to an MCQ. What you have to know is primarily that this bacteriophage has picked up the DNA from the donor and has ultimately got it to the recipient, and you can see that in the recipient the fusion has occurred. The donor DNA plus the recipient DNA, repeating, the donor DNA that the bacteriophage got and the recipient DNA, which was the own DNA of this particular bacteria, both of them have recombined, and that is when I say that genetic material is transferable and is successful. So can I say in transduction, DNA goes from one bacteria to another, and who is taking that DNA? Bacteriophage, which is actually a virus. But this was just one type of transduction. I called it generalized transduction. There's another type of transduction which is a little special, a little different, and that is specialized transduction. Again, no one's going to make you draw this entire flowchart because luckily we have an MCQ based exam to give. However, what's the basic difference that you guys have to spot? So you will say, ma'am, it's the same bacteria, it's the same donor bacteria that you have over here. It's the same bacteriophage that you have over here. Bacteriophage DNA I can see over here. Donor DNA I can see over here. This is exactly what was happening earlier. Earlier also you could see bacteriophage DNA, you could see donor DNA, but they always remained separate. The bacteriophage DNA had different follow-up. The donor DNA had a different follow-up. Here both of these end up becoming combined. Both of these means the bacteriophage DNA and the donor DNA, first they combine and then they split. So first they combine and then they split, but splitting or breakups are always with a lot of memories, right? When they combine, well and good, but when they split, can you see the red color? The bacteriophage DNA has not come out alone. It has taken a part of the donor DNA also. A bit of the donor DNA has also been taken. Like I said, breakups are always with memories, right? So when the bacteriophage red color DNA split back, it took a little bit of memory from the donor, and now can you see finally the kind of bacteriophages that you have a little bit of self and little bit of donor.
and that is what it is going to go and give to the recipient DNA or the recipient bacteria. So what did you realize? You'll say, ma'am, there's something extra that has happened in this specialized transduction, and that extra is the fusion and the split up. The fusion and the split up has happened, and that is considered as extra over here. So that, guys, is referred to as a specialized transduction. So two types of transductions. Number one, generalized, where everything remains separate. Number two, specialized, where there's a fusion and split up that is involved between the bacteriophage DNA and the donor DNA. That's the first thing, that is known as transduction. Let's move on to the second one. There's something known as transformation. Now that's also equally important. When I say transformation, I'll draw one picture for you and it'll make it very simple. So when we are talking about transformation, we say that, for example, if this is a bacteria, let's consider this to be a bacteria, and this bacteria has its own DNA. So you'll say, ma'am, let's make a circular DNA like this. It has its own bacterial DNA. But now transformation, what transformation is going to occur? It's going to take up some DNA from the environment. Is there anyone getting the DNA? No. It's just going to take up free DNA is taken up from the environment. Free DNA is going to be taken up from the surroundings, or it has been taken up from the environment. That is what you call as transformation. Very, very important again for your exams. Transformation means what? Just picking up the DNA from the environment. That's it. So is there any bacteriophage? Is there any bacteria, a virus, anything involved? No. Just the free DNA which is there in the environment is kind of absorbed. So transformation done. What is the last one? And unfortunately the toughest one, that is conjugation. So let's study conjugation now, after which we'll get down to solving a few MCQs also. So what do we understand by conjugation over here, guys? Please remember, there is again going to be a transfer, but who is going to do that transfer this time? So again we'll draw it. This happens to be the donor bacteria and this happens to be the recipient bacteria. Now having said that, who is going to attach between both of them, or how is the transfer going to occur? I've made a bridge between both of them. Yesterday, if you remember, when we were studying the different types of the different types of transfers, and we were studying the bacterial anatomy, we definitely studied that what is involved in the attachment is always a pili. Do you remember this? In the previous session we studied that what is attached or what is involved in the attachment is always going to be the pili. So same way, if someone asks you what helps in transfer in conjugation, so in conjugation it's always going to be the pili that is involved. So here you have, first let's see the donor bacteria. We call the donor bacteria as a male, and here you have a recipient bacteria. We call the recipient bacteria as a female. So you will say, ma'am, this means donor bacteria has its own DNA, like every bacteria has DNA. Recipient bacteria also has its own DNA. So like every bacteria, donor DNA, male DNA, female DNA. But what am I transferring? I am transferring something different. I am transferring something known as F factor. What is F factor? F factor is a fertility factor. F factor is referred to as a fertility factor, which basically means that the male always has a fertility factor. I call it F plus. The male always has a fertility factor, and now I want that the fertility factor should also go over here. That is what I want. I want this female did not have a fertility factor. That is why this female was F negative. But now I want that the fertility factor should also go over here. Now there's a catch, guys. When I say the fertility factor from here, the male has gone to the female, I don't mean that the male will become negative. No, it's a copy. It's like it will duplicate. One copy will stay here, one copy will go there. So both of them will end up becoming positive. None of them will remain negative. So remember, this just replicates, copy paste. It's going to make a copy and the copy is going to go here. So one copy shall remain here, one copy shall remain there. So you will say that the male was always F positive, but the female will now become F positive. So what happens? That is what they ask you in the paper. They ask you that initially you had something called a male. The male was always F positive, and you had a female, and the female was F negative. Means the male had the fertility factor, the female did not have the fertility factor. But in the end, when this transfer occurs, can I say both of them end up having the fertility factor? Both of them become F plus F plus. I hope that's okay with everyone. Yes. So that's what they ask you in the paper. F plus and F minus, when they conjugate, both end up becoming F plus F plus. And repeating, what is F plus or what is F? F means fertility factor. Where is this fertility factor present? It is present on a circular structure called plasmid. So if this is the plasmid, here there is a fertility factor on top of it. So if I ask you, is plasmid a part of the main DNA? Or is it extra chromosomal? Think logically. Is it a part of the main DNA of the bacteria or is it extra chromosomal? I think you know the answer. It is going to be extra chromosomal. Okay, well, having said that, the first combination is done. And after, I'll show you a total of 3 combinations. After that, we'll do a recap also. Let's come to the second combination. In fact, I'll just give you a quick recap. One by one, we'll write. I have to tell you total 3 scenarios. In the first scenario, I'm saying that if this is a male bacteria and this is a female bacteria and both of them have their DNAs like they do. Now I'm saying fertility factor has to go from here to here. So initially the male was F plus and the female was F minus. But now both of them have become F plus and F plus. That's the first combination because this has gone from here to here. Now let's look at the second combination. Again, this is your bacteria. Everyone can see this is the DNA of the bacteria and this is the fertility factor of the bacteria. This has a fertility factor. This bacteria is an F plus. So you'll say, ma'am, this means this is a male bacteria. It has a fertility factor. Now you'll say just give it to the female bacteria. That is what you are expecting. But before this goes to the female, both of these end up making a pact. Both of these end up fusing. This is all happening in the donor only right now. In the same bacteria, the main DNA and the plasmid, repeating, the main DNA and the fertility factor, they have recombined. They have recombined. What do you call this kind of a cell? This kind of a cell is known as HFR. That is high frequency recombinant. It is referred to as high frequency recombinant. So when the main DNA and the fertility factor fuse, you call it high frequency recombinant. Now let's consider a scenario. Now let me consider that over here I have something known as an HFR kind of a cell. What is an HFR kind of a cell? It has the DNA of the host bacteria and the fertility factor which has fused. They are going to now conjugate via pilus with the F negative, means a female. See, she has her DNA, but she doesn't have a fertility factor. She's a female, she's F negative, which basically means that now the combination is going to happen between an HFR and an F negative female. Between an HFR and an F negative female. What will happen? You'll say, ma'am, a part of it, a part of both, a part of the host, a part of the fertility factor is going to go. Repeating, a part of this and a part of this will go to the female. But what am I saying again and again? Only a part of the fertility factor will go. Do you think if I give incomplete fertility factor, will this female end up getting the fertility factor? If incomplete things are coming to it, no. You will say, ma'am, in this situation, the female will still remain negative. She will still remain negative because you're not sending the complete fertility factor. You're sending a part of the fertility factor. So what did I analyze out of this? That this still remains HFR. In the end, this will still remain HFR and the female will still remain F negative. So when there is a recombination or when there is a conjugation between HFR and F negative, the male still remains HFR. The female will get a part of two things, but only a part. So she will still remain F negative. That's scenario number 2. So repeating, when there is a recombination or when there is a conjugation between an HFR and an F negative female, the HFR remains as it is and the female is also going to remain F negative. She does not receive the fertility factor. Coming to the third situation, I know by now it must be getting a little, a little stressful because too many things coming up, but we'll be revising in the end. Now let's start again from the beginning. You'll say this is the donor, this is the male. Why am I calling it a male? Because I can see the DNA and I can see the fertility factor. So this is an F plus male. Now, when both of them try to, in the same DNA, when both of them integrate, do you agree you've come up with an HFR kind of male, high frequency recombinant? Okay. Third scenario. After recombining, I've still not reached the other bacteria. Everything is happening in the same bacteria. The same male bacteria, they fused, they formed HFR. Now, after combining, they split up. Another breakup coming. In the same male bacteria, now the DNA is again separate and the fertility factor is again separate. But what did we learn? Breakups are always with memories. So when the fertility factor comes back this time, it carries a little bit of the DNA of the host also. So repeating, first there was DNA separate, fertility factor separate. When both of them combined, we got an HFR. Now both of them have broken up. So what you have, that cell is known as F prime cell. Now when they break up, and this carries a little bit of memories from the DNA, this is known as an F prime. This F dash is known as an F prime cell. Now let's consider this male is an F prime male and this conjugates with a female. Female we know is always F negative. So now I'm talking about F prime conjugation with F negative. You'll say the F prime totally goes here. So the male was anyway F prime and now the female will also become F prime. So when F prime and F negative conjugate, both of them end up becoming F prime. Let's repeat, when F prime and F negative are going to conjugate, when F prime and F negative are going to conjugate, both of them are going to become F prime, F prime. So let's do a recap. These are all understanding things. Now let's do how will I quickly solve the question in the exam. So please remember, whether you're dealing with simple F plus or you're dealing with F prime, when the male is F plus and the male is F prime, the end result is both of them becoming the same. So when the male was F plus, both male and female ended up becoming F plus. When the male is F prime, both the male and female end up becoming F prime. So there's no confusion over here. F plus and F prime, end result, both of them end up becoming that. The only problem is in HFR. HFR cannot make the female positive. HFR cannot make the female positive. In HFR scenario, the female will always remain negative. So remember, F plus will make the female F plus, F prime will make the female F prime, but HFR will not be able to do that. HFR will not do that, means the female will still remain F negative. That's your final summary of the conjugations that they can ask for you. But honestly speaking, there is no way that you can analyze this without solving questions. I personally feel solving questions is the best way of doing this. So let's start with question number 1 on this topic. DNA transfer in a bacteria via a phage is known as which of the following? Conjugation, transduction, transformation, or translation. So DNA transfer in a bacteria, that is what I told you, one bacteria, other bacteria, and who is going to do it? It's a bacteriophage that's going to do it. And this is the first thing that I taught you, that was transduction. This was transduction. I hope you gradually get into the mode of solving these questions. Okay, let's move on to question number 2. Transduction is defined as what? Bacterial medial viral recombination, viral mediated viral recombination, viral mediated bacterial recombination, or bacterial mediated bacterial recombination. So big time confusion they've created with the options, right? So when I talk about this, what all can you see over here? Okay, so I see that transduction, let me go back to my basic knowledge. Transduction is that this is a bacteria and this is a bacteria. So it's definitely bacteria to bacteria. It's nothing to do with virus to virus. It's bacterial recombination, bacteria to bacteria. Who carries it from bacteria to bacteria? Bacteriophage. And what is bacteriophage? Like I taught you, bacteriophage is a DNA virus. So virus is the mediator. Virus is the mediator, and that is why the answer to this question happens to be option C, because virus is mediating the bacterial recombination. Virus carries the DNA from here 2 here. So viral mediated bacterial recombination. Let's move on to question number 3. Let's read. The cell in which the F factor carries along with it some chromosomal genes. Read again. F factor carries along with it some chromosomal genes. For those who've got the answer right, very good. This is an F prime cell. What exactly were they telling you? I'll ask you again. This is a bacteria. This is a bacteria, and this is the DNA of the bacteria. Further, this is said to be the fertility factor of the bacteria. This is said to be the fertility factor. Can I call this as the F positive bacteria? I hope yes. The male bacteria, the F positive bacteria. So this is fine. This is F plus. Now when they combine, let's draw the second scenario. When they combine means this was the DNA and into it the plasmid has combined. If they've combined, what do I call it now? I call it a recombinant. I call it an HFR bacteria. Situation number 3. Now they've split up. When they've split up, what happens? You'll say, ma'am, we have the same bacteria. The DNA has gone separate. The plasmid has gone separate, but breakups are always with memories. Means it's carried a part of the DNA from the host. That is what they had asked you. The cell in which the fertility factor, this fertility factor, carries along with it some chromosomal genes. Carries along with it some chromosomal genes is known as what? That is what we studied. That is known as an F prime cell. I hope the questions are making sense now, because this is something which could be very important but confusing in the exam. Let's move forward. Practice another question. F factor integrates with the bacterial chromosome to form what? So let's go back. F factor integrating. They are saying F factor and bacterial chromosome integrate. What is the integrated version known as? Integrated version is known as HFR. Do I have HFR in the options? Definitely yes. So the integrated version is referred to as HFR. Let's move on to the next question. A bacterial cell in which the F plasmid is integrated with the chromosome. Have they just put up the same thing in a different language? Again, you are combining the F plasmid with the DNA, and I know that this situation is known as high frequency recombinant. Like I said, they can give you the same question in different languages. Let's practice one last question. Fertility factor possessing chromosomal genes is known as. Again, fertility factor possessing chromosomal genes, something that we just studied. Again, a recap. The fertility factor possessing a few chromosomal genes, what do we have it as? We call it the F prime. Perfect. So I think everyone knows the answer. The fertility factor with chromosomal genes, F prime factor. Well, having said and done with this, I think we can move forward, and now we go on to the next concept that you have. As we had decided, we are going to do 2 things under today's class and today's session. First one was bacterial genetics, which is done, and the second one is sterilization and disinfection. So let's get going, guys. When I talk about sterilization and disinfection, the first thing that I want to teach you is that there will be 2 different kind of methods that I will be talking about. So what are the methods that we have under sterilization, disinfection? This is going to be number 1, the physical methods like heating or filtration or some kind of radiation, physical methods. Second type of method is going to be a chemical method. Chemical means I can use alcohol, I can use formaldehyde, I can use some gaseous material. So either I will do sterilization, disinfection with physical methods, or I will be doing the same with chemical methods. And first and most important type of physical method that we have is going to be heat. But please note, this heat gets you, or heating gets you, maximum number of questions, because heating is just not heating with a candle or heating under the sun. Heating is of 2 types. They say heating can be dry heat or heating can be moist heat. So that is exactly what I'm starting with. Repeating, heating can come as dry heat sterilization and heating can come as moist heat sterilization. What do we have under dry heat? Definitely, like I told you, in earlier days, we used to use candles. Obviously, no one uses candles now. Earlier days, they used to keep the things under sunlight. No one does all of that. So these things are not coming to you. But these 2 machines are definitely used. Hot air oven and incineration plant. These are the two heating techniques that they are using in dry heat sterilization in every hospital. So first I'll show you what a hot air oven looks like. It looks like any other oven in your house that probably is used for baking. So it's a typical oven, and what all can you keep inside it? You can keep any kind of glassware, means you have a flask. You have a flask that you're using in your lab, or you have test tubes that you are using in your lab. So all kind of glasswares, syringes, cotton swabs. If you're using cotton swabs in your lab, you can also add them, and liquid paraffin. Remember, this thing is something which is important. Greasy materials, liquid paraffin, paraffin wax, right? Wax, if you guys have always seen candle wax, you've seen how wax is, it's very greasy. So any greasy material, liquid paraffin is sterilized using a hot air oven. So obviously you'll ask me that, ma'am, if you're calling it a hot air oven, you also must be having some heating temperatures that you have to know. So remember, 160 degrees for 2 hours is the usual protocol. 160-degree temperature for 2 hours is the usual protocol that we have. And remember, for everything we'll be having a control. When I say control, means, see, whenever you're working in a lab, you have to make sure that are you doing the right thing? Is this machine calibrated? Is this machine working well? Is the electricity source okay? Are you using any expired material? So any testing that you do in the lab, any machine, any test, you do a control, because control will tell you, it's like a check. It's like a check that, yeah, everything is working fine and what you're doing is the right method of doing it. So for the control in all of these sterilization techniques, they use bacteria, they use bacterial spores, because they feel that at 160 degrees for 2 hours, if this spore is killed, this means all your glassware is also sterilized. That is the concept. So how do you'll say, ma'am, how do you have this? How do you put it inside the hot air oven? This comes in the form of strips. There are strips that come which have the Bacillus subtilis spore on it. You just put this strip inside the hot air oven. Whenever you want to put your flask and glass tubes, you put them. Along with that, you place one tiny little strip here, because at the end the color will change. Different colors come. You don't have to learn the color change, but at the end, when the color will change, you'll say, yes, this means the Bacillus subtilis spores have been killed. And if they have been killed, this means my machine was working fine, and this means my glassware is also perfect and I can easily use it now. So for every technique, please remember, we will be having a particular check or a control. And over here, we have used a lot of things. Now, please note, we can use Bacillus subtilis. Bacillus subtilis is also known as Bacillus atrophius. They are cousin brothers. They belong to the same family. Either we can use Bacillus subtilis, Bacillus atrophius, or Clostridium tetani. So either of these three things will be mentioned in the paper, but please remember, if Bacillus and Clostridium both are mentioned, then in that case, please go in for the Bacillus species that are there. So that's the hot air oven. What other thing that you have in your hospital, if you ever have seen, you would be knowing. And if not, then guys, today onwards or whenever you visit your hospital, please do see that there's a huge incineration plant somewhere at the back of your hospital, which is very, very important. It has two big, huge chambers in the hospital, and this can always be used for incinerating all the hospital waste, the animal waste, the human body part waste. So all of these kind of hospital wastes, the human body part waste, at a high temperature of 1100-1200 degrees centigrade. So remember, incineration also uses dry heat. And what do I mean by dry heat? There are no fumes, there is no steam, pure simple heating. For hot air oven, it is 160 degrees for 2 hours. For incineration, it is 1200 degrees. So if I ask you this question, that which of the following is this done for? That is, incineration is done for human body parts, syringes, body fluids, or gloves. Please remember, this is an FMG PYQ previous year question. Human body parts and hospital waste, incineration, and I have already told you the temperature, 1200 degrees centigrade. Moving on to the next question. Let's have a look at this. Greasy material, dusting powder, paraffin wax. Repeating, greasy material, paraffin wax. How would you want to sterilize them? We just studied that we are going to put them in a hot air oven. What fits into hot air oven? Gamma radiation, sunlight, dry heat sterilization, or autoclave? I think everyone knows this is a case of dry heat sterilization. Let's pick up the next one. Method of choice for sterilization of liquid paraffin. Method of choice for sterilization of liquid paraffin is known as. I think we are clear. Liquid paraffin, yes, liquid paraffin means hot air oven, and any kind of dry sterilization, dry heat, hot air oven. Sorry. Let's move forward. Pick up another question. Hot air oven efficiency is best checked by which bacteria? Sterothermophilus, subtilis, tetani, or streptococcus? So the correct answer to the question, like I told you, that sometimes for hot air oven controls, we have three things: Bacillus subtilis, which is there over here, Bacillus atrophius, and Clostridium tetani. So when both the options are there, the best one that you'll select will be Bacillus subtilis. Okay, so having done with this question and having done with dry heat sterilization, we can now move on to the moist heat sterilization. What all do we have under moist heat? There are three things that we've written. That is the moist heat sterilization under 100 degrees, the moist heat sterilization at 100 degrees, or above 100 degrees. So what all do we have below, or what all do we have below 100 degrees or less than 100 degrees are two techniques called pasteurization as well as inspissation. Okay, so we learn it as inspissation and pasteurization are less than 100 degrees. IPL, and I'm sure the fever is on to everyone. IPL, that is inspissation, pasteurization, are going to be below or are less than 100 degrees. What do we have at 100 degrees? At 100 degrees, so if you're talking about IPL, you have to talk about the BAT. That is boiling and tyndallization are at 100 degrees, and above 100 degrees, when we are talking about above 100 degrees, A for A, we consider autoclave. Let's do a recap. IPL, inspissation and pasteurization less than 100 degrees. BAT, that is at 100 degrees, we have boiling and tyndallization, and above 100 degrees, we have autoclave. Out of these, let's study them one by one in detail. So let's start with the first category, and that is going to be the pasteurization of milk, the very, very famous. And we know that below 100 degree pasteurization, IPL, inspissation and pasteurization are less than 100 degrees. So pasteurization has two methods. One thing I know, that the temperature in both the methods is going to be less than 100. So there are two methods we use for pasteurization of milk. One is the Holder method and one is the flash method. The Holder method is 6330. What do we mean by that? 63 degrees for 30 minutes. You're going to hold it for 30 minutes. Repeating, 6330. Whereas the flash method, flash means seconds. This is 7220, means 72 degrees for 20 seconds, and only seconds, because this has to happen in a flash. So you increase the temperature, but the timing has become seconds. So 7220, and after increasing it to 72 degrees for 20 seconds, you drop it down to 13 degrees. That is what you do with milk. So let us do a quick recap, guys. What are the two methods that we studied up till now? We studied something called the Holder method, which is 6330, 63 degree for 30 minutes, and there's something called a flash method, 7220, 72 degree for 20 seconds, and drop it down to 13 degrees. Which of them is now considered better? The flash method is now considered better because it can even kill something that the Holder method cannot, and that is Coxiella. Coxiella can be killed by the flash method. So that was the first technique, pasteurization. If I now ask you a question based on it, pasteurization of milk is done at what? So you again, please read all the options, think of what all we had studied. We had studied two methods, Holder and flash. Holder was 6330, flash was 7220, 63 degrees 30 minutes, 72 degrees 20 seconds. So we have 63 degrees 30 minutes. The first option is only looking right. 63 for 20, no. 72 for 30, no. 72 for 20, but minutes, no. Flash method is 72 for 20 seconds. So certainly the first one is the correct answer over here. Pasteurization is done. Let's go on to the other technique. The other technique is known as inspisation. That is also below 100. Remember how much below 100? I call it 80-20, 80-20, 80-20. Why did I repeat it 3 times? Because we are going to, or you can even learn it as 80-30 for that matter, because 80 degrees is what I'll maintain for 30 minutes. So let's learn it as 80-30. But this is, why did I repeat it 3 times? Because this goes on for 3 days. For example, everyone has heard of two culture media. Have we all heard of LJ and LSS, the two L medias? What are the two L medias doing out here? LJ, Lowenstein Jensen. I'm hoping everyone knows that this is done for tuberculosis. And LSS, Loeffler's serum slope, is for Corynebacterium. Both of them we'll be studying under our bacteriology crash course, but these two medias, the L's and L's, they are sterilized using inspisation. So I'll take the media, I'll heat it to 80 degrees for 30 minutes on day 1, then 80 degrees 30 minutes day 2, 80 degrees 30 minutes day 3, and then the cycle is complete. So remember 80-30, 80-30, 80-30 is what you have to consider over here. That is what is inspisation. So I hope below 100 degrees done, so that we can come down to at 100 degree. What was the mnemonic? For at 100 degree you had to remember BAT, that is boiling. Just like you boil water, we boil water for approximately 15-20 minutes, right? So that we can kill the bacteria and the germs in it. So that's a simple boiling. I know boiling of water will always be at 100 degrees, but there's a problem that boiling does not kill the spores. So you say that I want a particular technique where I can kill the spores also, and that technique is tyndallization. I've learned tyndallization as T20 matches. Everything about IPL and batting and T20 happening here. So T20 means tyndallization is a 20-minute cycle, but again for 3 days. So 100 degrees the temperature, 20 minutes because T20, tyndallization 20. So 120 day 1, 100 degree 20 minute day 2, 100 degrees 20 minutes day 3. So that is what you have in tyndallization. Let's do a recap. Boiling is a single step for 15 minutes. Tyndallization is 3 days, 100 degrees, 20, 20, 20 minutes. Done with all of that, we are now coming to above 100 degree, which is autoclave. How much above 100 degree? 121 degree, 15, 15. What do I mean by that? Let's explain. So what do you have over here? Guys, this is the classical picture that you have for an autoclave. And remember, autoclave is 121 degree centigrade that you are going to heat for 15 minutes, and you are going to keep a 15 psi. What is psi? Pressure. So we keep a temperature of 121 degree, timing of 15 minutes, pressure of 15 psi. That is what you use for autoclave. However, please remember, so any kind of glassware you want to keep, again, you can keep flasks, you can keep the test tubes, all of these can be done in autoclave. A lot of other things, for example, even if, other than instruments, in the OT, you will always see that autoclave will be present in every operation theatre. Why? Because all the aprons that are used in an operation theatre, all the sutures, the stitches, the sutures that are used, they have to be autoclaved. You will see this machine in every microbiology lab because all the culture media have to be autoclaved. You will see this in every micro and pathology lab because sputum samples have to be autoclaved. Why do you think I'm autoclaving the sputum samples? See, for example, in our country, you have a case of TB. Okay, so what will they send to your lab? What sample will they send to your lab? They'll send a sputum sample. Okay, you'll look at a sputum sample, you'll tell them whether the patient has TB or not. After that, to throw the sputum sample, to discard the sputum sample, you can't discard so many microorganisms in the environment. Before you discard or throw the sputum sample, before discarding, you autoclave it so that all the organisms are killed. So remember, all instruments, aprons, sutures, media, sputum samples, they need to be autoclaved. What's the catch point? all sutures except catgut. Catgut is not, will lose its strength. Catgut loses its strength, loses its elasticity if you autoclave it at such a high temperature. So you will never put autoclave, you'll never put catgut suture in autoclave. And similarly, LJ media and LSS media, these two L medias, did I tell you you're not doing them for autoclave? What are you doing to sterilize them? We just studied that for sterilization we are using inspisation. Inspisation is for the two L medias, that is the LJ medias and the LSS medias. You don't use catgut over here, you don't put LJ LSS in here. These are two exceptions that you have to know for the autoclave. And what is the picture that I had shown you for the autoclave? Here you go, with which they ask you a question. What is the control that is used in an autoclave? Finally, the control you want to know, is this temperature even attained? Have I attained a 121 degrees centigrade? So please remember, there is a bacteria known as Bacillus sterothermophilus. The word thermophilus tells you that this has the ability to survive at very high temperatures, but at 121 it will get killed. So how will I get to know that 121 has been attained when this organism is killed? Bacillus sterothermophilus is the control that we use. The story is not over. You'll say, ma'am, the usual temperature that we have, we've understood 121 degree for 15 minutes, 15 psi. But in case you are dealing and you want to kill the prion proteins, for the prions, I'll increase the temperature to 134 degrees and the timing, just put a dot. Not 15 minutes, it's 1.5 hours. So remember, 134 degrees centigrade for 1.5 hours. That is what you have to do for prion killing. Increase the temperature, increase the timing, the pressure can remain the same. Okay, so having said all of this, now if I want to do a very quick recap of what all have I studied up till now, I've studied three things. I think IPL and VAT are very useful mnemonics. So inspisation and pasteurization below 100 degrees, boiling and tyndallization at 100 degrees, and autoclave above 100 degrees. Now, if I want to obviously judge myself, the best way is to solve a question. So how are prions destroyed? Prions are best destroyed. Look at all the things written over here. Autoclaving at 135, hot air oven at 160, H2O2, that is hydrogen peroxide or sodium hypochlorite. I think we've just studied autoclaving at approximately 134 degrees centigrade for 1.5 hours is going to do the purpose, solve the purpose for us. So that is how we go about with the prion proteins. Well, having said that, when I got you to start with sterilization disinfection, I clearly told you that there are physical and chemical methods, and under the physical methods, the heating method is done. What are we left with? Let's move on to the next one. We also have something called as the radiation method. Radiation. So basically by radiation, I mean I have something called ionizing radiation and non-ionizing radiation. That is also something that we have. So let's pick up the next one. Heating done, dry heat done, moist heat done. Let's pick up radiation. Ionizing versus non-ionizing. What's the difference? Please remember, ionizing radiation has something called X-rays and gamma rays. Repeating, ionizing radiation has X-rays and gamma rays. And do they increase? Have you ever heard that you went in for an X-ray and temperature has increased? No, right? So they will not increase the temperature. There is no increase in temperature. So can I call it a cold technique? Can I call it cold radiation? Whereas for non-ionizing radiation like ultraviolet rays, infrared rays, they increase the temperature. They increase the temperature, and that is why I call them as the hot sterilization. Repeating, cold sterilization is the ionizing radiation because X-rays and gamma rays don't increase the temperature. Non-ionizing radiation like UV rays and infrared rays, they increase the temperature, and we call it hot sterilization. Why and when do I use them? That's the next point that you all have to know. So please remember, number one, ionizing radiation Catgut sutures. Did I tell you that you were not doing catgut sutures in autoclave? That is what we studied, right? That when you are dealing with autoclave, you can put all the sutures in autoclave but not catgut, because at this temperature, catgut is really going to suffer at 121. You don't put catgut. How do you sterilize catgut? Using ionizing radiation, the cold method. You don't subject it to heat. Other than that, you can use this ionizing radiation also for pre-packed syringes and grafts. Nowadays, a lot of transplants and grafts are taking up. So for grafts, you will be using them. Non-ionizing radiation means the ultraviolet rays and the infrared rays. These you will be using for OT fumigation. Fumes in the OT, basically the radiation in the OT for the OT sterilization. And biosafety cabinet. What is a biosafety cabinet? All of you, whenever you visited a microbiology lab, and you must have seen that the microbiologist always sits in this small kind of a box or a chamber, because you know the microbiologist is dealing with a lot of infectious samples which have come from all sort of patients who could have any kind of disease. So many culture plates he or she is dealing with, so he or she has to be kept in a sterilized environment so that the infection does not spread all over. So that small chamber in which that microbiologist is sitting is known as a biosafety cabinet. How do I sterilize it? Usually it's an ultraviolet light that I'm using for it. If someone asks you that what is the control for radiation, what is the control that we have for radiation? Remember Bacillus pumilus. Please remember, the way you write R for radiation is the same way you write P. So what is the control for radiation? Which bacillus? It is Bacillus pumilus. If I ask you the question out here, cold sterilization is done by, remember I told you cold was what? Cold was that we were not going to increase the temperature. The temperature was going to remain the same. And if you remember, ionizing radiation, what all does ionizing radiation comprise of? X-rays and gamma rays. Ionizing radiation, X-rays and gamma rays. Look at the others. Steam. Steam is obviously from water. The temperature will increase. Other than that, look at infrared rays, ultraviolet rays. Obviously, with them also the temperature increases. With ionizing radiation, X-rays and gamma rays, the temperature does not increase. Moving on to the next one. After this, we come back to something known as filtration. So, easiest way, all of you have seen an aquaguard or all of you have seen a water purifier. You've seen how water has to be filtered. Any aquaguard or water purifier that you look at, it will always have filters, candle and filters inside it. Similarly, nowadays, so much of air pollution that has come up, the air quality has gone bad. So a lot that is being sold in the market are air purifiers. Yes, which is a big market which is now coming up. What do the air purifiers, what do they say in their marketing? What kind of filters do they have? If you ever heard or read that point carefully, they say we are using HEPA filters. Very rarely do they say we are using ALPA filters. So there are two kind of filters, HEPA and ALPA. They basically filter out the bacteria. Obviously, ALPA is the best one that we have in the market, but certainly the best will be the most expensive. So most of the air purifiers are having HEPA filters that are being used. Earlier, they used to use something called a seeds filter. Even for water filtration or any liquid they wanted to filter, they used to use seeds filtration, but this is not used now. It was found that this seeds filter contains asbestos. Seeds filter contains asbestos, and do we all know that asbestos is carcinogenic? It can cause cancers. For example, if I'm wanting to filter water, and I make that water pass through this seeds filter, it will get elements of asbestos in it, and when we consume that water, it's a source of cancer. It's carcinogenic. So we don't use the seeds filter. Now we use HEPA and ALPA. HEPA is most common, but ALPA is the best. What is the pore size of these filters? What is the, for example, if this is a filter, what is the pore size that we have? The pore size is 0.22 microns. The pore size is 0.22 microns. And what is the control? We'll obviously have a control. Such a small pore size, such a small filter, the bacteria has to be very, very small, diminuta. Brevendi monas diminuta is what we use as the control out here. So for filtration, it is Brevendi monas diminutor. Having said that, why don't we do something? Why don't we make a list of what all controls we've studied up till now? We've not finished, but whatever best we can recall up till now. So try to repeat with me. The first technique that we studied was hot air, or you can say dry heat. So hot air oven, if I'm asking you, basically you're talking about dry heat sterilization. So hot air oven had three things, if you remember. Number one was Bacillus subtilis. Apart from Bacillus subtilis, we could have the cousin brother, that is Bacillus atrophius, and apart from that, we could also have Clostridium tetani. Moving on, coming to the next one, we had autoclave. Autoclave is also having something at a very high temperature, so Bacillus sterothermophilus. We had to use a high degree or a high temperature bacteria, Bacillus sterothermophilus. Coming to the next one, and that is radiation. We studied that R and P looks the same, so for radiation it is Bacillus pumilus. The next that we studied was filtration, and for filtration we studied something has to be very small, so we said Brevendi monas diminuta. Brevendi monas diminuta for the filtration technique. I hope up till here everyone is still in sync with us and everyone is being able to identify all these things, so that we can now move on forward and study the next question. Glutaraldehyde, we've entered chemicals. Up till now, we spoke about heat, dry heat, moist heat, filters, radiations, all kind of physical methods. Now we've entered into a chemical, that is glutaraldehyde, is used for all of the following except. So what is the answer out here? It is used for bronchoscopes, proctoscopes, endoscopes. It is not used for thermometer. Remember, glutaraldehyde is for all the scopes. Laryngoscope, bronchoscope, proctoscope, anything, endoscopic tube, any kind of scope is something that I will put with glutaraldehyde. So basically, the first set of chemicals that I wish to teach you all are alcohols and aldehydes. Most important. Alcohol that is being used, isopropyl alcohol. Everyone has seen this as a concentration of 70 to 80%. What do you dip inside alcohol? Something that you never ever clean. For a doctor, the biggest source of infection that he carries back home or you carry back to your house or your hostel is a stethoscope. Before leaving the lab, one should always sterilize it, because this is something you'll wash your hands, you'll take all your precautions, you'll wear a different set of clothes, but something that you've forgotten and you're carrying along with you to your library and to your hostel is a stethoscope, which has a very high source of organisms. So isopropyl alcohol for stethoscopes and thermometers. What do you use aldehydes for? We have two types of aldehydes, formaldehyde and glutaraldehyde. Glutaraldehyde I just taught you, what you very famously know as the solution Sidex. Sidex, glutaraldehyde for all the scopes, bronchoscope, laryngoscope, endoscope, proctoscope, colonoscope, any kind of scope, glutaraldehyde. Coming to formaldehyde, you will say formaldehyde has two forms. One thing is a liquid formalin, which actually I think you use in pathology. Pathology specimens, you use liquid formalin. That is why when you enter a path lab, or even for that matter in anatomy, the cadavers, they are also kept in formalin. That is why your eyes get that burning sensation whenever you enter an anatomy dissection hall, or whenever you enter the pathology lab, your eyes start burning, also because you don't like those places, but also because there's a lot of formalin that is over there, right? Okay, so having said that, that's the liquid version of it, whereas there's a gaseous version of formalin that is known as formaldehyde gas, which you use for OT fumigation. So what all have we studied for OT fumigation? If you remember, just 2 minutes ago, for OT fumigation, we studied non-ionizing radiation, and now for OT fumigation, the chemical that we are studying is the formaldehyde gas. So coming back to the question, I hope this is okay. Glutaraldehyde is used for all except thermometer, because for thermometers and stethoscope, we prefer isopropyl alcohol 70 to 80%. Okay, what are the other types that we have? What are the other chemicals? We also have phenols and halogens. Phenols, I think everyone knows, the Lysol, which is the floor disinfectant, or Savlon or Dettol, and you put all your sharp instruments in this, all those OL, Crisols, Savlon, Dettol, Lysol, all of these, and sharp instruments, cleaning the floors, all of these are phenols. Next one, next chemical, halogens. Halogens like iodine. I think everyone knows tincture iodine and betadine. You've seen that brownish color solution. So tincture iodine, betadine, chloride. Chloride is also a halogen, and our famous most, sodium hypochlorite 1%. So this is a question that you get in the paper. The most potent disinfectant. This is a PYQ again of the FMG and the NEETPG, also once asked in the AIMS exam. The most potent disinfectant out of all: alcohol, glutaraldehyde, the halogens, iodine or hypochlorite, sodium hypochlorite, and please remember 1%. So why am I emphasizing on 1%? Because earlier recommendations used to say that sodium hypochlorite we have to use at 10%, but that is done. No longer. We use sodium hypochlorite as 1%, and especially, what's the other question that you get? You are an intern and by mistake you spilled the blood on the floor. You are an intern, you are collecting blood from a patient, you're taking a blood sample. By mistake, your hand moves and the blood spills on the floor. What chemical are you going to use? 1% sodium hypochlorite. Okay, talking about the last chemical that we have, and that is going to be, we are talking all in chemicals, we've only been talking about things like liquids. For example, we are talking about formalin, glutaraldehyde, Lysol, or betadine, or liquids, alcohols, or liquids. But remember, even gas can cause sterilization. And what do you have to know out here? That when we are dealing with gaseous material, we are talking about which gas? Ethylene oxide. ETO, it might be written in the exam as ETO. Remember, ETO is ethylene oxide, and never ever use ethylene oxide as 100%. Never. You have to dilute it. You have to mix it with other gases like nitrogen and all. You use it. Now the latest recommendations say, earlier they used to say use it as a 10%. The latest recommendations say 3%. So remember, 3% ethylene oxide is something that we use. Why don't we use it as 100%? Because it is inflammable, it will catch fire. And number two, it is also said to be carcinogenic. If you use it as 100%, it will be carcinogenic as well as it will be inflammable. I don't want to take either of the risks. I use it as a 3%. Where do I use it? Cardiopulmonary machines. For the sterilization of cardiopulmonary machines, we use ethylene oxide. If someone asks you what is the control, remember G for G. For gas sterilization, the control is Bacillus globigi. So I think we made a list sometime back, and I finally can finish that list for all of you. The list of the different controls that we did. Let's add on to it. So if someone asks you what is the control for gas sterilization, you will say G for G, and that is going to be Bacillus globigi. One of the control questions is always expected. It's an all-time favorite. In fact, this has come in your previous years also. Bacillus globigi is used as a control for, I don't think this is going to be a problem. Bacillus globigi is used for the control of gas sterilization. Which gas am I talking about? Ethylene oxide. Let me take a test of other options also. What do we use for autoclave? High temperature. I hope you remember, we use Bacillus stereothermophilus. Coming to the next, what do we use for hot air oven? For hot air oven, we use Bacillus subtilis, or we have other things like Atrophius, or we have other things like Clostridium tetani. Then one more thing is written over here which we've not studied, and that is known as plasma sterilization. Although it's not very important for the FMG exam, I'll give you an overview. See, what are the three states of matter that we always study, taking you back into school time? You'll say we always study solid, liquid, gas. These are the three states of matter. But do you know the fourth state of matter? The fourth state of matter is plasma. So that is what they're saying. See, we've understood solids, we've used filters, we've used liquids like alcohol, glutaraldehyde, betadine, so many liquids we've used. We've also used gases like ethylene oxide, Finally, we say we use the fourth state of matter, that is we convert H2O2. Using H2O2 we make plasma, and plasma is used for sterilization. Please remember, for plasma also the control is Bacillus stereothermophilus. Reminding you, plasma is another state of matter. You are making it out of H2O2, and this also uses a similar control, you can say, as that for autoclave, and that is Bacillus stereothermophilus. So I'll add it over here. Autoclave number 1 and plasma sterilization, both of them are going to utilize Bacillus stereothermophilus. Now I can say that all your important control tables are done. All said and done, what's the last topic that you have to know? The last topic that you have to know is you are now on the other side. Means you have to do testing. You have to do testing and you have to tell me. So for example, I am opening a small little setup of my clinic of my own, and I want to find out that what is the best sterilization disinfection technique that I should have at my clinic. So I call you because you guys are experts. So I call you and I say I am sending a solution to you guys. This solution is a solution named X. You don't know whether this is an alcohol, whether this is iodine, whether this is hypochlorite. I say I have a solution in my lab, in my clinic. I want you to tell me that am I using the right thing for my sterilization and disinfection, or is this potent enough, is this efficient enough to sterilize my clinic? So you will say that there are 4 tests. I will put your chemical X through 4 steps. And what are those 4 tests that they ask you in the paper? Their names are little difficult, so be patient. The first one is phenol coefficient test or the Riedel-Walker test. Repeating, phenol coefficient test or the Riedel-Walker test. What do? The solution X that you have sent, they are going to compare this solution with phenol. They say that we know that phenol is a very very good disinfectant. So is your solution X matching with phenol? If it is matching with phenol, this means you can use it. If it is not matching with phenol, means you cannot use it. So phenol coefficient test or Riedel-Walker test is comparing your solution X with phenol and telling you. But do you see a little bit of a catch? You'll say, ma'am, you've only compared it with phenol. Phenol isn't the best disinfectant that you have, right? But the only thing you've compared it with is phenol. So maybe you've missed out on so many things. Have you been able to find out that whether this X will be able to kill fungus, whether this will be able to kill the organisms in the soil? Have you been able to find out that whether this will kill organic matter or not? No, you just compared it with phenol and done. So there is a drawback. That is why you come to testing number 2, which I call as the modified Riedel-Walker test. You're doing the same thing. See, the previous one was only Riedel-Walker test. Now you're doing a modified Riedel-Walker test, which means that again the phenol component is there, but you will also test it in the presence of organic matter like soil. Can it disinfect soil? Can it kill the yeast? Can it kill the fungus, the spores? So that is now known as the modified Riedel-Walker test or Chick-Martin test. Chick-Martin test is a modern name for the Riedel-Walker test. 2 tests are done. After this, you come to the next one. There are 2 questions that I want to ask you. Number 1, I want to ask you, how many times can I use this solution X? I've used it once today for cleaning my floor, or I've used it once today for I've dipped some instruments into it. And now if I want to use the same thing tomorrow, can I do it or not? So how will you tell me that, can it be reused? Who tells me about the reuse? The capacity. Can it be used repeatedly? Can it be used repeatedly? That is known as the capacity test. To test the capacity and the reusability, we have the Kelsey-Sykes test. We have the Kelsey-Sykes test. And finally, you will tell me that, ma'am, I have done all the tests at my level. Now you finally start using it in your clinic, and then you tell me whether you're getting good results or not. So that is known as the in-use test, known as the Kelsey and Maurer test. So repeating, these are confusing. One is known as Kelsey-Sykes and one is known as Kelsey-Maurer. So Kelsey is common. You'll have to learn what is Sykes and what is Maurer. So Sykes has the K sound to it. Sykes has the K sound to it. So this tells you about the capacity. Whereas Kelsey-Maurer has the U word in it. So this tells you about the in usability. So let's make a, I think best is that if we make a table out of it, we'll be very confident. So how do I do the testing of a disinfectant? You will say that, ma'am, there are total of 4 tests that you are going to do. The first test is labeled as the Riedel-Walker test. Riedel-Walker test is comparing it with what? Riedel-Walker test is basically comparing it with phenol. It is a phenol coefficient test that you do, but it's a disadvantage because you're only doing it with phenol. Second, you say, let's do a modification. Let us do modified Riedel-Walker test, and that has been given a very modern name, and that is known as the Chick-Martin test. That is referred to as the Chick-Martin test. Then you say that I want to know the capacity of the disinfectant. I want to know how many times can it be reused. Second, and after this, you want to know how many or how can I finally reuse it, or can I use it in my lab or not, practically, the in-usability. So for capacity test and for in-use test, the name starts with the same. These are Kelsey tests, but for capacity, K sound. That is, this is known as the Kelsey-Sykes test, and Morer's use U word. So this is going to be the Kelsey Morer test. Finally, we've got the 4 tests, one for phenol, one for modification, one for capacity, and one for in-use ability. That is what finishes off this chapter also for you. It finishes off both the things, that is bacterial genetics as well as sterilization and disinfection, and we can conveniently wrap up this session. So I hope you guys have been benefiting from the previous one also. In case you've missed out on the previous lesson, please go and view the general microbiology part. This is part 2 of it, and going forward, the part 3 and 4 will be having mycology, that is fungus. So do watch out for that as well. I hope you guys are able to grasp it, because microbio is not an easy subject, undoubtedly. But with few mnemonics, like I say here and there, we are always able to solve anything that's difficult. Okay, so thank you so much guys for joining in, and see you in the next session on the same YouTube channel. Thanks a lot. Study well. All the very best. Hi everyone, welcome back to the YouTube session of the Microbiology Crash Course. I'm Dr. Preeti Sharma, and today we'll be discussing in this session about mycology. Mycology meaning we'll be talking about fungus and the different types and etiologies that are important for the exam. We'll also be taking up all the images that are important for you alongside MCQs that have been asked in the previous years. So let's start right away. Topic of the day, the much famous, and especially after COVID, I think something that has taken up a lot of importance, fungus, that is mycology. Let's start right away with a question. Culture morphology is best studied by, and this happens to be a previous year question. So culture morphology is best studied by gram stain, lactophenol cotton blue, calcoflower white, and methenamine silver stains. So culture morphology, and the answer to this question is lactophenol cotton blue. So let's know what are the different stains that have been mentioned over here. I've got you an entire collage of all the different stains that could come up in the exam when it is with regard to the fungus. So the first one that you see over here, where all the fungal hyphae have become black in color, this is known as the GMS stain. What does GMS stand for? It stands for Gomori methenamine silver stain. Repeating, Gomori methenamine silver stain. Now there's a little bit of a trick that I always tell the students, that whenever there's a stain that has the word silver in it, like over here, please see Gomori methenamine silver. So whenever there is the word silver in a stain, means you're somewhere dealing with a blackish color, because silver will turn black. So you can see the fungus has become black in this case. Okay, so this is the first one. Let's move on to the next one, in which the fungal hyphae have become pink. So over here, this particular stain is referred to as the PAS stain. So fungal hyphae are PAS positive also. Coming to the third one, you see the background is very, very dark and something is becoming fluorescent. So this is referred to as the calcoflower white. Now, if you guys have seen the previous sessions and the previous lectures of the microbiology crash course, all of you already know that what kind of a stain is calcoflower white. We've done a mnemonic for it. That flower will basically be for fluorescent. So calcoflower white is going to give us a fluorescent, or it's going to be a fluorescent dye that will be there. Moving on to the fourth one, I can see a culture plate. So what is the culture medium on which the fungal samples are grown? So the fungal media are Sabauroutes dextrose agar, SDA, that is Sabauroutes dextrose agar. And if I've grown it, see, I've seen a whitish color, cottony whitish color colony over here. From this, if I want to grow it, take a little bit of a specimen, I want to see it under the microscope. So what am I trying to do? I am trying to look at the culture morphology. I'm trying to look at the culture and what morphology does it have on the microscope. So what color stain have I used? I've used a blue color stain, and that is referred to as lactophenol cotton blue, and that is exactly what I had asked you. Let's repeat the question, that if you want to study culture morphology, you will use LPCB, that is lactophenol cotton blue. Let's move on to the next one. I have two more stains in front, and both of these, the organism in both of them is nothing but Cryptococcus. I'm sure everyone has heard about Cryptococcus, and it's very famous for causing meningitis. So cryptococcal meningitis is definitely a very important question in the exam. Please remember, now when you're dealing with the case of Cryptococcus, what kind of samples do you think you're going to get? Number one, you might be dealing with a patient with cryptococcal meningitis, so they'll take out the CSF, cerebrospinal fluid. They'll do a lumbar puncture. They'll send you the CSF sample. That's one possibility. Second, person dealing with cryptococcal meningitis might die, or there might be an autopsy case. So you might get a biopsy tissue. You might get a brain biopsy. These are the two samples possible. Either in a living person, you could get a CSF sample, or in a case of death and autopsy, you could get a biopsy specimen. And in both the cases, the stain that I'll use for cryptococcus will be different. Now, this stain I've shown you, where the background gets a color and the organisms are unstained. I think everyone knows this is the very famous India ink stain. India ink basically is a kind of a negative stain. It's a kind of a negative stain. So if they ask you that you have a specimen of CSF and what kind of staining would you do? You would do India ink negative staining. Similarly, if you're dealing with a biopsy specimen of Cryptococcus, can you see this time the Cryptococcus has become rose pink in color, pinkish in color, because the stain that you've used is mucicarmine. The stain used is mucicarmine. So finally, these are the two things that I need to remember when I'm dealing with cryptococcal meningitis. Repeating, for CSF, it is India ink. For biopsy, it is going to be mucicarmine. So now I think everyone is clear. Let's do a very quick recap. Unlabeled recap. So the black color one becomes Gomori methenamine silver stain, pink color one becomes PAS, fluorescent one becomes calcoflower white, the culture media Sabauroutes dextrose agar, culture morphology lactophenol cotton blue, CSF sample for cryptococcus India ink, and we have the biopsy sample for cryptococcus. We will be having mucicarmine. So all said and done, let's attempt a question. The capsule of Cryptococcus in tissues is best seen by, and they've given you a picture, gram stain, India ink, mucicarmine or methenamine silver. So capsule of Cryptococcus, see, simple. When it comes to Cryptococcus, I know that either I can get a CSF sample or I can get a biopsy sample. If I get a CSF sample, I will be staining with the India ink. If I get a biopsy sample, I will be staining it with mucicarmine. Here they've told me biopsy sample. They've given me the picture also of mucicarmine. So the answer over here becomes the mucicarmine stain. If I change the question, the capsule of Cryptococcus in CSF, and they've given me the picture, yes, that is going to be the India ink negative staining. So these are stains, but now that when you're talking about Cryptococcus, why don't you tell me that finally, what is the study that helps us? So this is not for answering, this is for learning. Please remember that for Cryptococcus, we do LAT. Latex agglutination test is a very famous test that is performed for the final diagnosis of Cryptococcus. Repeating, latex agglutination test for Cryptococcus. Well, having done with all of these main stains, let's move ahead with the story and go on to this. Which of the following is not dimorphic? First, I want all of you to attempt it, and then definitely we can take up the discussion of this. Which of the following is not dimorphic? So you've got a couple of organisms over here, and I hope many of you have got this right. The answer to this is Cryptococcus neoformans is not dimorphic. So first, what do we understand by dimorphic, or what is the meaning of the word dimorphic fungus? Remember, the meaning of the word di means two, and morphic means morphology. It has two forms. It basically exists in two forms, and these two forms are yeast and mold. So it has a yeast form which will be very tiny yeast form, and mold form will look something like this. They will look like hyphae. You can also call the mold form as the hyphae form. So repeating, we have two forms that a fungus can have, the tiny yeast and the hyphal form, that is mold. Remember, the yeast form will be seen at a 37 degrees and the mold form will be seen at a 25 degrees. And how do we learn it? There's a very simple mnemonic. See, what does yeast rhyme with? Yeast rhymes with heat. And what does mold rhyme with? Mold tends to rhyme with cold. So remember, which of them is at a higher temperature and which of them is at a lower temperature? At a higher temperature, yeast will be at heat 37 degrees and mold will be at cold, that is a 25 degrees centigrade. So which are the organisms which can show this kind of a feature? That is, they can show the yeast form also, they can show the mold form also depending on changes in temperature. This is the mnemonic that you have to know. The examples of dimorphic fungus are, the mnemonic says, body heat probably changes shape. So you'll always remember that dimorphic was all to do with heat and cold. So we have body heat probably changes shape. What is with B? B for blastomyces, H for histoplasma, P for two things. We have Penicillium marnefi and Paracoccidioidomycosis, C for coccidioidomycosis, and S for Sporothrix schenki. Let's repeat. Body heat, B for blastomyces. Next we have body heat, right? So H will tell me that I'm dealing with histoplasma. Next, P has two things, Penicillium marnefi and Paracoccidioidomycosis. C has coccidioidomycosis and S has Sporothrix schenki. Coming back to the question, which of the one is not dimorphic? So what is the mnemonic? Body heat probably changes shape. So blastomyces is dimorphic, not there in the options. Histoplasma is dimorphic, it's there in the options. Paracoccidioidomycosis, not there in the options. Penicillium marnefi is also known as Talaromyces marnefi. Coccidioidomycosis, not there in the options. Sporothrix schenki, there in the options. So which of them is not dimorphic? Clearly the Cryptococcus neoformans is not dimorphic. I hope everyone is okay with this. Let's move forward. Done with all the dimorphic fungus, and now let me show you all of those that I have just written out to you. So right now I told you body heat probably changes shape. These are all the pictures: blastomyces, histoplasma, paracoccidio, penicillium, coccidio, sporothrix. All of them that we are going to study, but obviously one by one. So let's start. Let's start with the first one. Body heat probably changes shape means B for blastomycosis. B for blastomycosis, also known as the North American blastomycosis, the Gilchrist disease and the Chicago disease. One more thing guys, all of them are going to have a lot of names and most of the names will be named after a continent and a part of the world. So we obviously have to learn that, but something that is going to go common in all of them is systemic involvement. Dimorphic fungi are very famous for systemic involvement, means organs will be affected, and one of the most common organ affected are the lungs. Lung involvement is most commonly seen. So coming to the first one, that is blastomycosis, known as North American blastomycosis, North America, so also having the Gilchrist and the Chicago disease. How do we identify it? B for blastomycosis and B looks like an 8. So here what you see over here is a figure of 8 appearance. You see the classical figure of 8 appearance. Why do we see this figure of 8 appearance? This, for example, is the organism. This, for example, is the organism. Now it's budding. Now it's going to show budding. And when it will show budding, the base of the bud is very, very broad. It's a broad base. See, I'll show you the difference. If this is an organism and this kind of a bud arises, you'll say, ma'am, it's arising from a tip. It's a very narrow budding that is happening. That is not happening here. Here you can see a proper 8 is forming. The base is very, very broad. The base is very broad over here. So I can say B for blastomycosis, B for broad based budding. There is broad based budding that is noted, and that gives a figure of 8 appearance. Why is it black in color? Now you all know the stains, right? Why is the color black? Because what color stain have we used? We've used Gomori methenamine silver stain. Repeating, North American blastomycosis, Gilchrist or Chicago disease, figure of 8 appearance, B and 8 and broad-based budding all sound the same. Body heat. So heat, next one that we come to, histoplasma. Now before I start with histoplasmosis, let me tell you a trick over here. Just a small thing that you have to remember for the exam, that everything that you will have for histoplasma, everything that I'll tell you for histoplasma will be very, very similar to tuberculosis. Means what? When I'm dealing with the case of histoplasmosis, what do you think is going to be the most common organ affected? Lung involvement is very common, just like tuberculosis. Number two, what does it cause in the lungs? It causes caseating granuloma. It causes caseous necrosis. Just like tuberculosis, it causes caseous necrosis. Number three, can it result in a granuloma just like TB? Yes, it can. So patient will come to you with similar kind of complaints, that is cough and sputum and caseating granuloma, lung involvement. So very commonly histoplasma looks like TB. Then I also know that histoplasma is also known as what? It is again what part of the world it is common in? It's common in Ohio. So it's known as the Ohio's disease, or also known as the Darling's disease. Remember, Ohio's disease or Darling's disease is histoplasmosis. I know it's a dimorphic fungus, means yeast, heat, mold, cold, right? Do we remember that mnemonic? It has a yeast at heat, means at 37 degrees, and it has a mold form, mold cold, which means it has a mold form at 25 degrees. Let me see both the pictures. The yeast form is very, very tiny. When I say very tiny, I mean that it's only 2 to 4 microns. You can definitely see. First, I'll try to enlarge whatever is shown over here. See, this is one cell. What cell is this? This is the macrophage. This happens to be the macrophage. So I'll draw the nucleus of a macrophage over here. And all of us know that macrophage is something which is very, very hungry, forever hungry. It's going to be a foodie kind of a cell. So it will always eat up the histoplasma. You will see multiple dot-like structures inside it. So let's see this over here. You see a huge cell over here. This huge cell is nothing but a macrophage. You see the nucleus of a macrophage over here. And apart from that, can you all see those dot 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 organisms? Very tiny. And when I say very tiny, they are only 2 to 4 microns. The size of a yeast is only 2 to 4 microns. That's why it looks so tiny. Coming to the mold form. So first and foremost, what stain is this? You've used it from a culture, lactophenol cotton blue. And you'll say that over here, what we can certainly see, if I can take this picture, I probably would want to show it to you as an enlarged version. If I enlarge this photo further, you'll say that, ma'am, you're seeing some round structures and you see some conidia or spores coming out of it. This exactly same picture came in the previous year papers. So we can see that there is a conidia or spore coming out of it. So I can basically say that these are known as, what name have I given to them? The authors call them tuberculate macroconidia. The authors call it tuberculate macroconidia. So yes, there will be tiny ones also. There will be microconidia also. But these ones that you're seeing out here are the macroconidia. So do we have, for histoplasma, did I tell you that everything will be again sounding like TB? Like I said, the lung involvement, the necrosis, the granuloma. Can I say it has the tuberculate macroconidia that makes it again similar to TB? I hope that part is clear. So well, having said that, that's the summary of histoplasmosis. So remember your mnemonic, dimorphic fungus, body, heat, Probably stands for the number third organism called Paracoccidioidomycosis, which is known as South American blastomycosis. By now, I'm sure a little bit of confusion has already started. So why don't we start making a simultaneous table? I think if we start doing that, we'll be in a more comfortable position at the end. So let's make a table of all the names and all the diseases that we have to know. Let's make a common table, right? Okay, so the first one, we'll go in the order, body heat. So first one we are dealing with is blastomycosis. What is the other name that we have for blastomycosis? This is known as North American blastomycosis. This was North American blastomycosis, which also obviously goes by Chicago's disease and is also known as the Gilchrist disease. Moving on, body heat. So the second one that we had was histoplasmosis. Again, histoplasma is very common in Ohio region, so that is why they also call it the Ohio disease, and it is also referred to as something known as Darling's disease. This is a very famous previous year question regarding Darling's disease. Coming to the third one, that is paracoccidioidomycosis. That is where the confusion starts, guys. Paracoccidioidomycosis is known as South American blastomycosis. Now, in the exam, don't get confused which one is North American and which one is South American. So, obviously, first you will say B and P. These are the two confusing things. So, North American, B comes first. So we usually say north is above and south is below. So in the alphabetical chain, B is above and P is below. So if I say this is north and this is south, you will say B comes first, P comes later. So blastomycosis is North American blastomycosis. Paracoccidioidomycosis is the South American blastomycosis. And what does paracoccidioidomycosis have? It has the very famous two things. M&M means it has the Mickey Mouse appearance. Number one, it carries the Mickey Mouse appearance. You can very well see in the tissue, it has these spores which are looking like the ears of a Mickey Mouse. So number one, M for Mickey Mouse appearance. Number two, M for mariner wheel. So the wheel of a ship or the wheel of a pilot. So you can call it a pilot wheel or a mariner wheel appearance. And why is it black? Because we know that when I get a black color, the stain that I've used is the Gomori methenamine silver. So repeating, paracoccidioidomycosis, number one shows us the Mickey Mouse appearance, and number two shows us the pilot wheel or the mariner's wheel appearance. So body heat, probably. Remember with P there was one more thing, and that was Penicillium marnifi, which I told you is also known as the Talaromyces marnifi. So that's the other name. It is also known as Talaromyces marnifi, and the question that you get in this case is the natural reservoir. What is the natural reservoir of Penicillium marnifi or Talaromyces marnifi? Are going to be the bamboo rats. They are going to be the bamboo rats. So now let's see, what does this picture show us? So again, why is it blue in color? Because I know that whenever I'm dealing with culture morphology, we are dealing with a stain called lactophenol cotton blue. And can we see that the conidia have given an appearance of a brush? We call it a broom or a brush appearance. We call it a broom or a brush appearance. So please remember, Penicillium marnifi, we are going to have a broom brush appearance. So repeating, it has a broom brush appearance with a bamboo rat as a reservoir. Broom brush with bamboo rat. So with a broom, you are going to try and make the rat run away. That is how you learn it. So broom brush appearance with bamboo rat, natural reservoir is Penicillium marnifi. Where have we reached? Body heat, probably. Two P's done, changes shape. So C for coccidioidomycosis. I'll give you a very good way of learning coccidioidomycosis. I call it ABCD. What is ABCD in coccidioidomycosis? Number one, let's start with the D. What are the other names? Again, back to the names. This is known as desert rheumatism. So desert will mean they will give you some kind of history of a cave or a desert or a valley. So they also call it valley fever, or the Arizona desert is a very common history given in this case. So please remember some history that will be roaming around, either a desert or a valley, these kind of history. So it is known as desert rheumatism. rheumatism. When I say rheumatism, I classically understand that there is a significant involvement of joints that will occur. When I say there is valley fever, I understand that the patient is also going to have evidence of fever over here. So joint involvement, fever, desert and valley history, so D for all of that. C is going to tell me coccidioidomycosis, and what is B and A going to tell you? Barrel-shaped arthrospores. When you look at it under the culture, again blue color, lactophenol cotton blue, can you see these kind of boxes? These kind of boxes are known as barrel-shaped arthrospores. It's a very famous question. So repeating for all of you once again, what is ABCD? D means it is desert rheumatism or valley fever, C for coccidioidomycosis, and B and A for barrel-shaped arthrospores. So body heat probably changes shape, and coming to the last one, that is sporotrichosis. We learn this and after this we go in for questions. So what is sporotrichosis? Why do I call it sporotrichosis? Because what is the name of the organism that we have? The name of the organism is Sporothrix schenki, and this is also known as the rose gardener's disease. Now I'm going to tell you the story about a rose gardener. Everything in sporotrichosis will be revolving around the word rose, and we'll also have the history of a gardener. So whenever they'll want to give you a history of sporotrichosis, they'll always say there's a gardener, he's working in the rose gardens, and he's had a thorn prick. The thorn from the rose has basically pricked his finger or his feet. So there is a thorn prick history that is always given characteristically while roaming around in the garden. Okay, so when it will hit him on his finger or on his toes or on his feet or hand, basically I'm talking about cutaneous involvement. The cutaneous lesions will develop. I'll show you a picture. Have a look at this. Can you see the cutaneous lesion here, then here, then here, then here? There are multiple cutaneous lesions that are going to be there in sporotrichosis or rose gardener's disease. But why is it a thorn prick might have happened at one point, right? Maybe the thorn prick happened at this point. But then why are so many lesions seen? Because it travels along the lymphatic channels. Please remember, the organism is going to show us a traveling kind of a history along the lymphatic channels. That is why I say that it is a kind of a lymphocutaneous disorder. Coming further, now let's look at the microscopy part of it. Microscopically, it shows you cigar body and asteroid body. What are these? Have a look at this photo, guys, in front of you. I think without a doubt you can identify that this looks like a star. So first I'll want to highlight this photo and tell you what exactly is seen. In the center you see a round structure. In the center over here you see a round structure. This round structure is referred to as a cigar body. It's nothing but the round yeast. It's the yeast form of the fungus. So remember, this is the cigar body, and around the cigar body you can see these kind of starry projections. That is why this entire thing is together known as an asteroid body. Together it is known as an asteroid body. But what is in the center of the asteroid body? In the center of the asteroid body we have a cigar shaped body. So in repeating, in the center of the asteroid body we have a cigar shaped body, and now the entire thing like this is referred to as asteroid. Okay, now having said that, you would next want to see this picture. What is this picture? The blue color stain, lactophenol cotton blue. And what disease is it? Rose gardener's disease. So does this look like exactly a rose, a flower, a rose? It does, right? So please remember, in rose gardener's disease, we will see rosettes of the conidia. We see the presence of rosettes of conidia. So coming back, guys, microscopically, cigar body, we've seen it is surrounded by these pink projections, so we call it an asteroid body. On culture morphology, we see the rosettes of conidia. Photos you've already seen, they look like rose, so rosettes of conidia. That is why I told you in the beginning that everything out here will be to do with the rose. Rose gardener's disease, finger prick, so cutaneous and lymphoid involvement, there is cigar asteroid bodies, and there are rosettes of conidia. Well, if we've done all of these, body heat probably changes shape, and I have to finish this table. The last one, there was one more thing that you had to know, the other name for coccidioidomycosis that they give to you in the exam. ABCD, remember, ABCD means this was also known as the desert rheumatism, which is also referred to as valley fever. So repeating, out of all, these are the ones for which you have to know the separate names. I hope we've revised it. Now it's time to solve a few questions. So let's start. Let's read together. We have an 18-year-old school-going student who develops fever, cough, chest pain, and lymph node biopsy shows you granulomatous inflammation. So up till now, in our country, if I have fever in a patient, cough in a patient, chest pain, and lymph node showing me granuloma, honestly, the first thing that will come to my mind is tuberculosis. Then I see macrophages show oval structures measuring 2 to 4 microns, and all the options given to me do not have tuberculosis. Now tell me, macrophages eating a 2 to 4 micron structure. Does it remind you of this? Does it remind you of this macrophage which is eating these dot structures, that is histoplasma? And anyway, I had taught you everything about histoplasma will be similar to TB. So yes, the answer to this question is histoplasmosis. Coming to the next question that you have. Question number 2. A gardener who pricked her finger while working in the rose garden has developed a local pustule. I think by now you know the disease. There's a gardener and there's rose history given. There's developed a local pustule which has formed an ulcer. After cutaneous nodules, there is also involvement of local lymphatic drainage. Everything is fitting in well. Firstly, we have something called a rose garden that has been mentioned. Number 2, the kind of involvement that has been mentioned is along the lymphatic channels and skin, that is lymphocutaneous. So certainly what I'm talking about is going to be Sporothrix schenki or sporotrichosis. Let's move on to the next one. Cigar body is seen in, right now we studied two bodies, cigar body and asteroid body. So both of them, so I'll repeat again, the round cigar body and then you had the asteroid body, both of them were seen in Sporothrix schenki. Next question, let's read again. 20-year-old college student who's gone for some archaeological team as a part of the team is traveling to Arizona. I think I've got my answer here, but I'll still want to read. Okay, so what does this team do? The archaeologists, they basically help in finding out they excavate, they find out the different and the historical aspects in a particular cave. So they've gone to Arizona, they are excavating some ruins out of it, and 1 month after returning, she has developed a flu-like illness with cough, muscle pain, and fever. X-ray also shows you lung involvement and the spores are mentioned in the picture. What kind of spores are these? I think we know that I'm dealing with the ABCD organism. D for desert, which has been mentioned. C for coccidioidomycosis. B for barrel-shaped arthrospores, which I can see. And what is the other name for coccidio or desert fever? It's the valley fever, right? The valley fever is what is mentioned over here. Indirectly, I can get another question. Same thing. If they give me the picture and they say barrel-shaped arthrospores are seen in, again, always remember ABCD, barrel-shaped arthrospores will be seen in coccidioidomycosis. Let's move forward, move on to the next question. Okay, let's read. A girl who pricked her finger, again, this is something very similar to what I've just taught you. Pricked her finger, rose bushes, local pustule, progresses to an ulcer. Several nodules develop along the local lymphatic drainage. What have they done? It's the same history. They've only changed the options to confuse you. Otherwise, it's the same history. It's the same rose gardener's disease. It's the same skin and lymphatic drainage involvement. So nothing should be a worry over here. They're just changing the options in every exam. Otherwise, the history is remaining the same. Okay, coming to the last question of this particular set of organisms. Which of the following pathogen isolated from the sputum specimen will show the mold growth with tuberculate macroconidia and microconidia in a culture media? What have they written over here? It's a sputum sample. All of them are dimorphic fungi. All the dimorphic fungi can show involvement of lungs. So this much is fine. After that, they said tuberculate macroconidia. This photo we had just studied, which is the organism which Is everything similar to TB? Everything similar to TB is going to be histoplasma capsulatum. So whenever you see these TB kind of similarities coming your way, definitely go in for histoplasma. Okay, now one more question. So right now, if you remember, I was talking about the last organism that I spoke about, where there are multiple kind of, these lesions which are after a rose prick or a thorn prick, a Gardner's disease. You tend to confuse that very commonly with this question. I want you to read this question very carefully. There's an H&E stain of a nodular lesion on the arm of a forest worker, and that is shown in the picture. So you know what happens, you see that okay, there is skin involvement, and yes, there is a kind of a forest or a gardener only given, and many students end up marking sporotrichosis, but that's not the right answer. Please remember, the picture also makes equal importance. What is this photo showing you? It's showing you some brown color bodies. Let us zoom into those bodies, because the answer to this question is chromoblastomycosis. Guys, first and foremost, chromoblastomycosis is also referred to as verrucous dermatitis. It causes verrucous dermatitis. Why? Dermatitis will tell me that there is skin involvement. Verrucous, whenever I use the word verruca always means something like a wart. Something like a wart. So can we all see that cauliflower kind of warty lesion over there on the foot? That is what you see because these are forest workers. When they are forest workers, for obvious reasons, they are walking barefoot. So by mentioning the history of a forest worker, they are trying to tell you that the person is walking barefooted. So that is when this organism tends to enter, and that is why it results in this verrucous kind of a growth. And why do you call it chromo? Because it has a color. Chromo word will always mean color, right? And can we all see what color has it got? It's got a copper kind of a pigment, right? So we call these copper penny bodies. They look like copper coins. Once upon a time when there used to be copper coins, copper penny bodies, they have been given many other names. They are known as muriform bodies. They are also known as medlar bodies, and they are also known as sclerotic bodies. Unfortunately, we'll have to learn all these names because all of them come in the paper. Repeating, we have copper penny is the easiest because they look exactly like copper coins, medlar and muriform, and sclerotic bodies. Now coming back, you had nodular lesion, you had a forest worker, and you had these brown color bodies. So I think now everything is making sense. We are dealing with the case of chromoblastomycosis. So please remember, just by looking at a forest worker, please don't confuse whether you're dealing with the case of whether you're dealing with the case of sporothrix or you're dealing with the case of chromo. Look at the photos. The photos will tell you a lot about it. Okay, having said this, let's move on to the next question that happens to be this. There is a colony of aspergillus which is shown in the image below. This is the photo that is shown, and you have to identify that which of the following aspergillus is put out here. So first let me probably teach you, then we'll come back to this question. So these are the different types of aspergillus that you have to know for the exam. Firstly, we have aspergillus fumigatus, aspergillus flavus, and aspergillus niger. If I ask you out of these three, which is sounding like a black color, fumigatus, flavus, and niger, I think everyone knows niger word refers to black. So which of them will show you black colonies? Obviously, aspergillus niger will show you black colonies, undoubtedly. Now you're left with fumigatus and flavus. Fumigatus sounds like fumes. That's my mnemonic. It sounds like fumes and smoke. So which of them is going to give you a smoky green color? A smoky green color. So smoke and fumes. Fumigatus will give you the smoky green color. And flavus sounds like a flower. Flavus sounds like a flower, and flowers are nice and bright colors like yellow and green. Flowers are yellow and green colors. So let's repeat our mnemonic. Niger will always be black in color. Flavus and flowers will always be nice yellow, bright yellow, green colors. And fumigatus are fumes and smoke. So that's going to give us a smoky green color. Coming back, I asked you this. So first analyze what photo has been given. It's not bright yellow, green. So it's not flavus. It's not a case of black color. It's not totally black. So it's not niger. It's a classical one that comprises of smoky green color. It's a smoky green color, and I know smoky green means fumes. So this is a case of Aspergillus fumigatus. Repeating, smoky green, yellow green, black. You say, ma'am, I want to look at these under the microscope. So you've done the culture. On what have you done the culture? We always do the culture on SDA. So we've done a culture on the subaroutes, dextrose, agar, and now you want to look at them under the microscope. Can I say I am doing culture morphology? And if I'm doing culture morphology, there is a very famous stain, that blue color stain that we use, lactophenol cotton blue. So how do I get to know that this is a case of Aspergillus fumigatus? First and foremost, you have to see there will be a vesicle, a round structure over here and over here. Okay, so this is Aspergillus fumigatus, and there is going to be Aspergillus flavus, and there's going to be Aspergillus niger. I think I can easily pick out niger over here also, because even on this stain, niger will always keep its black color. The black color is not going anywhere. Niger is always and always going to keep that. So niger keeps the black color, and I think I can easily identify. My main confusion always happens between fumigatus and flavus. So when I say fumes, now you have to look very carefully. You see these dot 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 kind of conidia that are arising, but do you see they are not arising from this side and this side? No, they are only arising from the top part of the vesicle. They are only arising from the top part. See over here also, only from the top part, not from the sides, not from the sides. Again, only from the top, because guys, fumes and smoke will always be top. Fumes and smoke will always go top. So please remember, if the conidia only arise from the upper part of the vesicle, if the conidia only arise from the upper part of the vesicle, that is indicative of fumes and fumigatus. Whereas flavus is for flower. That is what we were learning, right? Flavus is for flower. So flower will have petals all around. Flower will have petals all around. Can you all appreciate? You'll say, ma'am, the origin of those conidia are from this end right up till this end. Every side and every circumference or part of the circumference of that vesicle is giving rise to the conidia, just like a flower. So maybe I'll ask you a question and then you'll be able to analyze. If I say lactophenol cotton blue testing has shown which of the following, and you have all the Aspergillus species mentioned, can you see that only the top portion is giving rise to the conidia? Only the top portion. From here and here nothing is coming out. Only the top portion is giving rise, like fumes. So if I'm talking about fumes, we are talking about Aspergillus fumigatus. Let's do a recap. When we talk about the cultures, remember fumigatus, fumes, smoky green, flavus, flower, yellow green, niger will be black. When I talk about lactophenol cotton blue, niger is still black. Flavus gives you full coverage like a flower, and fumigatus, like fumes, is only going to be along the upper part. Let's move on to the next question. In a patient, corneal scrapings, so you have corneal scrapings which reveal narrow angled septate hyphae. Which of the following is the likely etiological agent? Repeating, which of the following is the likely etiological agent? So let's do a recap. Firstly, eye involvement has been mentioned, and the main takeaway point is this: narrow angled septate hyphae. So let me show you two pictures. This is also a fungus and this is also a fungus. And why are they looking so pink? Taking you back to the very first slide. Under what stain does the fungus look very, very pink? Fungus looks extremely pink under the pass stain. So coming back, this is also a pass stain that I have put over here, and this is also a pass stain that we've put. But the first one I'm calling it as mucormycosis, and the second one that I'm calling it as is aspergillosis. And how did I so confidently put it out like this? Please remember, the way you write aspergillus, take up say the first 4 alphabets out of it. So when you write aspergillus, remember A will tell you that it is going to have acute angled branching. It is going to have an acute angle branching. So when I look at the branches over here, you'll say, yes ma'am, this is one hyphae and this is another one, and look at the angle between them, it's acute angle. This is one, this is another, angle between them, acute angle. So whenever I'm seeing a branching, I'm only seeing an acute angle coming out of it. So A for acute angle, and rearrange SPE, it sounds like septate. These are all septate hyphae. Can you all see carefully? There are lines in these hyphae. There are lines that are present. There are septa that are present. So we have septate hyphae, that is along with acute angle branching, aspergillus. Whereas what is mucor? Mucor is the complete opposite. If aspergillus was acute branching, mucor is going to be perpendicular branching. Mucor is going to be perpendicular branching, right? Perpendicular. If aspergillus is septate, mucor is going to be aseptate. So let me prove it. Can you see there's no line in between? It's completely smooth. Completely smooth. Are you seeing any septate? Not at all. There are no septate that I see. Completely smooth. So aseptate, and look at the branches coming out. Branches are coming out at 90 degrees. Branches are coming out at right angles. So mucor and aspergillus are complete opposites. Remember, A for acute angle branching and SPE for septate. But if I just show you this picture and I say which of them is broader, look at this. Aspergillus is very, very thin and mucor is very, very broad. So we say mucor is mota. That is how we learn it. Mucor is mota, and R for ribbon like. Mucor is a mota ribbon. That is how we've learnt it. Okay. Mucor is a mota ribbon. It is a ribbon like structure that you have. So now coming back to the question, they said narrow angled. When I see narrow angled, obviously we are talking about acute angle. And when they say septate hyphae, septate will tell you that we are dealing with A for acute angle and SPE for septate. So means we are dealing with aspergillus. And that was the final answer over here. I hope everyone's got the differences between aspergillus and mucor, because undoubtedly it's very, very important. Coming to the next question. The morphological feature of pneumocystis carinii infection, very, very important. Some people refer to it as pneumocystis carinii and some people refer to it as pneumocystis jirovecii. So please remember, they've asked you what is the morphological feature of pneumocystis carinii infection. These are the 4 options that have been given to you, and I hope you guys will be able to attempt. The answer to this is option number B. That is, exudate intraalveolar means lung involvement. Exudate is present with lots and lots of plasma cells. How do we learn it? Remember the points about pneumocystis carinii that you have to know. Firstly, please remember, we call it PCP. There are 2 meanings of PCP which you have to know. Number 1 meaning is pneumocystis carinii pneumonia. Means what infection does it causes? It causes pneumonia, lung involvement. So PCP means pneumocystis carinii pneumonia. I also call it PCP because it is a plasma cell pneumonia. If someone asks you what are the main cells that you have in pneumocystis carinii, you will say the main cells are plasma cells. That is why, guys, the answer to this question was plasma cell infiltrate. Repeating, pneumocystis carinii pneumonia, also known as plasma cell pneumonia, will show you lung involvement with lots and lots of plasma cells. Under what situation? Please remember, under an immunocompromised situation. Usually they give you something like an HIV positive person or a transplantation person, someone whose immunity is compromised, immunocompromised. Number 3, the only stain, it is only and only stained with what? All stains cannot stain pneumocystis carinii. It is only stained with this, a blackish color stain. I think we know the blackish color stain is Gomori methylamine silver stain. It is only stained with GMS. And what kind of an appearance does it give you? So you will say, in some places it's looking round, right? So I call it a ping pong ball appearance because it's looking round like a ping pong ball. I call it a ping pong ball appearance. But in some places, you'll say it looks as if the ball has been crushed. Someone has crushed the ball. So this is also referred to as the crushed ping pong ball appearance. The first name given to it is crushed ping pong ball appearance. Then some authors say, okay, it's looking like a hat. It's looking probably like a hat. So some people say that it has a hat appearance. Then there's one more imagination to it. They'll say some of them are like a hat and some of them are like a ball. So this is also what? Cup and saucer appearance. That's another name that is given to it. It is known as the cup and saucer appearance. So out of these, honestly telling you, the most likelihood of getting in the exam is crushed ping pong ball appearance. What are the key points? Pneumocystis carinii pneumonia, also known as plasma cell pneumonia, happens in immunocompromised patients. The only stain which can give it a color is gomori methenamine silver, and the only appearance that is most likely to come in the exam is the crushed ping pong ball appearance. I hope this question is clear to everyone. Let's move on to the next question. Which of the following is used for the cultivation of Rhinosporidium? Hella, Hep2, SDA or none? Which of the following is used for the cultivation? So first and foremost, look at the organism. What is this organism over here? Answer is D, none, but let's study about it first. This organism is rhino. Rhino means there's some kind of a nose involvement. These patients come to you with nasal polyps. These patients come to you with nasal polyps. So Rhinosporidium, what is the full name of the organism? It is Rhinosporidium seaberry. Rhinosporidium seaberry, and rhino will tell me that it comes to us with nasal polyps. So earlier, actually, honestly telling you that if we look at the history part of it, earlier it was said to be a fungus. It was said to belong to the mycology section. That is why we are, even today we are reading it here. But now they say this has been categorized as an aquatic parasite. It has been categorized as an aquatic protozoan, an aquatic parasite, and that is the recent update about Rhinosporidium seaberry. Now remember the clinical features I've told you. Patient will come to us with multiple nasal polyps, and what will you see under the microscope? So please remember, for Rhinosporidium, under the microscope you will see these big round structures, and these big round structures are referred to as sporangia. These are referred to as sporangia. Inside the sporangia, you have dot, which basically means you are dealing with endospores. So please remember, sporangia are present, those big structures with dot, spores inside it. That is what you have for Rhinosporidium. And if they ask you, how do you grow it? How do you cultivate or how do you grow it? Please remember, you cannot grow it. This is one of those organisms that cannot be cultivated. One very important point that will come to you in the exam. Please remember, they will ask you that Rhinosporidium seaberry cannot be cultivated. So let's write it down, because definitely I expect this. Cannot be cultivated. So all the things that are written over here. What is SDA? SDA is a culture medium. You can't cultivate it. Hep2 and HeLa, for those who've read virology, you know, Hep2 and HeLa are cell lines. So they are basically used for the growth of viruses. You can't grow Rhinosporidium on these also. You possibly cannot grow it, because they say that there is no aid or no mode of cultivation for this. So Rhinosporidium seaberry done. Let's move on to the next question. Okay, so here we are, and let's first attempt. There's a vitreous aspirate from a case of metastatic endophthalmitis, which on culture shows gram-positive round to oval cells 12 to 4 microns in size. The aspirate on gram staining shows the presence of pseudohyphae. Which of the following is the most likely etiological agent? You've got an eye involvement, endophthalmitis, and you've got something to do with a gram-positive organism, pseudohyphae. Out of all of these, please remember, pseudohyphae is a very common thing that you see with Candida. So when I'm talking about Candida, and in most of the cases we are talking about Candida albicans, Candida shows you two forms. Number one, it shows you the budding form, and number two, it shows you the pseudo hyphae form. And I'll draw it and I'll tell you what do I mean by that. When I say budding, for example, if this is Candida, you will say there's a bud coming out of it, budding. Then if this is Candida, one bud coming out from it, from here another bud is coming. So these tiny things are known as budding forms. What are pseudohyphae? This is Candida. The bud, instead of being like this, instead becomes elongated like this. From this, another bud arises, but instead of being round, that other bud is also going to be like this. Then the next bud is also going to be like this. So basically, when the budding form looks elongated like this, I call it a pseudo hyphae. It's not a true hyphae, it's just the budding form which has become very, very elongated. So remember, Candida shows you budding form as well as pseudo hyphae, and when the word pseudo hyphae is written with such a gram positive history given to us, we are dealing with Candida albicans. One more question that we have is that Reynolds broad phenomenon is seen with. This is a multiple choice question. Multiple answers are correct. Candida albicans, Candida dubliniensis, Gardnerella or Histoplasma. Remember, both the Candidas show you Reynolds broad phenomena. So what is Reynolds broad phenomena? Let's study that. What do we understand by Reynolds broad phenomena? It is known as the germ tube test, GTT. Reynolds broad phenomena is known as the germ tube test. What do you do in it? You take the Candida colony which you got from the patient and you put it in serum that you've obtained maybe from human blood. So serum 1 ml, Candida colony, and you keep these two things for 2 hours. After 2 hours, repeating, all that you take is serum and you take Candida colony. When you will incubate them, you will see that from, if this was the Candida, you will see a germ tube will originate from it. Similarly, if this is a Candida, you will see a germ tube coming out of it. Remember, this is the germ tube test which will only come positive for Candida albicans and Candida dubliniensis. Repeating, you will see a tube coming out of it. So repeating for you guys, Reynolds broad phenomena seen in 2 Candidas, not the others. Only in these 2 Candidas you see it, Candida albicans and Candida dubliniensis. Well, having done this, let's move on to the next question. And here you have the next one. So, identify the dermatophyte based on the macroconidia shown in the image below. Identify the dermatophyte based on the macroconidia shown in the image below. For those who've read it earlier, the answer is epidermophyton, and for those who've not, please read with me once again. So what am I talking about over here? What family have I entered into? I've entered into dermatophytes. I mean that these are organisms with derma involvement. What do I mean by derma involvement? Derma involvement means hair, skin, and nails. So if I first ask you this question, systemic, look at this will indirectly answer your query. Systemic infection is caused by all the fungi except what? So systemic means lung involvement, liver involvement, brain involvement, skin also could be a part of the system. So multi-system involvement. So you will say that cryptococcus, yes, cryptococcal meningitis, it involves the brain. All these histoplasma, paracoccidio, all these are dimorphic fungus. I have right now told you in the beginning of the session that dimorphic fungus will certainly involve multiple organs. So what is the one that does not involve the multiple organs? Answer to that question, dermatophytes, because right now I said dermatophytes, the name tells you that it has derma in it. So when I say dermatophytes, I am involving 3 things primarily. Skin for obvious reasons. Number 2, there is hair. And number 3, there is involvement of nail. Dermatophyte will have skin, hair and nail involvement. Okay, so let us see the types of dermatophytes. You can see I've written dermatophytes and I've written these random alphabets over here. What are the names of the dermatophytes? Please get the names right. There are 3 dermatophytes that you have to know. TME. Do you guys know what is the full form of TME? Where do we use TME? TME is our Telegram URL. Whenever you're searching for someone's Telegram channel, you type TME. So TME means Telegram. What is TME? T for trichophyton. The first dermatophyte is trichophyton. M for microsporum. E for epidermophyton. E for epidermophyton. So repeating, T for trichophyton, M for microsporum, E for epidermophyton. And now I have to tell you, do all of them involve the skin, hair and nails? Please listen very carefully. We'll write it down once again. When I'm dealing with the case of TME, means I have trichophyton. You will say, ma'am, this has the word tri in it. This has the word tri in it. This means this will involve the skin, the hair and the nails. All 3 things will be affected. What is the next one? Microsporum. Next one is microsporum, and the third one is E for epidermophyton. So you will say that all of them are coming under dermatophytes. So one thing is there, that skin involvement has to happen in all. Skin involvement is going to happen in all, but microsporum has skin and hair involvement, and epidermophyton N has skin and nail involvement in it. How do we learn it? Trichophyton has tri, all three are affected, skin, hair, nails. Micro, so now think logically, micro, what is thinner or what is smaller? Hair is thinner or nail thinner? Obviously everyone will agree hair thinner hota hai. Hair is thinner, so hair will be microsporum, and epidermophyton ends with an N, so this is where nail involvement will occur. So skin is seen in all, tri is for tri all three, micro thinner hair, and epidermophyton is N for nail. So I hope this question is clear for all of you. And if I ask you a similar question associated with it, which of the following infects hair, skin, and nails? Everything is affected. I think you will be able to answer trichophyton. Hair, skin, and nails. Let's do repeat. Trichosporum nahi tha. It's trichophyton, which has skin, hair, and nails. Microsporum and epidermophyton both have skin, but epidermophyton ends with an N, so that is skin and nails. And micro, thinner is hair, so that is skin and hair. Coming back, how do I differentiate them? I've understood what is TME, trichophyton, microsporum, epidermophyton. I differentiate them on the basis of macroconidia as well as microconidia. What are these macro and microconidia? Can you see these big structures over here? These big structures are macroconidia. And these tiny little dots that you're seeing, obviously they are micro. So macro will be bigger, micro will be smaller. Microconidia are arranged in a descending order. This means maximum microconidia are seen in trichophyton, next seen in microsporum, and least number are seen in epidermophyton. I'll prove that to you. Have a look at this photo. Can you see these tiny dots? They are maximally seen in trichophyton. In microsporum, you'll say, ma'am, I can only see one tiny dot. And in epidermophyton, you'll say, I just can't see any tiny dot. So they are in a descending order, decreasing order. Maximum, minimum, least. Whereas the macroconidia, there's a mnemonic for that, that is PSC. That is why I told you, TME stands for telegram. And I would tell you the telegram that I hold and I conduct, the telegram channel, which is by my name. So there were a couple of you who were also asking me about the telegram channel in the previous session as to where are all these PDFs going ahead. PDFs are being uploaded on the telegram channel, and the name of the telegram group and telegram channel is Pathology by Dr. Preeti Sharma. So that's the name of the telegram channel, Pathology by Dr. Preeti Sharma, and I think with that doubt I've also made a mnemonic, and the mnemonic is TME Preeti Sharma channel, which means PSC. TME PSC. What do I mean? TME PSC means the macroconidia will be pencil shape, spindle shape, club shape. Pencil shape, spindle shape, club shape. See, TME. Can you see T? Pencil shape. Macroconidia, pencil shape. Next one, spindle shape. I can see macroconidia, spindle shaped. Last one, club shaped. Yes, I can see macroconidia, club shaped. Why don't we look at all these things directly? Why don't we look at T, M and E directly under these blue color stains? What are these blue color stains? Lactophenol, cotton blue. So first, if I say what is the range of microconidia, micro, the smaller ones, you'll say TME, this has the maximum microconidia, this has few microconidia, this hardly has any microconidia. See, you can see in the first one, in trichophyton, there are maximum number of microconidia. In microsporum, they are lesser in number, few of them are there. In epidermophyton, I don't see any small structures. Now let's talk about the macroconidia. For macroconidia, there's a mnemonic P, so TME, and it was PSC. P for pencil shape. So trichophyton shows you these pencil shaped macroconidia. Microsporum shows you spindle shaped macroconidia that you have. And epidermophyton will show you the club-shaped microconidia. So if I take you back to the first question which we had attempted, and I asked you that what is this? So first, always analyze two things, micro and macro. Do you see any microconidia, the tiny dots? No, they are totally not there. So out of TME, microconidia were maximum in this, few in this, and none in epidermophyton. So right now I feel it is epidermophyton. Let me talk about the macroconidia. For TME, the macroconidia was PSC. Pencil shaped? No. Spindle shaped? No. Club shaped? Yes. This is definitely looking like a club. So the answer to this question becomes epidermophyton. These are very important dermatophytes that you have to know. Coming to the next question. Now I think all of you will be able to attempt. All are true regarding dermatophytes except. All are true regarding dermatophytes except. Let's go one by one. They are differentiated based on macroconidia. Are they? Yes, I think. TME shows you pencil shaped, spindle shaped, club shaped macroconidia. So that is a true statement. Epidermophyton infects skin and nails. You say epidermophyton ends with N, nails. Yes, that is correct. Microsporum has fusiform microconidia. So microsporum, what kind of microconidia? Spindle shape. Yes, it's definitely spindle. It looks like fusiform is nothing but spindle only. Microsporum has a fusiform macroconidia. Epidermophyton has abundant microconidia. Is that the case? Microconidia was maximum in trichophyton, then it was little bit in microsporum and negligible in epidermophyton. Does it have abundant microconidia? That's the false statement. It has least or absent microconidia. So I hope now with these mnemonics you'll easily be able to solve all of these. Let's come to the next. Tinea versicolor is caused by which of the following? Tinea versicolor is caused by which of the following? It's a very famous image-based question, and the answer to this question is Malassezia fur. Let me show you a picture. Tinea versicolor is a skin disorder. Repeating, when we are talking about tinea versicolor, the first thing that they will ask you is the organism, and the organism is referred to as Malassezia fur. And Malassezia fur is a derma problem, means can you see on the trunk of the patient? This is the trunk, the back, the shoulder, the neck, and you can see there are hypopigmented lesions. The trunk of the patient will show you hypopigmented lesions. When you look at it under the microscope, now what kind of microscopy is this? Very important, listen to me very carefully. Do you see any color to it? Any pink or black or blue? No color to it, it's colorless because this is referred to as a KOH mount. This is referred to as a KOH mount. We use a 10% KOH mount. So my first question to you guys is, when do I know that I have to put the sample in a 10% KOH mount? When do I analyze that? I analyze 10% KOH mount when I'm using samples with keratin. K for keratin, K for KOH. When keratin is present in my sample, where is keratin present? You will say skin has keratin, hair has keratin, nail has a lot of keratin, especially nail has, why is the nail so thick? Why is it so tough? It has a lot of keratin. So skin, hair and nails, basically all derma samples, all dermatology samples, they have to be kept in a KOH mount because the keratin will be digested. The keratin will be digested, which means that all of these will soften. All of these will soften because the keratin will be digested by KOH. So I get a derma sample. I look at it under KOH. Can you see there's a ball-like dot structure? Yes. And then there are some lengthy lines also. What is this known as? Some dot like of structures, some lengthy lines. This is known as the spaghetti and meatball appearance. This is referred to as the spaghetti and meatball appearance. So all those dot are the yeast. These are all the yeast. They give that meatball appearance, and all these hyphae, the long structures, hyphae, they look like spaghetti pasta. So spaghetti and meatball appearance is seen in Malassezia fur or Tinea versicolor. I look at it under the culture. I know for the fungus, I will always use Sabaurout's dextrose agar, SDA. But over here, I have understood, firstly, what is this appearance? This is referred to as the fried egg appearance. It looks like fried eggs. It looks like a sunny side up. It looks like a fried egg appearance. A fried egg appearance. But if I want to fry the eggs, I will always need oil. If I want to fry eggs, I'll always need oil to fry anything. And oil, in nowadays we say we should always go for healthy oils, and that is olive oil. So this is not a regular SDA. I have added something on top of it. This is SDA with olive oil because I found out that Malassezia furfur is fat loving. It is oil loving. It will grow in oil. So the eggs will get fried in oil. That is, they're fat loving, lipophilic. We say that Malassezia furfur is lipophilic. It is fat loving, oil loving. So what have I done? I've put olive oil on top of it. So I've easily learnt it that fried egg appearance, to fry the eggs, you need an oil. So SDA with olive oil will give you the fried egg appearance. Coming back, the question was, tinea versicolor is caused by what? It is Malassezia furfur. Okay, so having said that, coming to the very last question and the much hyped question following COVID with fungus is right in front of us. Beta 13D glucan testing is done for. So the last question that we have for the fungal etiology, beta 13D glucan testing is done for Candida, Fusarium, Aspergillus, Pneumocystis, all of the above. Before I make you attempt this question, there are two things which became very famous in COVID. There were two markers that became very famous. Remember, during the second wave of COVID, there was a big boom about black fungus, yellow fungus, white fungus, all those random colors and names that were given. That is when people got familiar that, okay, these are the tests that are done when I want to test for fungus. One test is known as beta D glucan and one test is known as galactomannan. What do you do it for? So please remember, beta D glucan is nothing but a normal cell wall component of the fungus. A normal fungus has it in its cell wall. Galactomannan is particularly in the aspergillus. So please remember one thing, galactomannan will primarily be used for aspergillus. So I've called it aspergillus galactomannan. Beta D glucan can be used for a lot of things, for Candida, Aspergillus, Histoplasma, Fusarium, dermatophytes, Pneumocystis jirovecii. It can be used for a lot of things. So what's easier? You learn what will you not use beta D glucan for. I will not use it or it will be negative for BMC. That is how I've learnt it. Beta D glucan testing is negative for B for blastomyces, number 1 blastomyces, M for mucor and BMC, C for cryptococcus. So please remember BMC. These are the three things for which beta D glucan will come out to be negative. Other than this, all the fungal organisms written, it will come out to be positive. Galactomannan can also be positive in a lot of other things, but I prefer using it only and only for aspergillus. So let's repeat. Beta D glucan is for all except BMC. What is BMC? Blastomyces, mucor and cryptococcus. Galactomannan, I prefer to keep it only and only for aspergillus. Coming back. Beta 13D glucan testing is done for. You will say, ma'am, it is done for all. I just had 3 exceptions. I just need to make sure that those exceptions are not there. What were the 3 exceptions? BMC, blastomyces, it's not over there. Mucor is not over here. Cryptococcus is not over here. This means it can be done for all of these. Answer to this question, all of the above. Repeating one last time and then we end the session. Beta D glucan for all except blastomyces, mucor and crypto. Galactomannan testing is done for primarily aspergillus, which finally gives us a rapid review and a finish to the fungal etiologies or the mycology chapter. And I hope you guys are enjoying microbiology, which usually people do not. But I hope it started to make a little bit of sense. For those who've not attended the previous lectures, remember the part 1 and part 2 of these series had general microbiology. Today we've ended up revising the fungal etiologies, and next we'll be going forward with bacteriology, parasitology and so on. So yes, hoping to see you in the next classes as well. Thank you so much guys for joining in, and any other feedback, most welcome. Anything you want that should be included or not in the future sessions, please do write it out to me in the comments below, and I'll try to incorporate it as much as I can. Thank you so much. Study well. All the very best. Hi everyone, welcome back to the YouTube session, and I'm Dr. Preeti Sharma, and I'll be taking the microbiology crash course for the upcoming exams, that is NEET as well as FMG. Now, well, if you're new here, then we've already conducted 3 sessions prior. We've had 2 sessions on general microbiology, 1 extensive session on mycology, that is fungus, and today we'll be starting off with bacteriology, all the important bacteria, the important topics, points, MCQs and images that you need to know for particular bacteria, and especially important for the upcoming exams. So yes, let's begin with bacteriology, and before I begin, I just want to tell everyone that all these crash courses that I'm conducting, or the classes that I'm taking on the YouTube channel, the PDF of these is also available. You can download them from the Telegram group. Either you are part of the Telegram group or channel or both, you can definitely download it from either, and the name of the channel is put up in the description below. You can follow that. It's Pathology by Dr. Preeti Sharma. You can click on the link, you can follow it, and you can download all the PDFs that are being taught out here. Well, let's begin with the session. So these are the set of bacteria that we have to start with. We are going to talk about cocci, and we are going to talk about bacilli. So first I'll talk about a general rule. Now the general rule says that all the cocci are going to be gram positive. All the cocci, they tend to be gram positive, whereas all the bacilli, they tend to be gram negative. So first I'm not going into these mnemonics right now. First I'm starting with all of these, the very first category, and that is referred to as the cocci. So when I say cocci, remember the cocci are gram positive except Neisseria, Moraxella and Velonella. Except for these 3, we have all the others are gram positive. Repeating, all are gram positive except Neisseria, Moraxella, Velonella. These are the only cocci which are going to be gram negative. So first let me begin, before I go on to the bacilli, I'm only going to stick to the cocci as of now, and I'll make you attempt a question first, and then we'll start with the discussion. The synergohymenotrophic toxin of Staph aureus happens to be PV, alpha hemolysin, beta hemolysin, gamma hemolysin. Whatever options you feel, you have to mark from here. So some of you know about this, some of you don't. So what I'll do, I'll teach you first and I'll get back to this question. We basically have to talk about all the different types of toxins that are there in Staphylococcus aureus. So first let's see, we have a lot of virulence factors. Why does Staphylococcus aureus cause all of its features that we are talking about? Number 1, it has something called protein A. Where is protein A present? That's a question. Protein A is present in the cell wall of Staphylococcus aureus. Repeating, protein A is present in the cell wall of Staphylococcus aureus. What does protein A do? Everything with A. It is going to have an anti-phagocytic property, means when Staphylococcus will enter our body, we will try to eat it up, we will try to phagocytose it. Protein A will protect it. It will make it anti-phagocytic. Similarly, it is another anti-complementary. It is also anti-complementary, and it also helps in a test known as the coagglutination test. Please remember, protein A has 3 uses: anti-phagocytic, anti-complementary, and it is used in the coagglutination test. So that is the use of protein A, which is present in the cell wall of the bacteria. Other than that, there are toxins. What toxins are there? Which break the blood. Hemolysins, alpha, beta, gamma, delta. Repeating, hemolysins will break the blood. Alpha, beta, gamma and delta hemolysins are present. There's something known as the PV toxin. If you want to know the full form, PV stands for Pantin-Valentine toxin. Even if you learn it as Pantin-Valentine, you learn it as PV, it's good enough. Please remember, the way you write PV, and out of this alpha, beta, gamma, remember the way you write gamma. Gamma looks very much like a V, right? So remember, the PV toxin and the gamma hemolysin are together working. They are said to be a unit. Synergy. They are said to be a unit. So they are together known as The synergohymenotropic toxin of Staph aureus. Repeating what all? We said the panten-valentine toxin, and the way you write V is the way you write gamma. So the panten-valentine toxin and the gamma hemolysin means the correct answer over here was option number D, that these two together are known as the synergohymenotropic toxin. Let's move forward. There's something known as the exfoliative toxin or epidermolytic toxin. What does this word tell me? Epidermo means some skin problem it will cause. Exfoliation, it will cause exfoliation of the skin. Everyone has heard of staphylococcal scalded skin syndrome, SSS. There is something known as staphylococcal scalded skin syndrome, and that is caused by this toxin. Similarly, toxic epidermolytic necrosis or TEN, all these epidermolytic kind of disorders or exfoliation of the skin kind of disorders like SSS and TEN, they are caused by Staph aureus. Coming to the last one, enterotoxin. The word entero tells me I'm now going to deal with an intestine. So what is the enterotoxin going to cause? Let's take it up on a separate slide. Remember, enterotoxin causes food poisoning. Obviously, entero will cause something to do with food poisoning, and that is what gets you the maximum questions. Remember, the incubation period of this food poisoning is less than 6 hours. It is 1 to 6 hours on an average. So less than 6 hours, if they give you a history in the exam, you are dealing with two things. If they say after consuming food, after eating food, within 6 hours, a person has landed up with food poisoning, either think of Staphylococcus aureus or think of Bacillus cereus. And which one is commoner? Remember, Staphylococcus aureus is more common. So how will you differentiate in the exam? They will say incubation period was less than 6 hours. How will you get to know whether it is Staphylococcus aureus or Bacillus cereus? You'll look out for the food. If they say there is any dairy or poultry consumption that has happened, dairy and poultry consumption, like for example in the previous NEET paper, there was a consumption, there was a birthday party, and everyone had consumed pastries. All the kids had consumed pastries. Now pastry definitely has a lot of dairy, creamy pastries they had consumed. It has a lot of milk, it has cream, it has dairy. So obviously you would have selected Staphylococcus aureus. So anything that is dairy and poultry, go for Staph aureus. If they mentioned C for Bacillus cereus, C for Chinese fried rice or Chinese food, you know what to select. So remember, dairy, poultry, select Staph aureus, Chinese fried rice, Chinese food, select Bacillus cereus. But if no food item is mentioned, if nothing is mentioned, only less than 6 hours is the time given, go for the commoner one, that is Staphylococcus aureus. Now please remember, enterotoxins are of many types. Enterotoxin A, B, C, D, like that. There are many types. All of them will cause food poisoning. All of them will cause food poisoning except enterotoxin F. Now that is going to be something fishy. Enterotoxin F is going to be something fishy. It doesn't, although it looks like entero, it sounds like intestine problem, but there is something fishy. And what is that fishy thing in it? It doesn't cause any kind of food poisoning. It causes TSST. I hope everyone has heard of something known as toxic shock syndrome toxin. Everyone's heard of toxic shock syndrome, TSS. So this toxin, toxin shock syndrome toxin, it causes TSS. And this toxin is known as a super antigen. It's not a regular antigen, it's a super antigen. So repeating guys, enterotoxin, entero word means intestine, but enterotoxin F is fishy. It doesn't cause intestine problem. It's a super antigen causing toxic shock syndrome. Having said that, why don't we attempt a few questions? If I say, which of the following staphylococcal toxin which is responsible for food poisoning? So for food poisoning, what all do we have? Beta exotoxin, enterotoxin, alpha toxin, and TSST. I think for food poisoning, intestine comes to our mind, and enterotoxin is the best answer that we have, right? Moving on to the next question. The protein A of Staphylococcus aureus. This is a PYQ of the FMG exam. The protein A of Staphylococcus aureus is present in cell wall, cell membrane. It's not seen in the Staph aureus, or it's present as a toxin. So please remember, it is correct answer we did in the beginning. Protein A is present in the cell wall of the bacteria. Coming to the next question. So this is definitely something that can help us. Out of the 20 children that went to a party, 11 developed abdominal pain, diarrhea with nausea and vomiting around 6 hours after food consumption. So out of the 20 children, 11 have had it, and the incubation period given to me is 6 hours. Most likely agent that you have. So I know that within 6 hours I have to think of Staphylococcus aureus and I have to think of Bacillus cereus. Let me look at the options. I only have Staphylococcus aureus in the options, so the answer becomes very, very simple. They've not given me the type of food item consumed, but only by looking at the incubation period, I can make a guess. Okay, now this means you've done all the toxins that it can cause. Now, if we ask you that what are food poisoning was just one thing that you learned. If I ask you what are all the things that Staphylococcus aureus does, you learn it by the mnemonic known as soft pains. Please remember, soft pains mnemonic will help you learn everything that you need to know. So what is it? Firstly, it causes all the soft tissue and skin infections. All the soft tissue and skin infections. Next, it causes OM, osteomyelitis. It causes osteomyelitis. F for food poisoning, we've done that. T for two conditions, TEN and TSS, I've told you the toxins. P for pneumonia and pneumatococcus. In fact, everything that I've highlighted in yellow, the most common cause for those things are Staphylococcus. So the most common cause for osteomyelitis, Staph aureus. Most common cause of pneumatococcus and pneumonia, Staph aureus. Most common cause of infective arthritis, Staph aureus. So repeating guys, we have S for skin soft tissue, O for osteomyelitis, food poisoning, 10 TSS, pneumonia pneumatococcus, acute endocarditis, infective arthritis, necrotizing fasciitis, lot of itis coming up way, acute endocarditis, infective arthritis, necrotizing fasciitis and sepsis. Out of which for OM, pneumatococcus and infective arthritis, it's the most common. So if you get this kind of a question saying secondary bacterial pneumonia in a post-influenza patient, post-influenza, post-viral infection, you can have a secondary bacterial infection. So secondary bacterial pneumonia, we just did pneumonias and pneumatococcus. We've just studied that Staphylococcus happens to be the commonest cause out here. I hope all these features are done, and then we can move on forward and understand what are the different types of enzymes that it produces. What are all the enzymes that Staphylococcus produces? You can see 5 enzymes written over here, and we've learnt it by the mnemonic Staphylococcus only. What do I mean by that? T for thermonuclease. Nuclease will remind you of DNAs. So remember, thermonuclease or DNAs is one. pH for phosphatase. C and C for catalase and coagulase. Now these are actually the most important. Staphylococcus aureus is catalase positive and coagulase positive. Repeating, thermonuclease and DNase, pH for phosphatase, C for catalase, C for coagulase. How does the catalase test look like? Catalase test looks like this, guys. Please remember the easiest way is, what is catalase? If I ask you what is the enzyme catalase capable of doing, you will say catalase is something which is capable of breaking H2O2. So what do I do? On this slide, I've taken 1 drop of H2O2. On this slide also, I've taken 1 drop of H2O2. On this, I've put, I don't know which organism is it. On this, I've put some kind of an organism on top of it. On this also, I've put some kind of organism. Right now, I don't know which is staph and which is not. Over here, I saw there are no bubbles, and over here, I saw that bubbles were produced. Remember, whenever bubbles will be produced, the catalase test will be said to be positive. When is the catalase test positive? When bubbles are produced. Why? Because catalase enzyme breaks H2O2. H2O2 is hydrogen peroxide, right? Catalase breaks H2O2 into water and oxygen, and oxygen is responsible for bubbles. So simple, if Staphylococcus aureus is there, catalase will be there, reaction will occur, oxygen will form, bubbles will form. So if catalase is there, bubbles will form. This means this organism was catalase positive. If catalase is not there, bubbles will not form. This means this organism is catalase negative. So Staphylococcus aureus is catalase positive. There's another test that is known as the coagulase test. It's also an enzyme. But there are two ways of doing it. You'll say, ma'am, like catalase test, you were doing it on a slide. Similarly, coagulase test, you can either do it on a slide or you can do it in a test tube. If you want to test for coagulase, either you can do it on a slide or you can do it on a test tube. The slide coagulase test is also known as the bound coagulase test, and the test tube coagulase test is known as the free coagulase test. How do I learn this? Because it's obviously becoming very overwhelming to learn so many things. Which is bound, which is free, which is slide, which is tube. Count the number of alphabets. Free has 4 alphabets. Tube also has 4 alphabets. So free and tube coagulase test are the same. Count the alphabets here. Slide has 5 alphabets. Bound has 5 alphabets. So slide and bound are the same test. That is how we learn it. So slide and bound coagulase test, tube and free coagulase test are the same. And remember, Staph aureus, intermedius and Hicus. Staph aureus, intermedius and Hicus show you both positive. Both of them are going to be positive under it. Whereas Staph lugdunensis, see, dunensis sounds like bound. Bound dunensis, if you turn the alphabets around, bound dunensis. So lugdunensis only shows you the bound test positive. Whereas Staphylococcus schliefferi sounds like free. So schliefferi will give you the free coagulase test positive. Repeating, guys, all these aureus, intermedius, Hicus, both of them are positive for both the tests. However, schliefferi is free test positive, lugdunensis is bound test positive. Let's attempt some questions. So if they ask you, which of the following organisms do not show the test below? Do not show. What is the test below? First, you primarily have to identify that. The test shown below is the test with the bubbles. The bubbly test is said to be the catalase test. It is said to be the catalase test. So they are asking you, which of the following organisms is not catalase positive or is catalase negative? Do you know the mnemonic for that? Remember, cats need space. Cats need space. So the catalase positive organisms, cats, space, that is Staphylococcus aureus, Pseudomonas, Aspergillus, Candida, Enterobacter. Let's repeat. Cats need space. So Staphylococcus aureus, Pseudomonas, Aspergillus, Candida, Enterobacter. Coming back, maybe we get something. So for catalase, the mnemonic that we said was space. Let's start repeating. S for Staphylococcus. Do we have Staphylococcus over here? It is catalase positive. P for Pseudomonas. Do we have Pseudomonas over here? Positive. A for Aspergillus, which is not over here. C for Candida. I don't see Candida over here. E for Enterobacter. Enterobacter is something which is not Enterococcus. Enterobacter would be definitely something to do with Enterobacteriaceae family. So that's an altogether different family. That is positive. Enterococcus is not catalase positive. Talking about Mycobacterium, that is where I want to complete the mnemonic. Cats need space and space is being made. That is how we learn. We say cats need space and space is being made. So space I know and being made will remind you of Mycobacterium. So yes, Mycobacterium is also catalase positive. Mycobacterium is also catalase positive. So again, cats need space. Let's learn. Cats means catalase needs space. Staphylococcus, Pseudomonas, Aspergillus, Candida, Enterobacter, and space is being made, which tells us that Mycobacterium is also catalase positive. Well, having said that, let's move on to the next question. Which of the following statements is incorrect about the test shown below? Or let me just add on before I get to that question. Let me ask you this question first. I think it will be a better thing to do. Which of the following organisms do not show the test below? That's the better question, and this is a NEETPG PYQ. This is a NEETPG and even the FMG students need to know this. So if you know this, you automatically know the other questions. Do not show. What test is this? Something said as positive, something said as negative. Okay, I don't know what test is it. Positive and negative is mentioned, but I can see that it has been done in a test tube and all the staphylococcal organisms have been mentioned over here. If it has been done in a test tube, it could be a coagulase test. It could be a coagulase test. And what is the other name for tube coagulase test? Tube has 4 alphabets. Free has 4 alphabets. Yes. So the free or the tube coagulase test is being done over here. Do I know there are 3 organisms, that is Staphylococcus aureus, Staphylococcus intermedius and Staphylococcus hicus, which show us both. They show us both tests positive, whether it is tube, whether it is slide. So Staphylococcus aureus will be positive, hicus will be positive. Let's talk about Staphylococcal Schliefferi. I have learned Schliefferi is free test positive, definitely. Lugdunensis is only the bound test or the slide coagulase test positive. So which of the following over here does not show the test below? Staphylococcus lugdunensis does not show this test. It shows the bound or the slide coagulase test. So I think the catalase and coagulase reactions seem to be well versed. And finally, we can go on to the diagnosis. How do we do the diagnosis of Staphylococcus aureus? Firstly, do you guys know when I write Staphylococcus, what's the meaning of the word? Why did they call it Staphylococcus? So now today you'll get to know. Coccus means ... bacteria, dot like round. You will say, yes ma'am, you can see these round bacteria. And why are they purple in color? Because I know all cocci are gram positive. These are gram positive cocci, so just call it coccus. Why did you call it staphylo? Staphylo word means bunch of grapes. Can you see they are arranging like a bunch of grapes? They are arranging like a bunch of grapes. So the staphyle word means bunch of grapes and coccus means the cocci bacteria ... Okay, then you say, ma'am, we've also called it Staphylococcus aureus. What is the reason for calling it Staphylococcus aureus? Because aureus means golden. The meaning of aureus is gold. Please remember Staphylococcus. Can you all see what kind of a culture is this? Please remember what you see over here is known as a nutrient agar. We can probably take it on a separate slide. Have a look at this, guys. See this? This is said to be a nutrient agar, and on a nutrient agar, I can see that there is a golden yellow pigment. There is a golden pigment that it has produced. Golden is aureus. That is why you call it Staphylococcus aureus. Golden pigment is produced, and the name of this pigment is Staphylo, because obviously it's Staphylococcus xanthine. Staphyloxanthine is the name of the pigment. That's on a nutrient agar. What is the next one? The classical red color one, and that is known as the blood agar. And please remember, in the blood agar, can you see there's a dot and a light area around it? A dot and the blackish area around it. Yes, blood is being broken, but very minimally. There is something known as a narrow zone of hemolysis. There is something known as a narrow zone of hemolysis. Please remember, Staphylococcus aureus on a nutrient agar shows you the golden yellow pigment, and on a blood agar shows you a narrow zone of hemolysis. Last one. What do you think are the selective media? What is the selective media for selective and differential? I'll tell you the difference between these two. What is the selective and the differential media for Staphylococcus aureus? And that goes by the name of MSA. Mannitol, salt, agar. Every word has a significance over here. What have I called it? I've called it mannitol, salt, agar. Firstly, agar will tell you that yes, it's a culture plate that you are using. Salt is what makes it selective. Salt is what makes it selective, because who can grow in salt? What do I understand by selective media? Only the desired bacteria will grow. Only and only the desired bacteria is going to grow. Means if you want to grow staphylococcus grows in salt, others don't grow in salt. So I'm using I'm using salt because staphylococcus grows in it. Okay, so salt has made it selective. And secondly, what has made it differential? Differential means difference between. What am I using over here? Mannitol. Mannitol has made it differential, means I want to find out, is it a mannitol fermenter or is it not a mannitol fermenter? I'm differentiating. Is it a mannitol fermenter or not? So that is why it makes it a differential medium. Please remember, when there is mannitol fermentation present, we are going to get a yellow color. So if we get a yellow color, means I'm dealing with a case of Staph aureus, means there is mannitol fermentation. If we don't get a yellow color, this means mannitol fermentation has not happened and this is not Staph aureus. Repeating for everyone, guys. Now when I come back on the culture, so you can say three things. Number one, I think everyone's understood what are you going to see on a nutrient agar. On a nutrient agar, you'll get a golden pigment. What will you see on a blood agar? You are going to see narrow zone of hemolysis. And what are you going to see on a mannitol salt agar? Mannitol fermentation will be noted. All of these point in favor of staph. So if I make you answer a question now, as per the images shown below, which of these does not correlate with the culture characteristics of Staphylococcus aureus? Which of these does not correlate with the culture characteristics of Staphylococcus aureus? Options given are 1, 2, 3 and 4. Let's start analyzing. Blood agar with a narrow zone. This is Staphylococcus aureus. Golden yellow pigment. Definitely yes. Mannitol fermentation. Definitely yes. The fourth one is the answer over here. It does not correlate. Why? Because if this is the organism, is this a narrow zone of breaking or it's a wide zone of hemolysis? A lot of red blood cells have broken. This is said to be a wide zone of hemolysis, and staph, as far as I know, staph does not show you a wide zone of hemolysis. It shows you a narrow zone over here. Wide zone of hemolysis is streptococcus, not staphylococcus. Streptococcus pyogenes shows you a wide zone of hemolysis. So that is exactly what we'll be reading next as well. And that is what tells you that this does not correlate with the culture characteristics. Let's move forward, going on to the next question. How does Staphylococcus aureus become resistant to methicillin? How does it become resistant to methicillin? For those who've read it, you know that the answer is going to be the MEC-A gene. And for those who've not, then now we are going to study it. So let's start with something very, very basic. What is the first thing that you would want to give? The first thing that you would want to give would be penicillins or beta-lactams. Penicillin or beta-lactams, but now it is said that there's a lot of resistance towards penicillin or beta-lactams that is noted because of what gene? Because Staphylococcus aureus produces something called beta-lactamase. If beta-lactams are trying to act on Staphylococcus will produce beta-lactamase. How do they make beta-lactamase? By the BLA gene. By the BLA gene, they make beta-lactamase. BLA gene for beta-lactamase, and that is what makes them resistant to penicillin. So if they become resistant to penicillin, what are you going to give them? You're going to give them methicillin. But now they are saying because these bacteria have the MEC-A gene, because they have the MEC-A gene, they are now becoming, see, methicillin, ME, because they have the MEC-A gene, they are becoming resistant to methicillin, known as methicillin resistant staph aureus. That is referred to as methicillin resistant staph aureus. And if there is methicillin resistance, that results in the treatment by vancomycin. Although now we even have vancomycin resistant staph aureus, so resistance is ongoing because of the inadvertent use of so many antibiotics that is happening. But yes, ultimately when so much of resistance comes, you have to do a culture sensitivity and find out that what is the antibiotic which will finally act upon it. So basic, first you give penicillin or beta-lactams, but if they have BLA gene and beta-lactamase, they will become resistant. So you give methicillin. If the bacteria has MEC-A gene, it will again become resistant. So you give vancomycin. Now coming back to the question once again, I had asked you, how does Staphylococcus aureus become resistant to methicillin? So methicillin resistant by the MEC-A gene. That was the answer over here. I hope everyone sorted with this set of information also, and we can move on to the next one. Organism causing IV biofilm. Organism causing IV biofilm. What is that organism? So, four options: Staph epidermidis, Actinobacter, meningococci, mycobacterium. So remember, staph epidermidis is the answer over here. What am I talking about? Please listen very carefully. There is something known as cons. What do you understand by cons? Cons means, right now, did I tell you that Staphylococcus aureus, when we were studying Staphylococcus aureus, did I tell you it is coagulase test positive? And then I told you the two types of coagulase, tube coagulase, slide coagulase. It was coagulase positive. But remember, there is a set of Staphylococcus which is coagulase negative, repeating, CO for coagulase and N for negative staphylococcus. So coagulase negative staphylococcus is what you have to know. How many types are there? Staph epidermidis and saprophyticus. Epidermidis gets you the maximum questions. Epidermidis makes a biofilm. Biofilm is something, this very commonly makes it in the IV cannulas. The IV cannulas, the intravenous cannulas that are put, that is where this grows and it makes a biofilm. What is the problem with the biofilm is that biofilm will make it very, very resistant to antibiotics. Biofilm is like a cage. It's like a protective cage. It will make it very difficult for the antibiotics to reach these organisms. So staph epidermidis has an IV biofilm, and also it is involved in prosthetic valve endocarditis. Prosthetic valve endocarditis association is also seen. Versus saprophyticus is very commonly seen with UTI. It's not that common, but when regard to UTI, yes, it can cause. So repeating, what we asked you over here was organism causing IV biofilm, and that is staph epidermidis. Repeating, these are coagulase negative staph. Either you have staph epidermidis or you have staph saprophyticus. This is the one that causes biofilm production. Let's move on to the next one. Staph epidermidis has become important due to biofilm formation, virulence, wide spectrum antibiotics, novobiocin resistance. So remember, the most important point as to why staph epidermidis is important nowadays, because of biofilm production. IV biofilm, that is exactly what we studied right now. Moving on to the next question. Granuloma in the lymph nodes is seen in all the infections except PYQ FMG exam. Granuloma in the lymph nodes is seen. So does TB show you granulomas? Definitely. Sarcoidosis? Certainly. So does Haemophilus ducrei. That also causes granulomatous inflammation. What does not cause granulomatous inflammation is Staphylococcus aureus. It does not cause granulomatous inflammation. Coming to the next one, which of the following organisms causes the fastest food poisoning? What did I understand by fastest? I told you anything under 6 hours. What were the two organisms under 6 hours? We said that we had Staphylococcus aureus and we had Bacillus cereus. And now let us find these options. Staphylococcus aureus is also there and Bacillus cereus is also there. So ideally this is not a perfectly framed question, but since it came as a PYQ, you'll have to select one, and the commoner one out of these two will be Staphylococcus aureus. Coming to the next question. A person ate some milk products in a party and after 6 hours started vomiting. So firstly, under 6 hours, again, under 6 hours, someone has had food poisoning, which means Staphylococcus aureus and Bacillus cereus. What is the difference in the food consumption? In Staphylococcus aureus, it is dairy and poultry. In Staphylococcus aureus, dairy and poultry. In Bacillus cereus, it is C for C, that is Chinese fried rice or Chinese food. Here you had dairy product, which means we are dealing with a case of Staphylococcus aureus. The same history comes to you with Chinese fried rice and your answer becomes Bacillus cereus. This also is a previous year question. Let's pick up the next question, guys. Another PYQ your way. So you'll analyze that this topic is their all-time favorite. Food poisoning 2 hours after intake of food. Anything less than 6 hours, again, Staph aureus, Bacillus cereus. Staph aureus and Bacillus cereus, though both are there in the options, I will have to select the commoner one. The commoner one is Staphylococcus aureus. Okay, all said and done. I think this set of bacteria is done and we can move on to the next one. And I know the question comes across as a very, very overwhelming question with too many lengthy options. Let's read the question, and then I think this one question finishes off everything about streptococcus that one is required to know. That's the beauty of this question. Let's read. Each of the following statements about the classification of streptococcus is correct except. So basically they've asked me which of these is the wrong statement. Whenever you read these kind of questions, make an analysis in your mind. What have they asked me? Have they asked me to pick the correct statements? No. They've said except. So in my mind, I've made an impression. Pick the one wrong statement. Find the one wrong statement out of these. So one by one, I'll keep coming back to these options because there are so many options written over here. But I think I first have to start with the classification of Streptococcus. That is something which is very, very important. The classification of Streptococcus we'll start with. So guys, when I start with the classification, how do I divide it? I divide it on the basis of hemolysis. How can it break red blood cells? How can it break the blood agar? So if I say that it only breaks it partially, it only breaks blood partially, or it breaks blood completely, or it does not break blood. Simple. So partial hemolysis, complete hemolysis, and gamma hemolysis means no hemolysis. Alpha, beta, gamma, streptococci is what we have. They will ask you the colors. So remember, when blood is not broken at all, there is no color. Blood will remain blood. Red color blood will remain red color blood. But when blood is broken down either completely or partially, on complete breakage, blood will become yellowish in color. On partial breakage, it will become green in color. How do I learn this? A little tough, right? So what have I written? We've written something called alpha, we've written something called beta, and we've written something called gamma. Gamma, you've understood there is no hemolysis, no color change, no hemolysis. Alpha, the way you write alpha is the same way that you write a G. So what color does alpha hemolysis give? Alpha hemolysis gives a green color. Similarly, the way you write a beta is the same way you write a Y. So what color does beta hemolysis give you? It gives you a yellow color. Repeating, alpha hemolysis gives you a green color, beta hemolysis gives you a yellow color, and gamma hemolysis does not give you any color. So coming to the alpha category, it has two things. It has Streptococcus viridans and Streptococcus pneumoniae. Repeating, Streptococcus viridans and Streptococcus pneumoniae come under alpha hemolysis. Under beta hemolysis, we further have a classification which we call as the Lancefield classification. And under gamma hemolysis, we have one organism called Enterococcus. So I think alpha and gamma are done. Alpha has viridans and pneumoniae. Gamma has Enterococcus. This beta is what will attract further attention. The beta is divided, beta Streptococcus has something called Lancefield classification. And what is Lancefield classification? Can you see I've highlighted a C out of it? So you'll ask me that, ma'am, first tell me, out of alpha, beta, gamma, Lancefield is done for what? It is the beta one that is classified further on the basis of Lancefield classification. And what is the basis of this classification? It says that it is going to divide it into many groups like group A, group B, group C, group D, so on and so forth. But this classification is based on the C for carbohydrate antigen in the cell wall. It is based on the carbohydrate antigen in the cell wall. Repeating, C for Lancefield classification, C for carbohydrate antigen in the cell wall. And this divides it into many groups like group A, group B, group C, group D. So for example, if I ever say I have a case of group A beta hemolytic streptococcus, or I'll say I have a case of group B beta hemolytic streptococcus, or I have a case of group C beta hemolytic streptococcus. So first I'll mention the group and then I'll tell you that yeah, I'm dealing with a beta hemolytic streptococcus. So for the beta, we have something called Lancefield classification, and that is based on the carbohydrate antigen. So when I come back to the question, guys, when I come back to the question, I do see one option over here. It says viridans streptococci are identified by Lancefield grouping. Firstly, tell me viridans. Viridans belongs to which category? As far as I just taught you, viridans and pneumoniae were belonging to the alpha category. Viridans is belonging to the alpha category. And is alpha category identified by Lancefield classification, or is the beta category identified by Lancefield classification? So I have straight away found my wrong statement. They've written that this is based on the carbohydrate antigen. This part is right. Lancefield is based on carbohydrate antigen, but it is done for the beta one. It is not done for viridans. It is not done for alpha. So the wrong statement, which one of these is wrong? The wrong statement is option C. One by one we'll study the other right ones also, but the wrong or the incorrect statement that we have is option C. Okay, having said this, let's move forward. Now, out of all of these, I am going to consider group A beta hemolytic streptococcus. And what's the name? Group A beta hemolytic streptococcus. Remember, it is known as streptococcus pyogenes. Group A beta hemolytic streptococcus is streptococcus pyogenes, and there's a superb mnemonic that you're going to get now. When we are talking about group A beta hemolytic streptococcus, group A beta hemolytic streptococcus, if it is written like this in the exam, don't get confused. Group A beta hemolytic streptococcus, streptococcus pyogenes. Everything with pyogenes will be pi, pi, y. That is how we've learned it. Number one, how do you see it? You'll see it as chains. Remember, how is streptococcus seen? Streptococcus is seen in the form of chains. What is the transport media for pyogenes? Pi, pi. The transport media for pyogenes is the Pike's media. What kind of colonies? You will say, ma'am, it causes like a pin. Like a pin, it causes pinpoints. So, pyogenes, Pike's media, pine point colonies, and how much of hemolysis? You'll say a very, very wide zone of hemolysis. It causes a wide zone. So, remember pi, 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 y. That is how we've learnt it. Pyogenes, transport media, Pike's media. Blood agar shows you pinpoint colonies, and it has a wide zone of hemolysis around it. That is what is said to be streptococcus pyogenes. I hope up till now it is making sense. Further, you'll want to study the biochemical reactions. That also you'll learn with the word streptococcus pyogenes only. Means what? You will say first and foremost, right now, we had done all the catalase positive organisms. Remember, we said cats need space and space being made. Do you remember that mnemonic? So, S was for Staphylococcus. We'll repeat. We had Staphylococcus, Pseudomonas, Aspergillus, Candida, Enterobacter, Mycobacterium. Streptococcus was not there. Streptococcus has nothing to do with catalase. It is a catalase negative organism. Coming to the next. Remember, it is S for sensitive. It is bacitracin sensitive. It is bacitracin sensitive. And PY plus. Pyogenes, I've written it as PY plus because it is PYR test positive. It's a very complicated reaction and you don't need to know the reaction as such for the exam. But you need to know that, remember, Streptococcus pyogenes is sensitive for bacitracin and PYR test is going to be positive. I hope that is somewhere making sense because we can move on to the question. Let's attempt this question, a lengthy one, so let's read it. An 8-year-old boy is brought to the emergency room with a 3-day history of fever, pretty high fever, 102 degree Fahrenheit, and abdominal pain. He complains of pain in his right knee as well as elbow. So there's joint involvement, there's high fever, there is abdominal pain. And 4 weeks ago, he was seen for sore throat and a rash. The throat culture had shown gram-positive cocci in chains and antibiotic was given. Remember, gram-positive cocci in chains. Gram-positive cocci means purple color and they are in chains. Have you started thinking of Streptococcus pyogenes? Because Streptococcus pyogenes also causes sore throat. Definitely. And it is present in chains. Further, I see physical examination, the temperature is high, the blood pressure, the heart rate are mentioned. So there's nothing very catchy in the heart rate and the blood pressure. We see that there is definitely tenderness in the joints that is there because arthralgia, arthritis is noted. Laboratory tests show C-reactive protein as positive. C-reactive protein is definitely an inflammatory marker. Bacterial infection, CRP will come positive. WBC 22,000. The normal WBC is 4,000 to 11,000. Over here, the WBC has come out to be elevated as 22,000. ECG shows you prolonged PR interval. Where are we moving towards? We are moving towards heart involvement. There is a prolonged PR interval. All of these are features of TLC being high, C-reactive protein being positive, the joint involvement that was mentioned over here, the fever that was mentioned over here, and finally the cardiac involvement mentioned over here. We are dealing with the case of rheumatic heart disease, rheumatic fever, rheumatic heart disease, and I know that Streptococcus pyogenes definitely causes rheumatic heart disease. Which of the following tests would be positive? Now tell me, have I taught you catalase and coagulase in Streptococcus? No, catalase and coagulase was Staphylococcus. Now I come to these antibiotics. Did I just now teach you that there is bacitracin sensitivity noted? So the answer over here definitely becomes option C, and that is sensitivity to bacitracin. So I'm giving you all a little bit of a homework. Now that we've studied Streptococcus pyogenes and we've studied everything around it in terms of micro, I want all of you to go and study the criteria for rheumatic heart disease, because everything mentioned here was a case of rheumatic heart disease. So I know you know the name of the criteria, the revised Jones criteria, just not Jones criteria, the revised Jones criteria. I want all of you to open up your medicine notes, read up the revised Jones criteria, something which is very, very important both in pediatrics and medicine. And once you're done, it's a kind of an attendance. Once you're done reading with the revised Jones criteria, you have to write a done and a thumbs up in the chat below. In the comments below, a done with the thumbs up will tell me that you were listening throughout the session, and secondly, that you've done your bit of homework. Okay, let's have another question discussion over here. Streptococcus pyogenes can be checked by which drug, which antibiotic? Did I just study? Streptococcus pyogenes is bacitracin sensitive. So which is the antibiotic or drug disc that we will use? It will be bacitracin. I hope that's okay. Group A, beta hemolytic streptococcus is done. Let's move on to the other group. Now let's go on to group B, beta hemolytic streptococcus, and that is known as Streptococcus agalacti. Streptococcus agalacti, which is involving, it can involve the young children also, the neonates also, as well as the adults. It can cause brain involvement as well. It can cause meningitis also, pneumonia also. So Streptococcus agalacti, like I said, it can affect the neonates, it can also affect the adults or older children, you can say. Rather than adults, I'll prefer going for the word older children. It can cause meningitis, it can cause pneumonia, and there are two tests that you have to learn for agalacti. What are the two tests? It is CAMP test positive and hippurate hydrolysis positive. Repeating, CAMP test positive and hippurate hydrolysis positive. Out of them, they will always ask you what is a CAMP test. Firstly, if you want to know the full form of CAMP, it is Christie Atkins Munch Peterson. It stands for Christie Atkins Munch Peterson, but these are names of scientists, and honestly, if you skip this information, there's nothing that you're going to lose out here. CAMP is the full form, I've told you, nothing to learn. But what is the CAMP test and what exactly am I seeing over here? That is something that I would want all of you to focus on. So what is the CAMP test, guys? You can see what kind of a medium have I taken up over here. I've taken something called the blood agar. You can see the reddish color media. I've taken something called a blood agar. And on a blood agar, what have I done? I have done a vertical streak of Staphylococcus aureus. I have done a vertical streak over here of Staphylococcus aureus, and perpendicular to it, one perpendicular line here, one perpendicular line here. As of now, I don't know what perpendicular line from the patient. This is patient number 1, and maybe this is a known patient number 2. Right now, I don't know, are they suffering from Streptococcus agalacti or not? So over here, what's the difference between patient 1 and patient 2? You said in patient 1, at the junction of your sample and Staphylococcus aureus, you saw an arrowhead that was formed. Whereas in patient number 2, there is no arrowhead that is formed. So remember, when the arrowhead will form, that will indicate that the CAMP test is positive. I'm sorry. When the arrowhead will form, that will indicate that the CAMP test is positive. So remember, you have to look out for this particular arrowhead here, then CAMP test is positive. If it's not there, this means the CAMP test is negative. Repeating, they will ask you for the labelings, guys. That's the most important thing out here. Vertically, what do you do? Staphylococcus aureus. Perpendicular, you put the patient sample, like over here Streptococcus agalacti, and you found that arrowhead. Very, very important test. Okay, having said that, you've done with, let us do a checklist. What all have we done? We had alpha, beta, gamma category, right? We had something called alpha, beta and gamma category. Out of that, the beta category is done. The Lansfield classification, we've done group A beta hemolytic streptococcus, which was Streptococcus pyogenes. We've done group B beta hemolytic streptococcus, which is Streptococcus agalacti. Let me move on to the next one. Let me move on to gamma hemolysis, then we'll come back to alpha. Let us move on to gamma hemolysis. And gamma hemolysis means what? Gamma means no hemolysis. There is no hemolysis that is going to occur. And what is there under gamma hemolysis is enterococcus. What is there under gamma hemolysis? It is enterococcus. So how do I learn enterococcus? From the name enterococcus itself. The mnemonic of enterococcus is enterococcus itself. So we'll try and decode the mnemonic. The way you write an E over here, you can make the E into a spectacle. You can make an E into a spectacle because this is a spectacle gram-positive cocci. The shape of it is that of a spectacle. So that's the first E, you've changed it into a spectacle. Now we come to the next, that is N. N means it has it has no motility. It does not show you hemolysis, gamma hemolysis, and NaCl 6.5% it can survive. Remember, no hemolysis, no motility, NaCl requirement 6.5%. T for tolerant, it is tolerant to heat. E for, I've changed the E again. Every time I get an E, I change it. First, I changed it into a spectacle looking. Now, I've changed it into a B because this grows in the presence of bile. How much percent bile? 40% bile. That is why if I ask you that you want to grow a particular enterococcus, which kind of medium will you use? You will say, ma'am, I'll use the BEA medium. BEA medium is bile, esculin, azide, agar. Even if you don't learn the full form, nothing to worry. Just remember, BEA is something that has bile, and if enterococcus will grow on it, what color has it got you? It's got you a black color. So remember, bile and black have to go with enterococcus. Bile and black have to go with enterococcus. Coming back, so the E I have changed into a B. Bile, esculin, azide, agar gives us a black color growth. Finally, we come to the last one, and that is R. There is some reaction which is positive, and which reaction is positive? PYR reaction is positive. PYR test is positive. So repeating guys, E for spectacle, N for non-motile, non-hemolytic, NaCl 6.5%, T for tolerant, E again for B, bile 40%, BEA, black color, and reaction PYR test positive. Why don't we go back to the question that we were studying? Are we able to rule out something over here? Enterococcus are group D streptococci and can be classified by their ability to grow on 6.5% NaCl. Did we just study that enterococcus can grow on a 6.5% NaCl? Yes, N for NaCl 6.5%. So that is certainly making sense out here. So this statement becomes the true statement. Fine. Having said that, now we are done with the alpha category. Now we are done with the beta category, the gamma category. We can straight away come down to the alpha category. What all comes under alpha? There are two things. Alpha hemolytic has viridins group versus pneumonia group, pneumococcus. Streptococcus viridins versus streptococcus streptopneumococcus or pneumonia. That is what comes under the alpha category. How do I differentiate between them? So remember, the difference is primarily on these two. Rather than shapes, the differences is primarily on these two. Remember, viridins has RI. You will always remember R for resistant. What is it resistant to? It is optochin resistant. It is optochin resistant. And I for insoluble. What insoluble? It is bile insoluble. Repeating for you guys, R for resistant. It is optochin resistant, and I for insoluble, it is going to be bile insoluble. If you've learnt viridins, pneumococcus is the opposite. This was optochin resistant, pneumococcus becomes optochin sensitive. This was bile insoluble, pneumococcus becomes soluble. So simple way of learning, pneumococcus is soluble and sensitive. It is soluble and sensitive, whereas viridins is going to be insoluble and resistant. Okay, so having said that, now I can again get back to the options, but before I get back, let me just finish it off. What do they cause? Common sense. What do you think Streptococcus pneumococcus will cause? It will obviously cause a case of pneumonia. It will cause pneumonia in it. And what does viridins cause? Viridins causes endocarditis. Viridins is very famous for causing endocarditis. Next question they ask you, viridins is present in chains. Viridins is an organism that is present in chains, and pneumococcus is an organism that is lens-shaped, lanceolate with a capsule. Let me show you a photo. This is the photo of Streptococcus pneumoniae or pneumococcus. Like I said, it is lanceolate with a capsule. It is lanceolate with a capsule. When I try to grow it on a culture, when we try to culture Streptococcus pneumoniae, we get something known as, either you can call it a Draughtsman colony, either you can call it a Draughtsman colony, or you can call it a carom coin colony. It is also known as a carom coin colony. So repeating, Draughtsman colony or carom coin colony. Why so? How does a carom coin look like? Carom coin is elevated in the periphery, depressed in the center. Does it look exactly like this? Elevated in the periphery, depressed in the center, elevated in the periphery, depressed in the center. Yes? So remember, carom coin or Draughtsman colony is what you see with Streptococcus pneumoniae. Plus, did you see that this lanceolate also had a capsule around it? That's a very, very important point. It is a capsulated organism, means Streptococcus pneumoniae is made up of capsular polysaccharide. And on the basis of capsular polysaccharide, you can divide it into many subtypes. There are almost over 90 subtypes that are known of Streptococcus pneumoniae on the basis of the capsular polysaccharide. So if I ask you that on the basis of capsular polysaccharide, can you do the typing of Streptococcus pneumoniae? Yes, the typing of it can be done. And there's a very famous reaction for any organism with capsule. Like for example, if you say, ma'am, Haemophilus influenza also has a capsule, Streptococcus pneumonia has a capsule. So all of them which have a capsule, they show something called the Quellung reaction positive. What do I mean by Quellung reaction? Please let's draw a diagram. For example, if this is the bacteria, this is a bacteria, and this is the capsule on top of it. So do you all agree that every capsule will definitely have an antigen? Every capsule will have an antigen, and I know that if I add anti-capsular antibody, if I add an antibody against it, ideally will the antigen antibody reaction occur? Will that ideally take place? Yes. This already has an antigen. If I add an antibody, then the reaction will take place. Don't you think this capsule will swell up in that case? If there is a reaction that will occur, if the reaction will occur, I will see that the capsule will swell up. There will be capsular swelling that will be noted. So please remember guys, that is known as the Quellung reaction. It's the swelling of the capsule on adding anti-capsular serum. That is what you get in a case of Streptococcus pneumoniae, or you also call it pneumococcus in that manner. Okay, now let me come back and visit the question. Let us read option first. Streptococcus pneumoniae are alpha hemolytic. Yes, alpha hemolytic are two things, viridins and Streptococcus pneumoniae, and these can be typed on the basis of the polysaccharide capsule. Right now, exactly what we studied. It definitely can. Let's go on to the last option. Although pneumococci and viridins are alpha hemolytic, correct, pneumococci and viridins are alpha hemolytic, they can be differentiated by bile solubility and susceptibility to optogen. Exactly my mnemonic. Do you remember the mnemonic for viridins? I have taught you that viridins R will tell me that it is optogen resistant, and I will tell me that it is bile insoluble. Whereas pneumococcus, for pneumococcus we said that it ends with an S, so everything is S. Bile, it is soluble, optogen, it is sensitive. So can they be differentiated on the basis of these findings? Certainly can. And finally, this question, like I said in the beginning, the beauty of this question is that the entire Streptococcus chapter can be finished within the domains of this, and I think we've managed to do that wonderfully. So please remember something very, very important. This entire question only per se is very important. Right. So, well, having said that, I think we can take up another question. A 30-year-old patient presented with fever presented with headaches and presented with vomitings. He had a splenectomy few years ago. What is the most probable organism? So actually this is again a question which can have multiple options, right? Whenever there is a person who's undergone splenectomy, maximum chances of infection happen to be with capsulated organisms like pneumococcus, H. influenza, even meningococcus. Pneumococcus, H. influenza, meningococcus, basically these are the kind of things which probably get you, which have a capsule, right? These are the things which have a capsule, and spleen helps you fight capsulated organisms. Please remember, spleen is something that helps you fight against capsulated organisms. And now I'm saying that the spleen is removed. The spleen has been removed. This means you cannot fight against, you will be susceptible. Person will be susceptible to all these capsulated. So like I said, there are two best answers that you'll get over here. Pneumococcus, meningococcus. Going in the order of more importance, pneumococcus stands better. But yes, both of them are important because capsulated organisms are being mentioned over here. So I suppose that is done. Let's move on to the next question. A patient presents with signs of pneumonia. The bacterium obtained from sputum was a gram-positive coccus which showed alpha hemolysis on sheep agar. Repeating, you have a case of pneumonia and alpha hemolysis. Under alpha group, I only know two organisms. One is viridins and the other is going to be pneumococcus. But remember, pneumococcus is something which is obviously going to cause pneumonia. So I know I'm dealing with the case of streptococcus pneumoniae. Which of the following test will help you give the diagnosis? Do you remember how do we differentiate between viridins and pneumococcus? Number one, we look for bile solubility. Number two, we look for optochin. Out of those two, I have bile solubility mentioned over here. And I know pneumococcus ends with S. It is bile soluble. That's the answer. Moving on to the next one, the easiest one I can say of the session, Draughtsman appearance of colonies. Right now I told you Draughtsman appearance of colony, also known as the carom coin colonies. They are associated with Streptococcus pneumoniae, not general Streptococcus, Streptococcus pneumoniae. That is nothing but pneumococcus. So yes, that is the final answer that you have. I hope all the set of PYQs seem sorted for everyone. They're not very tough. Staph and strepto seem to be done. In fact, we'll also take up one more set of organisms over here. If you remember, when we started off, the first thing that I told you was all the cocci are gram-positive except Neisseria, Moraxella and Velonella. All of them are gram-positive except Neisseria, Moraxella and Velonella. And now I can finally come to the negative one, means I can come to Neisseria. And this means that I am still dealing with those cocci, but now I have made them gram-negative. So we have two types of Neisseria in front of us, Neisseria meningitidis, which will always cause meningitis, and Neisseria gonorrhoeae, which will always cause gonorrhea, means genital infections. Before I start going with them, are they naturally present in the body? Do they have a natural habitat in our body? Yes, they do. They definitely cause diseases, but they have a natural habitat. Remember, Neisseria meningitidis is commonly present and normally present in the nasopharynx, whereas Neisseria gonorrhoeae is present in the genital region, and this is their natural habitat. So Neisseria meningitidis, near the meningitis, near the brain, you have nasopharynx, and Neisseria gonorrhoeae is for genital. Next, how do you differentiate them? We differentiate them on their ability to ferment something. So please remember, when we are talking about Neisseria meningitidis, it has M as well as G. Means it will ferment maltose as well as glucose. Whereas Neisseria gonorrhoeae only has G, so it will only ferment glucose, not maltose. Repeating, they will ask you that Neisseria meningitidis ferments maltose, glucose both. You will say meningitidis has M also, G also, so maltose and glucose both. Whereas gonorrhea does not have M, it does not ferment maltose, it only ferments glucose. So can I say glucose fermentation is being carried out by both? It is the main differentiating point is maltose fermentation, because maltose is something which only meningitidis will ferment and not gonorrhea. Yes. Moving on, coming to the next, how do they look like? What is the difference in their appearance? The shape over there. So remember meningitis, the way you write meningitis, you write an M. You can make a lens-like appearance out of it. It's a lanceolate or a lens-like appearance, and it has a capsule around it. Repeating, the way you write M, you are going to make a lens out of it and it has a capsule around it. Whereas gonorrhea, the way you write G and G, you can make kidneys out of it. So gonorrhea is said to be the bean-shaped, or it is said to be the kidney-shaped. Remember, the meningitis is lanceolate or lens-shaped with a capsule, whereas bean-shaped gonorrhea is going to be like a G, like a kidney. Out of them, which of them has a capsule? Repeating again, meningitis has a capsule. Moving on, what do they cause? Common sense. Meningitis will cause meningitis and gonorrhea will cause genital infections, that is STD. I don't think you need a mnemonic for that, right? Meningitis will cause meningitis and gonorrhea will cause STD. What else? Meningitis is also famous for causing bilateral adrenal hemorrhage. It is very famous for causing adrenal gland. It causes bilateral adrenal gland hemorrhage. That is also known as Waterhouse-Friderichsen syndrome. Waterhouse-Friderichsen syndrome is something which is bilateral adrenal gland hemorrhage, which is seen with Neisseria meningitidis. Next, Neisseria gonorrhoeae. I know it causes STD. I also know in ophthal that it causes ophthalmia neonatorum in the neonates. How do they get the infection? How do the neonates get this infection? While they are passing through the birth canal. The mother has Neisseria gonorrhoeae, and while they are passing through the birth canal during birth, during a normal vaginal delivery, that is when they get the infection and they present with ophthalmia neonatorum. What else? Neisseria gonorrhoeae can also cause the FHC syndrome. What is FHC syndrome? Fitzhugh-Curtis syndrome. Fitzhugh-Curtis syndrome is nothing but a case of perihepatitis, inflammation around the liver, fibrosis around the liver. Perihepatitis happens in Fitzhugh-Curtis syndrome. There are many other organisms. Even chlamydia can cause Fitzhugh-Curtis syndrome. We will be studying about that eventually. But one organism as of now which can cause it is Neisseria gonorrhoeae. Okay, all said and done. Let's do a quick recap. When I'm dealing with meningitis, obviously it will cause meningitis and Waterhouse-Friderichsen syndrome. Gonorrhea will cause STD, child passing through the canal, ophthalmia neonatorum, and complication Fitzhugh-Curtis syndrome. Coming to the next one, that is what is the media? Which media do you grow it in? Both of them can be grown in a charcoal based media called Thayer-Martin medium. Both the Neisseria, Thayer-Martin medium. If you want to learn something additional, gonorrhoeae can also be grown in the modified New York agar. But what you have to learn is the Thayer-Martin medium for both the Neisseria. And what is the transport media? The transport media is the Stewart's or the Amy's media. The Stewart's or the Amy's media happens to be the transport media. So let's repeat: Thayer-Martin medium for growth. Stewart, do you all remember there was a movie, a cartoon movie, animated movie called Stuart Little? It was on a mouse, Stuart Little. So Stuart Little was always a mouse, always running around, always moving around. So moving around, transportation. Stewart and Amy's media, transport media, and Thayer-Martin medium is the growth medium. Finally, what is any one of them which has any kind of a vaccine? Yes. Remember Neisseria meningitidis, the meningococcal vaccine. The meningococcal vaccine has all these types in it. First, please remember, when we are talking about Neisseria meningitidis has capsule. Like we said, it looks like a lens and it has a capsule. So capsule polysaccharides are type A, type B, type C, type Y, type W135. Repeating, what all is this? We have A, B, C, Y and W135 on the basis of capsule. These are the types of Neisseria. So meningococcus A, B, C, Y, W135. Remember, out of all these, all of them are used in the vaccine, but B is not used in the vaccine. B is not used in the vaccine. Please remember, that's again a PYQ that you guys have got. So let's start probably attempting one or two questions, and I think that will make more sense. The site where Neisseria meningitidis bacteria harbor normally. Remember, Neisseria meningitidis present normally. We said that they are present in skin, genitals, nasopharynx or lower GIT. They are normally present in nasopharynx. So Neisseria meningitidis in nasopharynx, and what's in the genitals? Neisseria gonorrhoeae was present in the genitals. Okay, moving on to the next question. Which of the following is a gram-negative diplococci? Firstly, cocci. Let's pick up the cocci. You'll say, ma'am, right now we've studied Staphylococcus. It's a coccus. We've studied Streptococcus. That is also a coccus. Neisseria is also a coccus. Corynebacterium diphtheria is not a coccus. We haven't studied it as of now. It's a bacillus. So firstly, that doesn't make any sense. Option is ruled out. They've asked me cocci. I know that all cocci are gram positive. But I know that Neisseria meningitidis. What were the three exceptions? Exceptions: Neisseria, Moraxella, Velonella. Neisseria, Moraxella, Velonella, they are gram negative, and that is what they've asked me. They've asked me the cocci, which is gram negative. So Neisseria over here is the best answer that I have. Moving on to the next one. Meningococcal vaccine cannot be used for which of the following serotypes? Do you remember? There were 5 serotypes that we studied. A, B, C, Y and W135, out of which I said which of them is not there for a vaccine, or meningococcal vaccine is not used against which strain? It is not used against serotype B. Coming to the next one. Which of the following is a kidney-shaped bacteria? Again, very, very simple. Kidney-shaped. I just told you there's a G and there's a G and you make a kidney out of it. So, Neisseria gonorrhoeae or gonococcus, Neisseria gonorrhoeae or gonococcus is a kidney-shaped bacteria. As we proceed in bacteriology, we will be doing the shapes of all of these undoubtedly. But as of now, yes, Neisseria gonorrhoeae is going to be a kidney-shaped bacteria. So when we come back, these are all that you had to know with regard to the cocci. Now moving forward, we'll also be going to the bacilli. Now usually I say that all the bacilli are gram negative. All the bacilli are gram negative except McDonald's. So let's start with that. All the bacilli means bacilli will no longer be dot dot. Bacilli will be rod shaped. All the bacilli are going to be gram negative except Mycobacterium, Anthrax, Clostridium, Diphtheria, McDonald NO, Nocardia, Actinomyces, Listeria. I know right now all of a sudden it will feel as if a big list has come on top of us, and we can't learn too much of it, and there's no pressure right now because we are gradually going to read all of these organisms and they'll organically enter into our system. As of now, I know that all the bacilli are going to be gram negative, but there's one bacillus which is I'm going to teach you right now, and that is Corynebacterium diphtheria. So that is what we'll be starting off with next. Please remember, when I'm talking about Corynebacterium diphtheria, it is a bacilli, but it is not gram negative. It belongs to that rare variety, the McDonald variety of gram positive bacilli. Gram positive bacilli, and it has nothing else. It doesn't have any motility. It has no spore forming ability. It has no capsule problem in it. So no motility, no sporing, no capsule, nothing at all. And it's belonging to that McDonald variety. Repeating, all the bacilli are going to be gram negative except for McDonald's. And right now, out of all of those, I'm only telling you to learn D for diphtheria, Corynebacterium diphtheria. So first question that will come to your mind is that how many types of these do you have? Actually, we just call it Corynebacterium. We just call it as a family of Corynebacteria, and everyone please listen to me for say 15 more minutes, and then we'll take up all the questions in the end. First, let us read everything that is important for Corynebacteria, and then we proceed to the questions. So when I'm talking about Corynebacteria, there is something called Corynebacterium diphtheria, which is known as Loeffler's bacilli. Everything about Corynebacterium diphtheria will have the word Loeffler in it because the primary scientist for all the contribution was Loeffler. So remember, Corynebacterium diphtheria, Loeffler's bacillus. Then there are non-diphtheroids. Non-diphtheroids include, listen carefully, Corynebacterium ulcerans. What do I learn? U for ulcerans, and L for which organ can you see that is involved over here? Lung involvement is seen. So Corynebacterium ulcerans, U for ulcerans, L for lung involvement, lung infection. Corynebacterium pseudotuberculosis. So pseudotuberculosis, what do you say? P for, it is also known as the Pries-Nockard bacillus, and S for, this causes infection in animals, that is sheep. Repeating, Corynebacterium ulcerans, U for ulcerans, L for lung involvement. Corynebacterium pseudotuberculosis, P for Pries-Nockard bacillus, S for sheep. And Corynebacterium minutissimum. Corynebacterium minutissimum causes a red erythro. Erythro always means red. It causes a red color rash on the skin. Obviously, skin rash, a red coral red rash it causes. So these are the non-diphtheroids. Right now, I'm focusing on Corynebacterium diphtheria, which is Loeffler's bacillus, and it is said, you'll say, okay, ma'am, what all does it cause? First, I'll tell you what disease does it cause? Common sense. Corynebacterium diphtheria will obviously cause a disease called diphtheria, right? So what is diphtheria? If I ask you that, diphtheria can be of many types. Diphtheria can happen in the respiratory tract. It can happen in the tonsillar location. Along the respiratory tract, it can happen in the larynx. So I'll show you some areas and some pictures which will help you. So the first picture that you see out here, you can see that there is a pseudo membrane that is formed. There is a pseudo membrane that is formed. Can you see in which area? It is formed in the soft palate and the tonsillar area, because diphtheria very classically involves the most common location affected is the tonsillar area, the facial area, the respiratory area. But the most dangerous, you do understand that laryngopharynx, if a membrane, imagine if this kind of a membrane is formed on the larynx, a person would not be able to breathe. There will be respiratory obstruction that will occur, and that will make it life threatening. So if someone asks you which is the most dangerous, dangerous type of Corynebacterium diphtheria manifestation, that is in the larynx. What else? It also involves the lymph nodes and the area of the neck. The neck becomes very swollen and edematous. So you call it the bull's neck. Remember, there's a membrane formed, there is bull's neck formed. The membrane in the tonsillar region is the most common location, and in the larynx is the most dangerous location. That is what diphtheria is. But does that mean that every Corynebacterium diphtheria will end up having resulting in diphtheria in the patient? Means each one of us, whenever this enters into our body, we will land up with diphtheria? No, because all the Corynebacterium diphtheria are not toxigenic. All of them are not carrying the toxin. Some of them carry the toxin. Which toxin am I talking about? Obviously, the tox gene. For toxin production, Corynebacterium diphtheria should have the tox gene. So, for example, these are two Corynebacterium diphtherias that you have. This one has the tox gene, this one does not have the tox gene. This has the toxin gene, so you will say, ma'am, this is going to be toxigenic. It is going to make toxin, it is going to result in infection. And this one you will call as non-toxigenic. So, basically, it's whether they have the tox gene or not. So, for example, this one has the tox gene. This one is toxigenic. It is causing the disease. Can it pass on the gene to some other Corynebacterium? Can it pass it on to this one? This one doesn't have the tox gene. Can it pass it on? Yes. Betaphage, bacteriophage. Bacteriophage can take up the tox gene from here and pass it on to here. What is that known as? Because we studied it in the second part of general microbiology. That is bacteriophage transferring one gene from here to here. That is known as transduction. So can I say that all Corynebacterium diphtheria don't have the toxin? Some of them have the toxin, some of them don't. But if you want to transfer the toxin from one place to another, you can use bacteriophage or transduction. So they will also ask you that how do you get to know? How do you get to know whether you're, if I give you bacteria number 1 and I give you bacteria number 2, this is also Corynebacterium, this is also Corynebacterium. I ask you which of them is toxic and which of them is not. Which of them is toxigenic, which of them is non-toxigenic. What test will you do? This testing methodology is known as LX gel precipitation test. This methodology is LX gel precipitation test. And listen to me very carefully, what are we doing in it? What preparations do I have to do? I will take a filter paper. I'm going to take a brown color filter paper over here, and I'm going to put antitoxin of Corynebacterium, not the toxin. I've put an antitoxin solution on this filter paper and kept it over here. Perpendicular to it, I'll put 3 lines. What are these 3 lines? Over here, I'll put a filter paper, and I know this is not from the patient. It is a known Corynebacterium diphtheria, which I know it has a toxin. That Corynebacterium diphtheria I will put over here. So think logically, the toxin, I know it has a toxin. So the toxin is over here, and on the filter paper, you have anyway put the antitoxin. Will a reaction occur? If the toxin is here and the antitoxin is here, you will say, yes, ma'am, you can see those precipitin lines forming. Those precipitin lines will tell me that reaction has occurred, toxin was there. What have I done over here? Abhi tak I have not gone to the patient. Here I had put a toxigenic strain. Here I have put a non-toxigenic strain. It does not have the toxin. Now do you think any lines will form? This does not have the toxin. Will any lines form? It will not. So first I know that over here I had put a toxigenic strain, so lines had formed. Over here I have put a non-toxigenic strain, so lines have not formed. And now finally in the center I put the unknown, I put the sample. I don't know whether my patient has a toxigenic strain or a non-toxigenic strain, but when I put the sample over here, I saw these lines. Now tell me, if the lines are coming positive, is it a toxigenic or a non-toxigenic strain? You will say, ma'am, it is a toxigenic strain. So have you understood the utility of the LX gel precipitation test? Firstly, it's just a toxin-antitoxin reaction. If you see the precipitin lines, toxin is there. If you don't see the precipitin lines, the toxin is not there. And is the toxin always present? Not at all. Toxin is transported via what? Via a bacteriophage. That's the story of toxin. Now let's look at how it looks under the microscope. So first and foremost, we know Corynebacterium diphtheria is a bacilli. Bacilli will always look lengthy like a rod. Perfect. I have studied all bacilli are gram negative, but Corynebacterium diphtheria belonged to the McDonald mnemonic. So it is gram positive. That is why you can see that it has come out purple in color, because it is gram positive. But do you see some weird shapes? I, V, L, because Corynebacterium diphtheria is very famous for having the Chinese letter or the cuneiform appearance. It is very famous for having Chinese letter or cuneiform appearance. Okay. Further, if you ever have to zoom into it, you will see that Corynebacterium diphtheria looks something like this. You'll see some knobs at the terminal portions. You will see some knobs. I'll show you another picture. Have a look at this. Can you see, pick up any one? Can you see there are 2 prominent round circles at the poles? There are prominent circles that are present. What are those known as? Those are known as forenames. Volutin granules. They are at the poles, so we call them bipolar granules. Babes-Ernst granules and metachromatic granules. There are 4 names, all 4 of them are the names of the same thing. Let's repeat. What are the 4 things? We have volutin granules, bipolar granules, Babes-Ernst granules and metachromatic granules. And what are the stains for them? There are 3 stains that you can use. Pan stains can be used for staining the volutin granules. Ponder stain, Albert stain, Neisser stain. Everyone learn with me. When I want to stain these round granules, I have Ponder stain, Albert stain, Neisser stain, Pan stains are there. What is the picture given here of? The picture here is of Albert stain. What is Albert stain? Albert stain is the Tim stain. What is the Tim stain meaning? Means the first thing in Albert that I will be adding will be toluidine blue. The very first thing that I will be adding will be toluidine blue. The next thing that I will be adding will be iodine. It will be iodine. And the third thing that I will be putting up over here is going to be malachite green. We will have malachite green. So let's do a recap. Number 1, we'll have toluidine blue, a blue purple kind of a stain, then iodine, and then a greenish stain. So remember, the bacteria is going to come green. This malachite green is going to make the bacteria green, and finally what is going to make it purple? The toluidine blue will make the volutin granules purple. Now does it look like this? Let's repeat. If you see, the organism has stained green because of malachite green, and the volutin granules have stained purple because of toluidine blue. So volutin granules or bipolar or Babes-Ernst or metachromatic granules, they are all stained purple because of toluidine blue, and the organism has been stained green because of malachite green. Remember, this is the Tim staining protocol of Albert staining. But other than Albert, what are the other stains that we can use? We can use Ponders, we can use Albert, and we can use Neisser stain. But are volutin granules specific? Are they only and only seen in Corynebacterium diphtheria? No. They are also seen in other organisms. Remember, what is the mnemonic for volutin granules? One thing you will remember, Corynebacterium diphtheria, definitely. But other than Corynebacterium diphtheria, you see it in V, N, G, S. First letter, last letter, first letter, last letter. So, G and S means Gardnerella and Spirillum. Repeating, Gardnerella and Spirillum. V and N, I make a little bit of a change. V is also looking like Y, so yeast, and N is also looking like M, so Mycobacterium tuberculosis. Let's do a recap. We have number one, we have the volutin V, so V looks like a Y, so I say yeast. N looks like an M, so MTB. G for Gardnerella and S for Spirillum, along with Corynebacterium diphtheria. All of them have volutin granules. But if I ask you, what are these granules? If someone asks you, what is the composition of these volutin granules? Remember, they are made up of energy. They have nothing but energy. They are energy sources. They are polymetaphosphate. They are polymetaphosphate. Again, something that could come to you in the potential question, I would say. Volutin granules are polymetaphosphate. I hope that is okay with everyone. So if you want to identify, first you look at it under the gram stain, the Chinese letter appearance. Next you look at the volutin granules, and finally you say, let's grow it on culture media. What are the culture media that you have for the diagnosis of diphtheria? Culture media are two: enriched culture media, selective culture media. We must know the difference. What is an enriched medium in general? Whenever I say something is enriched, will mean you have made it very, very rich. Means I have added a lot of things in this media. I have made it very, very rich. I have given it a lot of food. In this media, I've kept so much of food and so much of nutrients I've kept inside it that this diphtheria, when I put it on top of this media, it will get all that food and grow very, very fast. So when I say I'm dealing with an enriched media, it means that the media has a lot of nutrients, a lot of food, and the bacteria is going to have a feast. The bacteria is going to have a party out there because the bacteria will come across so much of food, so much of nutrients, it will eat quickly, it will become big, fat and grow, and if it grows, I'll be able to see it. That is an enriched media. Remember, enriched, if we see the example that I've given over here is LSS, Loeffler Sedum Slope. Did I tell you in the beginning that everything with diphtheria will have the word Loeffler in it? Corynebacterium diphtheria is also known as the Loeffler's bacillus. So the name of the enriched media for diphtheria is Loeffler Sedum Slope. One more very interesting thing that I'll point out over here. Count the number of alphabets in enriched. It has 8 alphabets. Count the number of alphabets in Loeffler. 8 alphabets. And how many hours does the bacteria take to grow in this? The bacteria takes 6 to 8 hours. Repeating, enriched has 8 alphabets, Loeffler has 8 alphabets. 8 hours is the time taken for the bacteria to grow in an enriched Loeffler medium. Versus a selective medium. Selective means selectively only this will grow. Every other bacteria, staph, strepto, all other bacteria will be suppressed. Selectively, only one thing is going to shine out, and that is Corynebacterium diphtheria. PTA, potassium tellurite agar, which takes very long, 48 hours. And can you see this blackish color? This blackish color is what you get on a potassium tellurite agar. So repeating for all of you guys, enriched 8 alphabets, Loeffler 8 alphabets, takes 8 hours. Selective media is potassium tellurite agar, which takes 48 hours. So if someone asks you which is the fastest media or the faster method of growing diphtheria, the faster method is going to be the enriched media. So when we are doing the testings, let's do a quick recap. First, you do a gram stain Chinese letter. Next, you do the Albert staining for volutin granules. Next, you start growing it on enriched and selective media. Third, you can do the LX gel precipitation test because you want to find out the toxigenicity or the non-toxigenicity. And finally, nowadays no one does it, but Schick test is also a test done for susceptibility. What is this? Schick test, for those who remember, Schick test is a type of a type 3 hypersensitivity reaction. It is done on humans. It is done on humans to find out the susceptibility. Is the human susceptible or is he immune? Basically, you want to find out two things. Is this human being still susceptible to diphtheria? Will he have diphtheria or has he become immune? Is he susceptible or is he immune? That will be determined by the Schick test. So whenever in the exam they say that the Schick test has come out to be positive, what is the interpretation that you would probably take out there? Repeating, when they say in the exam that the positive Schick test is noted, let's start with the questions. Positive Schick test is noted, it indicates that the person is susceptible to diphtheria, that the person is basically susceptible to diphtheria. That is what you have. Okay, moving on, let's practice other questions starting from this. Corynebacterium diphtheria is what? I've already done, it's a bacilli. And I know that all bacilli are gram negative, but McDonald bacilli are different. Gram positive bacillus. It is a gram positive bacillus. PYQ FMG exam. Coming to the next question. Diphtheria toxin acts by. Now this could be something new. I have taught you everything about the diphtheria toxin. Did we teach you something about the tox gene being present or absent and how the bacteriophage can transfer it from one place to another? But finally, how does it act? What is the mechanism of action? Remember, it inhibits the protein synthesis. It inhibits the protein synthesis, and that is how diphtheria happens. Unfortunately, no mnemonic for this. Something that you'll have to mug up. Diphtheria toxin inhibits protein synthesis. Question number 3, FMG PYQ. Which of the following is the most severe form of diphtheria? Which of the following is the most severe form of diphtheria? So I think everyone knows the respiratory involvement, the larynx involvement. Involvement of the laryngopharynx will definitely be the most severe form. Moving forward, Corynebacterium species causing erythrasma. The first thing that I taught you, Corynebacterium species causing erythrasma. So do you remember we had something called Corynebacterium minutissimum? Minutissimum causes that red color, coral red color rash that I was teaching you. Yes. So minutissimum for erythrasma. Next one. PYQ, NEETPG. Presnocard bacillus. Presnocard bacillus, which of these is known as that? So presnocard bacillus, Mycobacterium, Corynebacterium ulcerans, pseudotuberculosis or Bacillus cereus, and the answer is pseudotuberculosis. Do you remember the mnemonic? P and S. P for the presnocard bacillus and S was that it causes infection in the sheep. That is what we had studied. It causes infection in the sheep. Let's see if we have more questions. Corynebacterium diphtheria is arranged in what? It is arranged in which alphabet arrangement? Chinese letter, or Chinese letter I told you also known as cuneiform arrangement. Chinese letter or cuneiform arrangement is what it is seen in. And tomorrow and in the next sessions, I will be telling you the same question again and again because all these are left. So by the end of bacteriology, we should be able to answer all of these that are there. Moving on to the next question. False about Corynebacterium diphtheria. This is a good one. This is a PYQ again from NEETPG. False about Corynebacterium diphtheria. Also the last question of this session. Deep invasion is not seen. Did you see, was there a membrane forming superficially or was it going inside the larynx, going inside the tonsils? No. You said that, ma'am, there was a superficial pseudo membrane that was formed. So it remains superficial only. Deep invasion is not seen. That is correct. It remains superficial. It does not go deep. Next, LX gel precipitation test is done for knowing toxigenicity. Yes, do you remember that filter paper wala? And I told you whether the lines will form or not. Is it done to know the toxigenicity? Correct. Metachromatic granules are seen. Again, correct. Did we study volutin granules, known as Babes-Ernst granules, known as bipolar granules, known as metachromatic granules? Yes, metachromatic granules are seen. Last one. Toxigenicity is by chromosomal changes. No, does chromosome determine it, or is bacteriophage going to actually transfer it? Toxigenicity is by transfer from a bacteriophage, means by which genetic technique toxigenicity is dependent on transduction. So which of the following statements was false over here? Option number D was the answer over here. Well, with that, I think it's been quite an extensive session. We've done a lot of bacteria from staph, entire family of strepto, Neisseria, meningitis, gonorrhea, entire Corynebacterium is done. And in the next session, this is exactly what we'll be starting with. We'll be going ahead with the spore-forming bacilli. So we've got bacillus species, many of them, and a lot of Clostridium species also coming up. So yes, that will be what the next session will be, along with many other bacteria as well. We'll be wrapping it up in the next session. So yes, that is how it goes. Thank you guys for joining in. I hope you guys are benefiting from the same. I've given you a tiny little homework of reading the revised Jones criteria. I hope you'll be doing that with a thumbs up in the chat box that you've done it. And I'll be meeting you in the next session with many more bacteria and a continuation of the microbiology crash course. Thank you so much, and a recap PDFs are available on the Telegram groups and channel, and the link for that is already available in the description below, so please you can download it from there. Thanks a lot. Have a great day. Study well. All the very best. Hi everyone, welcome back to the YouTube session of the Microbiology Crash Course. For all of those who are new out here, I'm Dr. Preeti Sharma, and I am an educator for pathology and microbiology. Well, we are continuing with the Microbio Crash Course for the upcoming NEET, NEXT and FMG exam students, and we've already completed 4-5 essential components of this particular crash course. We finished with 2 parts of general microbiology, we finished with a very important section that is fungus or mycology, and we've also finished a portion of bacteriology. Today we'll be taking the bacteriology forward and dealing with the other bacteria that come under it. So for all of those who've probably missed out on the previous videos, they are available on the platform. They are available under the video section. There's a separate playlist that is there, so all of you can certainly look at all the microbiology videos from there. Also, as with regard to the PDF of this particular, all these classes that are going on, the PDFs of these will be available on the Telegram group. I've put the link to the Telegram group in the description below. The Telegram channel and the group, the description below has the name. It goes by the name of Pathology by Dr. Preeti Sharma. So you can follow me on the Telegram or Instagram or Facebook groups, and you would be able to download the PDF from Telegram and Facebook. Well, having said that, let's begin with the bacteria. And the set of bacteria that we have to cover today become the spore-forming bacteria. When I say spore-forming bacteria, the ones which make spores, there are 2 that come under it. One is going to be Bacillus. Bacillus, having the A immediate to it, is an aerobe. It requires oxygen. And Clostridium is claustrophobic, means less oxygen. So Clostridium happens to be an anaerobe. So please remember, first and foremost, Bacillus, A, so it's an aerobe, and Clostridium reminds us of claustrophobic, so that is an anaerobe. So let's begin with the very first question, and then we'll study accordingly. Question number 1 out here, guys. Which of the following is not a virulence factor of Bacillus anthracis? Which of the following is not a virulence factor? Labile toxin, edema toxin, lethal toxin, or capsular polysaccharide? And the correct answer to this is labile toxin is not a virulence factor. So you would obviously ask me, what are the virulence factors that are there for Bacillus anthracis? So remember, Bacillus anthrax, that you commonly know it as, it has 2 things. Number 1, it has a capsule, and number 2, it has a toxin. Both of them are responsible for its virulence. Now, I think when we were studying the first part of general microbiology, we studied that all capsules are made up of polysaccharides. I think that's a very famous thing that we studied. All capsules are made up of polysaccharides except Bacillus anthracis. That is where it comes across as an exception, except Bacillus anthracis, where the capsule is made up of polypeptide. Other than that, all other capsules in the subject with all the bacteria, they are made up of polysaccharide. saccharides. What about the toxin? The toxin has three parts to it, which you call as the EPL. So remember, many students learn it as the English Premier League. So if you wish to learn it that way, what is EPL? Three parts of the toxin. So remember, EPL have to work together. EPL have to work as a combination. Only then the toxin is going to make its activity. So what is EPL? E for edema factor, P for protective factor, and L for lethal factor. So E for edema factor. Edema means how does it cause edema? By increasing the cyclic AMP. It causes edema. It makes sure that the bacteria is protected. It makes sure that it can cause killing. It can cause destruction. That is lethal. Remember, edema, protective, lethal. EPL are the three factors. Now when I come back to the question, which of the following is not a virulence factor? So is the capsule a virulence factor? Yes. And what is the capsule of Bacillus anthracis made up of? It is made up of polypeptide. It is that exception. So it is a virulence factor. Other than that, EPL are virulence factors. So E for edema toxin, P for protective toxin, and L for lethal toxin. Basically, there is nothing known as a labile toxin, and that was the question that you had. Now that you've understood everything about anthracis, why don't we study all the other things associated with it? How many types of anthrax are known? Very easy. Once you know this, you'll be able to answer a lot of questions. There are three types of anthrax that we have. The anthrax affecting the skin, so I call it cutaneous. Anthrax affecting the lungs, so I call it pulmonary anthrax. And anthrax affecting the intestines, so we call it intestinal anthrax. Simple. Cutaneous, pulmonary, intestine. So how do these happen? How does the bacteria enter our body? How does it cause skin and lung and intestine involvement? So for skin, remember, we call it Hyde-Potter's disease. Have you all seen the workers who carry animal skin? I'll show you a picture. Have you seen that whenever the workers carry animal skin, they always carry it on their neck and on their shoulders. So as if they are carrying a rug, as if they are carrying a bag, right? So whenever we have to carry something, we carry it on our neck and our shoulders. That is the area. So imagine if they are carrying animal skin, and this is definitely coming from the animals. So if they are carrying the hides and the skins of the animals on their bare backs, so can you say that that is why this is referred to as the Hyde-Potter's disease? It is known as Hyde-Potter's. What is Hyde-Potter? Hide is nothing but animal skin. And porters are people who carry those heavy things on their back. So these are the porters. They are the people who are carrying animal skin, and that is why they have this blackish color lesion. And this will be a classical history. That blackish color lesion will either be on the neck or the shoulder or the upper back, because that is the area where they are carrying all of those animal skins, right? So please remember, cutaneous anthrax known as the Hyde-Potter's disease. Coming to the next one, pulmonary anthrax. Why would someone have pulmonary anthrax? Pulmonary means inhaling. Pulmonary means inhaling, right? We would inhale it. Why would we inhale something? People who work in the wool industry. So wool has very thin fibers, right? So these fibers are inhaled. Wool sorters. So all the workers who sort wool, who work in the wool industry, when they inhale these fibers, it reaches the lungs and it causes pulmonary anthrax. It causes pneumonia. Please remember, this is the form of anthrax which is also an agent of bioterrorism, because inhalation definitely comes under bioterrorism. Lastly, if someone has uncooked meat, if someone is having consumption of uncooked meat, which kind of anthrax would occur in that case? Intestinal anthrax will occur. Intestine inflammation will occur. Repeating, if they carry the skin of the animals on their back, cutaneous anthrax. If they inhale it, it is known as pulmonary anthrax. If we eat animal material, it is intestinal anthrax. Which out of these results in the formation of a malignant pustule, or also known as eschar? Malignant pustule, also known as eschar. So remember, this is a cutaneous finding. Do you remember I showed you this blackish color lesion on the skin? So this blackish color lesion on the skin is known as a malignant pustule or an eschar. You will ask me that why are you calling it malignant? Does it have anything to do with cancer? No. I'm calling it malignant because look at how it's appearing. The appearance is pretty bad. It is quite invasive. It is quite dirty looking, blackish, eroded. Only because of the bad looking appearance, I call it a malignant pustule. Otherwise, it has nothing to do with cancer as such. Okay, so the three types of anthrax are pretty much sorted: the cutaneous one, the pulmonary one, and the intestinal one. Let's come to the diagnosis. When you look at it under the microscope, you can very well see that yes, these are kind of bacilli. They are long, they are bacilli. And do you see that if, for example, if this is the organism, you will say that in the center, you are seeing a whitish area. There is the area that is not picking up the stain. What is that area? That area is nothing but the spores. Did we just study that Bacillus and Clostridium are spore forming organisms? So please remember guys, when you are dealing with this, either look at the appearance, look at the kind of appearance. Either you can call it the boxcar appearance, or you can also call it the bamboo stick appearance, because it looks like this, like cars, box cars. Either you can call it box car appearance, or you can call it a bamboo stick appearance, and you see it on a gram stain. If you try to grow it on culture media, on agar, you get a medusa head. What is a medusa head? This, you won't get a picture, but definitely something that is important to know. If you grow it on an agar, you get a medusa head appearance. Number 3, if you grow it on gelatin, if you put Bacillus anthracis inside gelatin, it will give you a very important finding. See, have a look at it. This is a test tube. This test tube, what have I filled it with? This test tube has been filled with gelatin. Do you see what has happened to gelatin? Gelatin is broken over here. It is broken a lot over here. So I can say gelatin has been liquefied. It has been broken. Where has the gelatin been liquefied more? It's maximum at the surface. Maximum gelatin is broken at the surface, and as you are going down, the amount of gelatin breakage is decreasing. So can I say Bacillus anthracis is acting maximally on the surface and least at the bottom? Why? It is breaking gelatin maximally at the surface and least at the bottom. Why? Because oxygen is present at the surface and oxygen is not present at the bottom. And is anthrax aerobic? Yes, that is the first thing we studied. Anthrax is an aerobic organism. It works in oxygen. On the surface, it's getting oxygen. It will break the gelatin. On the lower part, it's not getting oxygen, so it will not break the gelatin. And what kind of an appearance do I get out here? We get the inverted fir tree appearance. It's an opposite tree, upside down tree, inverted fir tree appearance. Number 4, what does Bacillus anthracis show us on a penicillin agar? PE for penicillin, PE for pearls. Remember, on a regular agar, if you guys remember, on a regular agar, it was showing us medusa head appearance. On a penicillin agar, it's showing us a string of pearls appearance. Next, what is the selective media? Something that we'll have to mug up. The selective media is the plett media. And what is the antigen antibody test that we can do, which is hardly done nowadays? It's more for MCQs. The antigen antibody testing is the Ascoli's thermoprecipitant test. So Ascoli's thermoprecipitant test also happens to stand for anthrax. So let's do a recap of all 6 things. Number 1, on gram staining, it looks like boxcar or bamboo stick. On agar, it shows you medusa head appearance. On a penicillin agar, it will show us pearls, string of pearls appearance. On gelatin, it shows us inverted fir tree appearance. Selective media is plett, and Ascoli's antigen antibody reaction is also for anthrax. Now that we've studied everything, why don't we start doing a few questions? All of the following are properties characteristic of Bacillus anthracis except motility on wet mount examination, medusa head colonies, poly-D-glutamic acid capsule, and boxcar appearance. All of the following are characteristics. So firstly, did we see the boxcar appearance? Yes. Either you can call it boxcar or you can call it the bamboo stick appearance. Does it have a poly-D-glutamic acid capsule? Anthrax. Yes, it had a poly-D-glutamic acid is what? It's nothing but protein. It's nothing but polypeptide. And does it have a polypeptide capsule? Definitely yes. Does it show you medusa head colonies? Again, yes. So what does it not show you? I have not told you that it's motile. It's not at all motile. I've not shown you any feature of motility out here. So it is a non-motile organism. This was the first question. Let's practice a few more. Moving on to the next one. Which of the following is not a cultural characteristic of Bacillus anthracis? Medusa head, string of pearls, inverted fir tree, bamboo stick. You'll say all of these are features of Bacillus anthracis. I asked you which is not a cultural feature of Bacillus anthracis. Culture. So if you see, Medusa head is on agar, it's on a culture plate. String of pearls is on what kind of culture plate? It's on a penicillin agar. So that is also a culture characteristic. Inverted fir tree appearance is on which agar? It's on gelatin agar. So that is also a culture characteristic. Bamboo stick appearance is not on a culture. Bamboo stick appearance is on gram staining. So that is why they had asked you which of these is not a cultural characteristic. Bamboo stick appearance is a characteristic but not a cultural one. It's a gram stain characteristic. Moving on to the next question. Which disease occurs after eating uncooked meat? I think it's very, very obvious over here because of the bias also. With uncooked meat, Bacillus anthracis, and which type? You had three types of anthrax: cutaneous, pulmonary, intestinal. So, but obvious, there's going to be intestinal anthrax. Intestinal anthrax is what is going to be associated with uncooked meat. Okay, having said that, let's move forward. Something that we studied probably in the previous session as well. The organism responsible for toxicity due to Chinese fried rice. The organism responsible for toxicity due to Chinese fried rice is going to be, so I told you a mnemonic, that Chinese fried rice and cereus. We are now moving on to the discussion of Bacillus cereus. Please remember, it's the food poisoning one. Where have I taught you this earlier? Where did we study that there are two organisms, Staph aureus and Bacillus cereus? Staph aureus also causes food poisoning in less than 6 hours, whereas Bacillus cereus also causes the same in less than 6 hours. So they are both causes of early food poisoning, short incubation food poisoning. When I talk about Bacillus cereus, one thing is certain, that there is food poisoning that is involved, and we keep studying it is to do with Chinese food, Chinese fried rice. So cereus for C, C for cereus, and C for Chinese. But actually this food poisoning is of two types. One in which the patient comes to us with vomitings, and the second food poisoning is in which the patient comes to us with diarrhea. So there are two kinds of food poisoning. Remember, the vomiting one has a Chinese fried rice and a short incubation period. It's not the diarrheal one. The diarrheal one has a lengthy incubation period of 8 to 16 hours, and it is by consumption of probably poultry and vegetables, so solid food, not just Chinese food. Remember, easier way of learning it, emetic. So when we eat food, the shorter route of the food coming out is vomiting. The longer route of the food coming out are the stools. So the shorter incubation period is for the vomiting type, and the longer incubation period is for the diarrheal type. So remember, when I say that Staph aureus and Bacillus cereus have the same incubation period, that is less than 6 hours, I hope you've understood that which kind of Bacillus cereus am I talking about. I'm talking about the emetic type. I'm talking about the emetic type. Okay, so yes, that was the question. Chinese fried rice, Bacillus cereus. Let's move on to the next. The following are true about Bacillus cereus introduced or induced diarrhea, except. So first and foremost, they're talking about Bacillus cereus, and which type of it? They're talking about the diarrheal type of it. Okay, and which of the following are true except? So is it transmitted by parenteral route? Incubation period is longer. It is caused by enterotoxins. It is usually associated with fried rice or Chinese food which is reheated. So please remember, I know that when I'm talking about Bacillus cereus, be it the emetic type or be it the diarrheal type. See, for example, Chinese food can go in both, right? Because emetic type is also Chinese food and diarrheal type is because of meat and veggies. Meat and veggies also ultimately goes in Chinese food only. So both of them are caused by Chinese food. So is it associated with fried rice or Chinese food which is reheated? Definitely yes. If it is causing food poisoning, it has to have a toxin which is going to be enterotoxin involving the intestine. So that is also true. For the diarrheal type, is the incubation period longer? Yes. If you remember correctly, for the emetic type, it is less than 6 hours, whereas for diarrheal type, it is approximately 8 to 16 hours. So definitely for the diarrheal type, it's longer. Can it be transmitted by parenteral route, means via blood or needles? No, obviously, that is not sounding right. When it's causing food poisoning, it's not something that will go via blood or get transmitted via needles. It's definitely going to be eating. So it's going to be definitely a form of eating or oral consumption. If you want to know that what is the selective media, you don't have to know the full forms. That's the good news. I've only got to the full form because somewhere for your knowledge you can know. But MIPA and PEMBA. Remember, when we are talking about the lab diagnosis of Bacillus cereus, three words should remain in your mind. Firstly, it is motile. It can move around. That is how it's causing diarrhea also, vomiting also. It's moving around. And the two selective media are MIPA and PEMBA. As I said, knowing the full form is not important. If you wish to know, it's on the screen in front of you, but again, not important. MIPA, PEMBA, mobile. These three words come to you. Bacillus cereus is done. I hope that's okay with everyone and we can move on forward. If you remember, all the organisms under Bacillus that I was talking about, be it anthrax or cereus or all the others that we've spoken earlier, they are all aerobic. They need oxygen, whereas the ones that come under Clostridium are anaerobic, and that is exactly what I'm starting with next. All that come under Clostridia are anaerobes, so this means they do not need oxygen. They do not need oxygen. So what is the classification? I know right now, as I said, sometimes we see some tables in microbiology or even other subjects like a biochemistry or a pharmacology, and it's kind of overwhelming because it gives you too much of information in a text manner. Whenever you come across anything like that, the best way is to segregate it into shorter tables, make your mnemonics, visual memory, image-based mnemonics, and that's the only way of cracking such things. So let's try and decode this table also. What have they written about Clostridium? They have said that, what all do you know? Do you know Clostridium is a spore-forming organism? Yes, we know that. Clostridium will make spores. Where are the spores present? Let me show you a picture of spores. Can you see that this is an organism and then there's a round spore over here? Similarly, this is a bacteria, then there is a round spore over here. So basically, they ask you the location of the spore. Look at these 4 pictures. They will tell you the location. Where do you think is the spore located over here? You'll say right at the tip. Right at the tip. Over here also, right at the tip. So can I say in these two, the spore is terminal in location. The spore is terminal. Okay, where is the spore over here? You will say exactly in the center. So one more is that the spore can be central. Next, you will say, ma'am, here also it's at the end, but not exactly at the tip, a little away from the tip. So can I say this is subterminal? What are the 3 locations, guys? Either right at the tip, terminal spore, right in the center, central spore, a little away from the tip, and that is what you'll call as a subterminal spore. Remember, the most common out of these are going to be subterminal. The most common of all. So if you say, for example, Clostridium perfringens, anything or the most common spore that you can have is subterminal. But for Clostridium tetani and Clostridium tertium, they have TE in them. For Clostridium tetani and Clostridium tertium, we have TE and TE for terminal. Very simple. Tetani and tertium have terminal spores. Then you'll come back and you'll say that fine, this is also terminal and this is also terminal. What's the difference? So I'll say that one of them, I'm very confident that this one is Clostridium tetani and this one is Clostridium tertium. You'll say, ma'am, both of them are terminal. How did you get to know? Because please remember, when you're writing tertium, you come across TER. Tertium gives us a tennis racket appearance. Tertium gives us a tennis racket appearance. TER for tertium, tennis racket. So tennis racket will always look something like this, a little long spore, like a badminton racket or a tennis racket. Whereas when you're talking about Clostridium tetani, this is going to be a drumstick appearance. Has everyone seen drumsticks? Drumsticks are round. If you look at the organism, the spore will be round like this, like a drumstick. So remember, when you see a round spore at the terminal, it's drumstick like tetani. And when you see this oval spore at the terminal, that is tertium, tennis racket appearance. What is common to them? TE for terminal, TE for terminal. But I hope you remember, TER is the tennis racket appearance. Coming to the next. When is the spore in the center? Means when is the spore right in the center? Whenever I put anything in the center, I'm dividing the organism into 2 parts. I'm creating a bifid. Remember, bifermentins has a central spore. So let's do a recap, guys. All the spores in all the organisms are usually subterminal, but in Clostridium tetani and in Clostridium tertium, the spores are going to be terminal and terminal. Whereas in bifermentins, dividing it into by fid, the spore is central in location. I hope the first part of the table is done, because now we move on to the next part. Clostridium species are unencapsulated and they are motile. What did I learn? Clostridium is not going to have a capsule and is no inhibition. It has no capsule and is going to move around everywhere. It has no capsule and it's going to move around everywhere, except exceptions are asked. So what do I want to tell you? When you started off your journey, NEET UG days, get back to NEET UG days, get back to 11th and 12th class, get back to those days where what were your subjects? Physics, chemistry and biology. Physics, chemistry and biology. For which exam did you take up PCB as your subjects? You took it up for an exam called Pre-Medical Entrance Test, PMT. So that's your mnemonic. You took PCB for PMT, right? So what is that? What am I trying to say? All the Clostridia are unencapsulated, but which are the ones which are capsulated? Which are the ones which have a capsule present in it? You will say Clostridium P for perfringens and Clostridium B for butyricum. So remember, PCB tells you Clostridium perfringens and Clostridium butyricum, they are capsulated, rest all of them are unencapsulated. Coming to the next, I told you all of them are motile, all of them can move here and there. But PMT, which are the ones which are non-motile, which don't have motility? Again, P for Clostridium perfringens and T for Clostridium tetani type 6. Repeating, perfringens and tetani type 6 are non-motile. So I would want to do a very quick recap. The two mnemonics that I have with me are PCB. And why did we take PCB? Because what did we want to crack? We wanted to crack. So for crack, I'll write this. We wanted to kill the exam PMT. So I took PCB because I wanted to kill PMT. But then there were some of us who were a little overconfident in life, and I would include myself also in those set of students who, apart from PCB, also took maths, who also took mathematics, who took math because they thought that why not even IIT, or why not keeping all my options open, or why not getting a better score. And all of us know that if we were inclined towards PCB, then our math was horrible, and I really don't know why all of us were so over ambitious and still took it up. And also, just in case you were one of those, please type it and let me know, so that I know that I was not the only foolish person who decided to do that when I knew that my aptitude was for PCB, and then I burdened myself with mathematics only to keep my options open. So in case you're also of the same, you've been through the same, let me know. Anyway, so we took PCB and we wanted to kill PMT. So let's see. All the Clostridia species are unencapsulated, but which is the one which is going to have a capsule? Capsule is going to be present in Clostridium perfringens and Clostridium butyricum. Okay, next, which of them did not, or which of them, or all of them motile? Definitely all of them are motile. But which are the ones which are going to be non-motile? The non-motile ones are again going to be Clostridium perfringens and Clostridium tetani type 6. That's the final summary. So I hope all of you are okay with the final summary, and we can probably attempt a question. Which Clostridium species is capsulated? As soon as you get this, remember capsulated, PCB. Capsulated which all? Perfringens, yes, capsulated. Butyricum, yes, capsulated. So what is the correct answer? The correct answer is both A and B are capsulated. So I hope finally these three tables are making sense. The spores, the capsule, the motility, because now we can move on to the next. Gas gangrene is caused by all except. This was a previous year question in the FMG exam. Gas gangrene is caused by all except. So the answer is, I think, even if you don't know gas gangrene, but you know for a fact that tetani will cause tetanus. Tetani will obviously cause tetanus and not gas gangrene. So definitely gas gangrene is caused by all the others. So let us start with this. What all am I talking about? Gas gangrene most commonly in 60% of the cases is Clostridium perfringens. Remember, in the earlier days, Clostridium perfringens was also known as Clostridium welchi. Clostridium welchi and Clostridium perfringens is the same. It causes gas gangrene. What do I mean by that? Gangrene means something is going to die, which means the muscle is going to die. There is myonecrosis that is going to happen. There is myonecrosis that is going to happen. So gas gangrene, which is going to be seen in 60% of the cases by Clostridium perfringens. Other than that, septicum and novi. Septicum and Clostridium novi, they also cause gas gangrene. Repeating, we have perfringens, then we have septicum, and then we have novi. All of these are responsible for gas gangrene. Out of these, obviously the question that you'll get will be maximally on perfringens. So let me come back to the question. Again, like I said, what all causes gas gangrene? It's caused by perfringens, it is caused by septicum, and it is caused by novi. Perfringens, septicum, novi. So tetanus does not cause it. Out of these, they will definitely ask you a question on the lab diagnosis of Clostridium perfringens, and I can see a lot of things written in front of me. And yes, that is exactly what is important. So one by one, we are going to come down to everything about Clostridium perfringens. Rather than seeing it like this, I would show you one by one as separate photos. I think that will be a better thing to do, right? So first and foremost, whenever I talk about Clostridium and it reminds me of claustrophobic, claustrophobic, the thing that comes to your mind is anaerobic. It does not need oxygen. And anything that does not need oxygen will be grown in an anaerobic culture media. What is the anaerobic culture media where there is no oxygen present? So anaerobic culture media is RCMB, Robertson's cooked meat broth. RCMB, that is Robertson's cooked meat broth, that is not having oxygen. That is for all the anaerobic organisms. Point number one. Point number two, it shows us, I'm going to now tell you many fancy names. It's going to show us target hemolysis. What is target hemolysis? Have a look at this. Target. So all of you have played that dartboard game. Of course you have, I'm sure. The archery and the dartboard games. So you know for a fact that there's a bullseye that you always have to hit. I still have one of these in my room because I think it's a game which my childhood went away, but this did not. And this is still a very famous pastime for me. And I think many of you also. So now we have magnetic ones as well and many others, which is a different discussion that we'll have some other time. But yes, I'm sure everyone's seen a dartboard and you've seen those bullseye archery boards. So they have a lot of lines around it so that you can hit in different zones, right? Similarly, the kind of blood breakage that is caused by Clostridium perfringens is target hemolysis, which basically tells you that you can see one clearing here and another clearing around it. Similarly, one clearing here and another clearing around it. So there's a double zone of hemolysis, also known as the target hemolysis, which you see with Clostridium perfringens. That is the next point. Coming to the third one, Nagler's reaction is positive. What is Nagler's reaction? Let's have a look at this picture out here. We call it a Nagler plate. What is a Nagler plate? It's basically a culture plate which is going to have egg yolk in it. It's basically a culture plate which is going to have egg yolk in it. Why have we added? Why is this whitish? Because of egg yolk. Egg yolk is something which is very rich in lecithin. Egg yolk is extremely rich in lecithin. So basically there's a lot of lecithin out here. And from the patient, I have added Clostridium perfringens. Can you see what has happened? All the area around Clostridium perfringens has become white. Why? Because please remember, Clostridium perfringens has something called alpha toxin. And do you know what is alpha toxin? Alpha toxin is lecithinase. Alpha toxin is lecithinase, right? So please remember, lecithinase will go and break the lecithin. Lecithinase will go and break the lecithin, and that is why you get an opaque appearance over here. So that is the Nagler's reaction. Repeating, Nagler's reaction is due to which toxin of Clostridium perfringens? It is because of the alpha toxin. And what is the other name for alpha toxin? Alpha toxin is also known as lecithinase. Coming to the next one, and that's the reverse camp test positive. Before I start off with this, I hope everyone remembers from our basic introduction in the previous session that there was something known as a camp test. And CAMP test was for an organism known as Streptococcus agalacti. It was for an organism called Streptococcus agalacti. Now I'm saying reverse CAMP test, which is for Clostridium perfringens. And now this is known as reverse CAMP test. So what do I know under reverse CAMP test? Vertically, I have put Clostridium perfringens. Horizontally, I have put Streptococcus agalacti. So please remember, always. Now I'll tell you the difference. We'll draw two images so that we know the difference between a CAMP test and we know the difference between a reverse CAMP test. These two is what we want to know. So what is CAMP test versus reverse CAMP test? Remember, in both of them, and I'm repeating this again, in both of them, the vertical line and a horizontal line will be there. There will be a vertical line and there will be a horizontal line. The horizontal line will always be of Streptococcus agalacti. The horizontal line will always be of Streptococcus agalacti. It's the vertical line that will vary. If the vertical line is Staphylococcus aureus, this means you are dealing with a case of, or you are performing a case of CAMP testing. You are trying to go in for CAMP testing. On the other hand, if the vertical line has something known as Clostridium perfringens, then you've done a reverse methodology and gone in for reverse CAMP test. So basically you realized that the horizontal line is always going to be agalacti. Agalacti is always going to be horizontal. The vertical line in CAMP test is Staphylococcus aureus, whereas in reverse CAMP test, the vertical line is Clostridium perfringens. And over here, the vertical line was Clostridium perfringens, so this happens to be the reverse CAMP test. Coming to the last one, it shows us stormy fermentation with litmus milk. Although no one will ask you this picture, but can you see that there is a litmus milk and there is some stormy bubbliness that you see at the top? Like I said, no one's going to ask you this, but with litmus milk test, stormy clot reaction is noted. Let's do a recap. We have to grow it in Robertson's cooked meat broth. Target hemolysis is seen. Nagler's reaction is going to be positive. Reverse CAMP test is noted, and the stormy fermentation with litmus milk is what we see out here. So I hope that's okay. Clostridium perfringens, all fancy names coming up your way. Okay, let's move on to the next one. Maybe you can answer this question now. Nagler's reaction is predominantly associated with which toxin of Clostridium perfringens? Nagler's reaction is predominantly associated with, so if you remember, Nagler's reaction was the one that was utilizing egg yolk. And if I have egg yolk, how will I break egg yolk? So you had studied that egg yolk has lecithin, and if you want to break egg yolk, you will have to use lecithinase. Now, which of these was lecithinase? Alpha toxin of Clostridium perfringens was lecithinase. Do I have any of these two in the options? Yes, I have lecithinase in the options, and that becomes my answer. Let's move on to the next question. Which of the following is incorrect about the toxin leading to the disease below? Which of the following is incorrect about the toxin leading to the disease below? So this is the disease that has been shown. First we'll identify this. It looks as if the man is smiling. This condition is known as rhesus sardonicus. This condition is referred to as rhesus sardonicus, which basically means that it looks as if, is the patient actually smiling? Well, definitely not. This is a classical case of tetanus, and obviously once if a patient has tetanus, he's definitely not smiling. But why does it look like as if he's smiling? Because all the facial muscles, all the muscles have basically contracted. So there's a contraction, there's a spasm that has occurred. This results in spasm of the muscles, and once all the muscles will undergo spasm, all the muscles of the face, they undergo spasm, it looks as if the patient is grinning or smiling. That is known as rhesus sardonicus. So if I want to answer this set of questions, I will need a little bit of theory base for myself. When I'm dealing with Clostridium tetani, it has two toxins which are responsible for everything. It has two toxins which are responsible. Number one is known as tetnolysin and the other is known as tetnospasmin. Repeating, tetnolysin, tetnospasmin. If I say that ultimately there has to be spasm of the muscles have to contract, there's going to be a muscle spasm, which of them would ideally be the one that will cause it? You will say obviously the spasmin one will have the main role. The main role in causing the muscle spasm is going to be technospasmin. And how does it act? At which level does it act? It at a neuromuscular junction. At which level does it act? It acts presynaptically. Does this remind you of some very famous question that you get in forensic, where there's another poisoning that also causes the same kind of muscle spasms, but that doesn't act presynaptically, that acts postsynaptically? I hope you all remember the strychnine poisoning. Strychnine poisoning, which is again definitely a question that you can, anytime get in the exam. Strychnine poisoning is something that they will tell you for postsynaptic attack. So that is going to cause postsynaptic effect, and then it is going to cause spasm of the muscles. So please remember, for tetanus it is presynaptically. Tetanus is going to be presynaptically. And who is the main one causing it? Tetnospasmin is causing it. So how do I differentiate between tetnolysin and tetnospasmin? Because both of them are heat labile. Both of them are heat labile, but their oxygen affinity varies. Remember, tetnolysin OL versus tetnospasmin. So tetnolysin is oxygen labile and tetnospasmin is oxygen stable. Repeating, tetnolysin is oxygen labile and tetnospasmin is oxygen stable. Let's come back and let's start reading it. Which of the following is incorrect? Are both of them heat labile? Definitely true. Okay, is it oxygen stable? So the toxin which is leading to spasm, I am talking about tetnospasmin. I'm talking about tetnospasmin. Means, is it oxygen stable? Yes, it is oxygen stable. Does it act postsynaptically? Think again. That is the false statement. I just told you that when we are dealing with a case of tetanus, it does not act postsynaptically, it acts presynaptically. And what does it do? It definitely, I think everyone knows this much of physiology, that it inhibits the release of GABA. It inhibits the release of GABA and it acts presynaptically. That is why there is a spasm of the muscles that is created. Well, okay, having said that, I suppose we can move forward and go on to the next set of questions. Botulism. Botulism causes what? Descending flaccid paralysis, descending spastic paralysis, ascending paralysis, or ascending spastic paralysis. I think in terms of the answers, botulinum or botulism causes descending flaccid paralysis. And which organism am I talking about over here? I'm talking about Clostridium botulinum. In fact, I think it's a very, very obvious kind of a question that when someone asks you that for botulinum, what is the toxin that you have to know? The toxin is the botulinum toxin. And in fact, guys, if you've read a little bit of the past literature, you would know that the most toxic substance, which is the most lethal to mankind, the most lethal and the toxic substance to mankind is Clostridium botulinum, or this botulinum toxin. And what does it cause? It causes food poisoning. It causes food poisoning. Number one, it causes paralysis. Number two, repeating, what does Clostridium botulinum cause? Number one, it causes food poisoning. Number two, it causes flaccid paralysis. So remember F and F. Clostridium botulinum causes flaccid paralysis and food poisoning. Obviously, if it causes food poisoning, what kind of a toxin does it have? Intestine, food poisoning, enterotoxin. And what kind of food does it grow on? It tends to survive in canned food. And all the students who are living in foreign countries and who consume a lot of these canned foods in hostels, always remember that there is that's the reason we check for the expiry date. And even if it is within the period of consumption, even if it has not reached the expiry date, but you must have seen that they've always written on the can that if the can appears damaged, or if the can appears very swollen, or if it's become rounded and there's some gas kind of production, or they say if you see any bubbles on the food, you should not consume it. Primarily why? Because Clostridium botulinum tends to grow on canned food. So whenever you you open a canned item, even if it's not reached the expiry date, but the can is looking very swollen, or if you see bubbles on the surface of the food as soon as you open it, do not consume that. Okay, so enterotoxin on the canned food causes food poisoning. Similarly, it causes flaccid paralysis. And obviously, if it causes paralysis, which toxin is it? Neurotoxin. So finally, what are the two toxins of Clostridium botulinum? One of them is going to be neurotoxin. One of them is going to be enterotoxin. The entero one will cause food poisoning and the neuro one will cause flaccid paralysis, and that is the answer to the question. It causes descending flaccid paralysis. So can I say in other words that tetanus was something that was causing spastic paralysis, it was causing spasm, and botulinum is something that is going to cause food poisoning and flaccid paralysis, FNF? Well, having done that, let's move on to the next question. A hospitalized patient developed severe watery diarrhea and dehydration following a long-term course of antibiotics. The histopathological image is shown below, and which of the following toxins are responsible for the production of this condition? So firstly, you have a case of diarrhea, so this means intestine is involved. So much of diarrhea has happened that obviously patient has become dehydrated, and this is a very important history given next. This has happened after a very long-term course of antibiotics. Remember, after long-term usage of not just cephalosporins, not just third generation cephalosporins, but any kind of antibiotics. After a long-term usage of antibiotics, what is the patient at a risk of? Patient is at a risk of Clostridium difficile infection leading to PMEC. Pseudomembranous enterocolitis, remember, leading to PMEC by Clostridium difficile. So they will always give you a long-term usage of antibiotics or cephalosporins. That is why the patient will have Clostridium difficile infection. That is when the patient will have pseudomembranous enterocolitis. How does pseudomembranous enterocolitis happen? First and foremost, the question. This happens because of two toxins called toxin A and toxin B. So remember, Clostridium difficile has two toxins. It has something called toxin A and it has toxin B, and these are the ones responsible for this condition. Further, how does the patient present? The patient is going to present with severe watery diarrhea. Why is the patient going to present with severe kind of watery diarrhea? Because let's see what has happened to the intestine. This is an intestine. Normally, you'll always draw the intestine like this, closed. What have I done over here? Instead of it being closed, I've opened it. This is the open version of the intestine. And all of you can appreciate that there is a definitive membrane, dirty, yellowish, necrotic membrane that is present on the mucosal aspect. That's the next question that you get. It is present on the mucosal aspect. So a necrotic membrane on the mucosal aspect, that is what you know for Clostridium difficile. And when you look at it under the microscope, you will say that there's a lava that has erupted. Everyone's seen a volcano. We all know that in a volcano, a lot of lava is going to come out. So from the intestine, this dirty lava material is coming out. We call it a volcano-like eruption. So if you see a dirty membrane on the mucosa and you see a volcano-like eruption, like you saw over here, they gave you the characteristic volcano lava eruption. You're dealing with a case of Clostridium difficile, and toxin A and B are responsible for these. Okay, so coming to the next question. Clostridium difficile, still on the same bacteria, can be spread through all except. Clostridium difficile can be spread through all except orofecal route, direct, hand to hand, or needles. Think logically. This is basically a kind of diarrhea, intestine involvement. It's a kind of diarrhea, it's a kind of an intestine involvement. So if I think logically, how do things reach the intestine? Definitely by hand to hand contact, direct contact, orofecal contact. Reaching the intestine via needles, that is sounding a little weird, and that is what by common sense you had to rule out, that Clostridium difficile is not something that is going to spread via the parenteral route. It is not something that is going to go via the needles, and that is why this is the wrong statement over here. Finally, if you say you want to do the diagnosis, what is the media? Again, two names that you learn, but you don't need to know the full forms per se. CCFA, CCYA. So remember, for Clostridium difficile, Clostridium difficile, sil has a characteristic, it has a C sound and it has an F sound. So remember that when you're talking about difficile, we'll write it in that manner. When you're talking about difficile, we have to talk about CCFA, CCYA. So difficile has CCFA and CCYA. If you want to know their full forms in front of you, most welcome to learn but not required. CCFA, CCYA, these are the two that you have. So let's do a recap of Clostridium difficile. Number one, this is always happening after a long-term use of cephalosporins. How does it reach? It reaches by direct contact or orophycal route or hand-to-hand contact, never by needles. This organism has two toxins, toxin A and toxin B. What do they cause? They cause watery diarrhea in the patient. If I want to look at it under the microscope, I will see there is a pseudomembrane. There is a dirty membrane that is formed on the mucosal aspect. Microscopically, we see a volcano lava-like eruption, and in terms of the media, we see that there is CCFA, CCYA media that are used. Well, I guess we've done all the Clostridia. Why don't we take up a few of the pre-VIQs? That will be good. All of the following organisms cause gas gangrene except. So quick test. These are PYQs of the FMG exam. All of the following organisms cause gas gangrene except. So we have studied for gas gangrene Clostridium perfringens, Clostridium septicum, Clostridium novi. Novi is not here. Welchi. Welchi and perfringens is the same thing. So which of them does not cause gas gangrene? Clostridium difficile. Clostridium difficile causes pseudomembranous enterocolitis. Next question. Necrotizing enteritis, the very, very, very important question. Necrotizing enteritis. So first and foremost, septicum we've not studied only because it's not at all important. Enteritis, when I say enteritis, difficile is sounding right. You'll say, ma'am, it causes pseudomembranous enterocolitis, but they've not written pseudomembranous enterocolitis. They've written necrotizing enterocolitis, so not difficile also. Tetani, I know, causes tetanus, that smiling face, that tetanus, rhizus sardonicus. Actually, please remember, necrotizing enteritis can be caused by Clostridium perfringens, and that is exactly what I want to add on to your knowledge, that Clostridium perfringens or Clostridium tetani not only causes necrosis of the muscle, but it can also cause necrosis in the intestine. Basically, it can also cause necrotizing enteritis. That is something that I hope you guys will definitely remember. So answer for Clostridium perfringens. Moving on to the next question. 3-year-old boy fell down and injured his leg while he was playing on the ground. After a few days, there are crepitations felt from the injured area. What could be the probable causative organism? Corynebacterium diphtheria, Pseudomonas aeruginosa, Clostridium tetani, or Clostridium welchi. So please remember, a 3-year-old boy has fallen down. As soon as I see the history of an injury and a fall, I always start thinking of tetani. Right now I think of tetani, but when I saw later, after a few days, there are crepitations felt from the injured area. Now that is something I don't see with tetani. When do we see crepitations? What are crepitations? All of those, these are, we consider them as signs of getting old. Do you sometimes move your fingers or move your neck or your back or your knee or some joint and you get that click kind of a sound? There's a click sound that comes maybe from your fingers or your neck, or when you're sitting for too long and then you stand up from your back and your knee, that those click kind of sounds are known as crepitations. But in this 3-year-old young boy, there are crepitations which are coming from those injured area. That is because gas production has happened over here. Crepitations are an indication of gas, and over here after injury they are talking about gas gangrene. So when they are talking about gas gangrene, the organism that comes to my mind is either Clostridium welchi or it is going to be Clostridium perfringens. Okay, so yes, having said this, I think we can move on to the last question of this family. Drumstick appearance. Drumstick. Think. Corynebacterium diphtheria, Clostridium tetani, Neisseria meningitidis, and Streptococcus pneumoniae. And by now we've done all, so we are going to discuss all. When we talk about the drumstick appearance, remember the answer to this question, Clostridium tetani. Because what kind of a spore does it have? TE, it has a terminal spore. So Clostridium tetani. Okay, in the previous crash course I had taught you Corynebacterium diphtheria has what kind of an appearance? It has a very classical Chinese letter or cuneiform appearance. It has a Chinese letter or cuneiform appearance. What about Neisseria meningitidis? The way you write M, we had said that we'll call it lanceolate, we'll call it lens-like. So Clostridium or Neisseria meningitidis is lanceolate, and Streptococcus pneumonia also. This is also going to be a diplococcus with a capsule. This is also going to present in a similar manner. So over here, drumstick appearance, Clostridium tetani. Well, having said that, we do wrap up the spore forming organisms, and we can definitely move on to the next family, and that is Enterobacteriaceae family. When we are talking about Enterobacteriaceae family, we have certain, I know for a fact that they are all gram negative. We are dealing with all gram negative organisms, but there's a certain kind of, general rule that we follow for the Enterobacteriaceae family. So please remember, when we are dealing, before I start with questions, when we are dealing with Enterobacteriaceae family, remember all of them are going to be motile, but Ella are not motile. All of them are motile, but Shigella and Klebsiella, these are immotile. They are non-motile. Repeating, all are motile, but Ella, they are immotile. Shigella, Klebsiella. All of them are catalase positive, oxidase negative. So all the organisms under this, they are catalase positive and oxidase negative, but Shigella, Ella. Remember, Shigella dysenteriae type 1. Shigella dysenteriae type 1 is catalase negative. Repeating, all are catalase positive, but Shigella, Ella, it is catalase negative. So last time repetition, all are motile, but Shigella and Klebsiella are immotile. All are catalase positive, but Shigella dysenteriae type 1 is catalase negative. So if I ask you this question, which of the following is a non-motile bacteria? Maybe you've not read all, or maybe you have. So if I ask you, is something that we studied, so you'll say, yes, we studied Ella are immotile, Klebsiella, Shigella, they are immotile. So the answer over here is very obvious, and this was a PYQ in the FMG exam. Shigella and Klebsiella, Ella are immotile. Coming to the next one. Catalase negative organism of the Enterobacteriaceae family. All of them are catalase positive except one organism, and that was Shigella dysenteriae type 1. Shigella dysenteriae type 1 is said to be a catalase negative organism. So I think the basic is pretty much clear, and we can move forward with the classification. How do we identify this entire Enterobacteriaceae family? This Enterobacteriaceae family is identified as lactose fermenter, non-lactose fermenter, and late lactose fermenter. Very simple. Either they ferment lactose, or they don't ferment lactose, or they ferment lactose slowly and after a while. So late lactose fermentation. LF, NLF, LLF. That is how we learn it. Lactose fermenter, non-lactose fermenter, late lactose fermenter. Remember, the only two lactose fermenters that you have over here are E. coli and Klebsiella. E. coli and Klebsiella are lactose fermenters. All the others, Salmonella, Shigella, Proteus, Yersinia, everything else of this family then automatically becomes a non-lactose fermenter. So if you remember coli and Klebsiella करेंगे. Coli and Klebsiella ferment करेंगे. That is how we've learnt it. Coli, Klebsiella करेंगे. Everyone else is not going to ferment lactose. And then we have Shigella soni. Shigella soni. Soni means keep sleeping. Wakes up very late, probably just one of us. Shigella soni keeps on sleeping, wakes up very late, so that is a late lactose fermenter. Let's repeat. Klebsiella and coli karenge ferment. They will be fermenters. Everything else, Salmonella, Shigella, Proteus, Yersinia, all Shigellas, all others, they are non-lactose fermenter except Shigella soni, because it keeps sleeping, wakes up late. That is a late lactose fermenter. So how did we identify? On what, what is the way of identifying lactose fermentation? What are the two media that we use for lactose fermentation? Number one, we have something known as a McConkey agar. Number one media that we have is a McConkey agar, and the number two media that we have is the clad media. What are they telling you? In both of them, we find out lactose fermentation versus non-lactose fermentation. In clad media also, we find out lactose fermentation versus non-lactose fermentation. But the colors are different. Over here, for lactose fermentation in McConkey agar, you get a pink color. Whereas when we are talking about clad media for lactose fermentation, over here there is a yellow color. There is a yellow color that is obtained. So repeating that when we are dealing with McConkey agar, lactose fermentation pink color. When we are dealing with clad media, the lactose fermentation is going to give you a yellow color. So you can see all this yellow color over here, lactose fermentation. So the next question will be that color change is happening. Color change is happening, which means there's going to be a medium, there's going to be an indicator. What is the indicator for McConkey agar? So firstly, what all does McConkey agar contain? It contains plant. What is plant? P for peptone, L for lactose, A for agar, N for neutral red, T for taurocolate. Let's repeat. P for peptone and agar. Every medium has peptone and agar. L for lactose, obviously, because you want to find out whether lactose is fermented or not. N for neutral red, because that is the indicator. That will change the color. That will change it to pink or yellow. That's the indicator. And T for taurocolate, because bile salts are needed. Similarly in clad, so if I ask you that what is the indicator, what is the color indicator that you have for McConkey agar and what is the color indicator that you have for clad? So remember, for McConkey agar, the color indicator that you will tell me will be neutral red. Whereas for clad, can you see it's all bluish in color? Because the indicator is bromothymol blue. The indicator is bromothymol blue. These are exactly two questions that I have. What is the indicator used in McConkey agar and what is the indicator used in clad? And I think now you can tell me what was the indicator in McConkey agar. McConkey agar was something by the mnemonic plant, right? So N for neutral red, N for neutral red, and for clad the blue color one. So for clad it is bromothymol blue. I hope these two PYQs will also be clear for all of you. Now there's no problem in solving them. So McConkey also tells me lactose fermentation by a pink color. Clad also tells me lactose fermentation by a yellow color. But what is better? If I ask you McConkey is better or clad is better, and as per the latest guidelines, remember clad is better than McConkey. Clad is going to be better than McConkey. So now my question to you is, why is clad better? Clad is better, and now my question is why so? So when am I using, if I broadly ask you what are all these organisms causing? E. coli, you see ma'am, E. coli very famously causes urinary tract infection. Klebsiella can also cause urinary tract infection. Proteus can also cause urinary tract infection. So basically these are all media that you will use when you're dealing with a patient of UTI. So UTI can be caused by gram-negative organisms like all these you studied, E. coli, Klebsiella, Proteus. So you'll say all gram-negatives. So will gram-negatives grow on McConkey? Will they grow on clad? Yes, they will grow on both. So why is clad better? Because sometimes UTI can also be caused by gram-positive organisms like Staphylococcus aureus can also cause UTI, like Candida can also cause UTI. Now the problem is that McConkey doesn't grow them. McConkey only gives importance to gram-negative organisms. It gives more importance to gram-negative organisms. Clad on the other hand, clad is going to be giving importance to everyone. It will allow the gram-negative and the gram-positive organisms to grow. It will allow gram-negative and the gram-positive organisms to grow. So can I say McConkey is very, very inhibitory. It is not letting the it's not letting the positivity come out only. It's not letting the gram-positive organisms grow. But clad is less inhibitory. Clad, remember, why did I highlight these two alphabets? Clad is less inhibitory. It allows everyone to flourish. It allows everyone to grow. And I'll show you two questions of the previous year of AIMS and NEET. Very important. Both of them are the same questions, only the language was different. Let's read. Clad medium is considered better than McConkey agar for what reason? You know your reason? What does it allow? You will say first option. It differentiates lactose fermenters from non-lactose fermenters. Both of them do that. CLED also differentiates, McConkey also differentiates. But why is CLED better? It allows everyone to grow. It allows both gram-positive and gram-negative organisms to grow. Means it allows staph and candida. What are they? Staph is gram-positive, candida is gram-positive, and CLED is better because it allows everyone to grow. It is less inhibitory. It allows everyone to grow. Next question. CLED medium is considered better than McConkey for the following reason. What is the reason? It will allow everyone to grow. So let's read. It supports gram positive and gram negative. Yes, I found the reason in the first option only. It definitely supports everyone. It supports gram positive as well as gram negative organisms. So remember, what is better? CLED over McConkey. Okay, having said that, coming back on the basis of this, what are the two lactose fermenters? कौन करेंगे? Ferment करेंगे. Coli and Klebsiella ferment करेंगे. So you will say, ma'am, this means that if I try to grow them on a McConkey agar, means E. coli will also give me a pink color because it is lactose fermenter. Any lactose fermenter, E. coli will also give me a pink color. Klebsiella will also give me a pink color. How do I differentiate? So for example, you get a pink color over here. How will you tell me that whether this is E. coli or whether this is Klebsiella? Problem ho gaya? Both of them will give a pink color because please remember, Klebsiella not only gives a pink color but also gives something called, can you see it's looking very shiny and mucoid? It's pink, okay. So on the basis of pink, you will say, ma'am, pink could be E. coli also, it could be Klebsiella also. But Klebsiella is having a capsule. Klebsiella is a capsulated organism, and anything that's capsulated always gives these shiny mucoid colonies. Anything that's capsulated will always give shiny mucoid colonies. So E. coli doesn't have a capsule. E. coli doesn't have a capsule. Klebsiella has a capsule. Remember, Klebsiella has a capsule, and that is why it's going to give these kind of shiny mucoid colonies. And mucoid means if you try to pick it up, it will stretch. If I try to pick it up, it's going to stretch like this. It's going to be stringy like this. So please remember, when you are getting mucoid and stringy colonies, that is indicative that the organism has a capsule. And over here, out of E. coli and Klebsiella, the best answer for a capsule is going to be Klebsiella. I hope the differences are sorted. Coming back, I've understood how Klebsiella is going to not only give me a pink color but also a mucoid colony. Now coming back to E. coli. Before I push you into lots of questions, what all does E. coli cause, guys? I hope you remember E. coli, two main things. E. coli can cause a lot of things, but the two main things that it causes are UTI and diarrhea. These are the questions, UTI and diarrhea, these are the ones that they're going to ask you in the exam. So first and foremost, let's talk about UTI. UTI, urinary tract infection. What sample will you take from the patient? When you're suspecting urinary tract infection, common sense, we'll take a urine sample from the patient. What kind of a urine sample? We have to take a clean catch. Means the patient will be instructed to clean the genitalia beforehand. Clean catch, early morning preferably. Early morning, midstream urine sample. So clean catch, early morning, midstream. What do I mean by midstream? You will have to instruct the patient that when you go to pass urine early morning, divide the stream of urine into three parts. So first pass 10-20 ml of urine, don't collect that. Then collect the next 10-20 ml of urine, and after that again throw away the remaining 10-20 ml. So you don't collect the first part of urine, you don't collect the last part of urine, you are only interested in the midstream sample. That sample is what you collect. And what is the culture that you will grow it on? Which is better, McConkey or CLED? We've studied CLED is better, CLED is being used nowadays. So you'll find out whether there's E. coli, whether there's Klebsiella, you'll find out. What is the criteria for diagnosis of UTI? For the diagnosis of UTI, we use something known as the CAS criteria. What is CAS criteria? CAS criteria says that You will give a diagnosis of UTI when the bacteria are more than 10 to the power 5. 1, 2, 3, 4, 5. When the bacteria are more than 10 to the power 5, that is when you are going to give a diagnosis. Means 1 lakh colony forming units per ml. When there is more than 10 to the power 5, why have this criteria been kept? Why not 10 to the power 4 or 10 to the power 3, 2, 1? Because are bacteria only present in case of UTI? No. Aren't bacteria normally also present? Don't we have bacteria in the urinary tract which act as commensals? So maybe there is E. coli as a commensal. 10 to the power 2 are seen. Will you give it as a case of UTI? No. If you want to call it a disease, if you want to call it pathogenic and disease, you must cross 10 to the power 5 colony forming units, CFU. However, there are some exceptions to the CAS criteria. When do we consider a count less than 10 to the power 5 also significant? Remember, you will consider it significant in case of a SPA. What is a SPA? S means suprapubic aspiration. Means what? Has the urine come out via the normal route? Has it come out via the urethra, via the urinary tract? No. You have directly punctured the bladder and taken the urine. You have directly punctured the bladder, suprapubic aspiration. Are you going to, so if urine would have come from the direct route and urine would have come through the urethra, then the commensals would have been there. Then you would have had to wait for the 10 to the power 5 cutoff to call it pathogenic. But when you've directly taken the sample from the bladder, then the commensals are not there. Then even 1 or 2 bacteria are significant. Then even a 10 to the power 2, 10 to the power 3 is going to be significant. So in case of suprapubic aspiration, don't consider this cutoff. Next, in gram-positive organisms, like staph and candida, they are not commensals of the urinary tract. They are not commensals. So even if they have a count of 10 to the power 2, 3, you consider them as significant. Similarly, A for antibiotics and diuretics. Many a times in our country, patient will say that, from a local chemist, I took some antibiotics, then I have come to you for the sample. I've been taking antibiotics since 5 days. After that, I'm feeling that I'm not showing much of improvement. So now I've come to you to give my urine sample. So antibiotic will anyway decrease the bacterial load. So you won't wait for 10 to the power 5. Below that also you will give the diagnosis. Same way patient says diuretic. If the patient is on diuretic, this means the urine is already diluted. The urine is coming to you as a diluted sample. Then again you don't wait for 10 to the power 5 cutoff. So repeating, what is the significance, or finally what is going to be the mnemonic for the CAS criteria exceptions? Firstly, CAS criteria says that you call significant UTI over 10 to the power 5. However, if we have suprapubic aspiration, gram-positive organisms like Staph aureus and Candida, or any antibiotic diuretic therapy, we don't follow the CAS criteria. This is when E. coli causes UTI. What about E. coli causing diarrhea? Now that is going to be very, very tough. Obviously, if I've said it's very, very tough, means I'm trying to just scare you because it's very, very tough in the textbooks, means we are going to simplify it. So first let's simplify it and then we'll move ahead with lots of questions, right? These are all the diarrhea strains that you have for E. coli, all the diarrhea strains. And can you see what is the difference? EPEC, so P alphabet, then changes to T, so ETEC, changes to IEIEC, HEHEC, and EAEC. Remember, these are all the different strains of E. coli that can cause diarrhea. What do I have to know? So E will always stand for entero. Okay, so it's going to be entero something E. coli. For example, I say entero something E. coli, entero something E. coli, entero something E. coli. So over here, entero pathogenic E. coli. What does enteropathogenic E. coli cause? Enteropathogenic E. coli causes P for pathogenic, P for pediatric. P for pathogenic, P for pediatric, it causes infantile diarrhea. Enteropathogenic E. coli causes P for pathogenic, P for pediatric, infantile diarrhea. ETEC, enterotoxigenic E. coli, T for toxigenic, T for traveler's diarrhea. Easy to learn. Enterotoxigenic E. coli causes traveler's diarrhea. Entero-invasive E. coli, invasion, something that is going in the cells. It's not going only till the intestine, it's going inside the intestine, inside the cells. Anything that will invade will cause blood to come out. Blood in the stools, what is that condition known as? Blood in the stools is known as dysentery. So because it is invading, it causes bleeding in the stools, that is dysentery. Enterohemorrhagic E. coli, so it will cause some bleeding, it will cause some hemorrhage, and it causes HUS. What is HUS? For those who are unaware, HUS stands for hemolytic uremic syndrome. HUS stands for hemolytic uremic syndrome. Hemolytic means blood is going to break. Blood is going to break. Uremic means kidney is going to get affected. Kidney is going to get affected. For those who don't know, please remember that this hemolytic uremic syndrome has a very classical triad. It has a very classical triad that you have to follow in the exam. And what is that triad? We call it the RAT triad. We call it the RAT triad. Means when you're dealing with hemolytic uremic syndrome, R stands for renal insufficiency. There is kidney involvement that occurs. A stands for anemia, because I think you know hemolytic uremic. So which kind of anemia? Hemolytic anemia. And also there is something that is known as thrombocytopenia. There is also known as thrombocytopenia. There is also thrombus formation that occurs. Clot formation occurs, thrombocytopenia can occur. So remember RAT, renal insufficiency, anemia, and thrombus and thrombocytopenia. Hemolytic uremic syndrome caused by hemolytic, so hemorrhagic E. coli, enterohemorrhagic E. coli. The only one for which you have to learn the strain. What strain of EHEC causes hemolytic uremic syndrome? O157H7. O157H7 causes hemolytic uremic syndrome, enterohemorrhagic E. coli. Lastly, enteroaggregative E. coli. Now, guys, what kind of diarrhea? Persistent diarrhea. How do I learn that? Enteroaggregative. You and I, we are together in this crash course, microbiology crash course. We really don't like the subject. Let's be very honest about it, because the subject has a lot of cramming up and too much of mugging up. So we don't like it. We keep forgetting it. It's volatile. So what have we decided? We've decided we'll do a crash course. All of us will sit down together. When all of us sit down together, united we stand, we mean we are going to make a persistent impact. If we decide cumulatively, if we decide collectively that we are going to study and we are going to crack this subject, we will definitely do that, right? So when all of us get together, we have more impact. So similarly, when all these aggregate together, when there is enteroaggregative E. coli, they have a longer time of diarrhea, persistent diarrhea. And one more thing, when you try to grow it on HEP2, what is HEP2? HEP2 is a kind of a cell line. HEP2 is a kind of a cell line. So please remember, on HEP2 cell lines, it gives a stacked brick appearance. Not any of these, only enteroaggregative E. coli gives a stacked brick appearance. Stacked brick means something like bricks. Again, can I learn it like aggregated bricks? Bricks which have been kept together, bricks which have been aggregated. So everything will make sense now. Enteroaggregative E. coli causes aggregated brick appearance or stacked brick appearance, and once we aggregate, long term, means persistent diarrhea. Let's do a very rapid revision of all these mnemonics that we've just studied, guys. Firstly, enteropathogenic E. coli, P for pathogenic, P for pediatric, infantile diarrhea. Enterotoxigenic E. coli means T for T, traveler's diarrhea. Enteroinvasive, invasion will cause bleeding in the stools, dysentery. Enterohemorrhagic, hemolytic uremic syndrome, O157H7. Enteroaggregative, long-term, persistent diarrhea, aggregated bricks, stacked brick appearance on HEP2 cell lines. These are all the diarrhea strains. Repeating, enteropathogenic, enterotoxigenic, enteroinvasive E. coli, enterohemorrhagic E. coli, and enteroaggregative E. coli. All these are important E. coli strains which cause diarrhea. Well, having said that, I think we are ready for some questions. Which organism causes HUS, hemolytic uremic syndrome? We just studied, okay, when I read the options, I think there's no confusion at all. Answer to the question E. coli, but which kind of E. coli? Hemolytic uremic syndrome, HUS, is caused by enterohemorrhagic E. coli. And did I teach you any strain also? O157H7 strain of enterohemorrhagic E. coli causes hemolytic uremic syndrome. Next, again, indirect way of asking you. Hemolytic uremic syndrome, short forms given. Now we know it. Hemolytic uremic syndrome is caused by EIEC, EPEC, ETEC, or EHEC. So, enterohemorrhagic E. coli. Coming to question 3. Traveler's diarrhea is caused by traveler's diarrhea. IEC, PEC, TEC, or HEC. T for T. Traveler's diarrhea is caused by enterotoxigenic E. coli. See how the questions are becoming so smooth and so simple to follow. Next question. All of these are PYQs of the FMG exam. Antimicrobials are given in which type of diarrhea? Antimicrobials or antibiotics are given in which kind of diarrhea? So do you give it in diarrhea caused by osmosis or secretory diarrhea? Or do you give it in something like a viral diarrhea, that is rotavirus? Or you give it in travelers. Travelers diarrhea is which E. coli? Enterotoxigenic E. coli, basically bacteria. So in which of them do you give antimicrobials? Which kind of diarrhea? Obviously we give antimicrobials and antibiotics when we are dealing with bacterial etiologies, not for viruses, not for osmotic or secretory kind of diarrhea. Antimicrobials are obviously given for bacterial, travelers, ETEC type of diarrhea. Next, which of the following shows the stacked brick appearance on cell lines? Stacked brick appearance. I also called it aggregated brick appearance. So which of them? Remember entero-aggregative E. coli. Entero-aggregative E. coli. So that is going to show you the stacked brick appearance. Let's move forward. Okay, let's take up the next question, guys. 5-year-old girl attended a birthday party at a local fast food restaurant. After 48 hours, she developed cramping abdominal pain and a low-grade fever and had 5 episodes of loose bloody stools. She is taken to the emergency room next morning as diarrhea continued and she looked pale. So overall, right now it looks like a food poisoning. Fast food restaurant with fever, with pain and loose bloody stools are there. And because her diarrhea is continuing, she started looking very, very pale. She's taken to the emergency room. On examination, she is hypotensive with tachycardia. So obviously so much of blood loss has happened, so much of water loss has happened. So obviously she is becoming hypotensive with tachycardia and her blood reports have shown hemoglobin 8. Is she coming across as anemic? Is she coming across as anemic? Yes. Then they found out that there is thrombocytopenia. Okay. So they've also found out that her platelet count is low. There is thrombocytopenia and there is evidence of hemolysis. So which kind of anemia is there? Hemolytic anemia is there. Is this sounding like some kind of a triad? When I look at the options also, did we study something called RAT? Only this part was missing in the question. The renal involvement is not mentioned. Other than that, hemolytic anemia is mentioned, thrombocytopenia is mentioned. Are we indirectly targeting a case of hemolytic uremic syndrome? See, diarrhea causing organism following which there is hemolytic uremic syndrome, enterohemorrhagic E. coli. And enterohemorrhagic E. coli, which strain? It is O157H7. That is how a question of EHEC will come in the paper. Coming to the next question. The blood of a patient is suspected to have pyogenic live abscess was sent for culture. So there's a patient who has pyogenic, it would be a liver abscess actually. Pyogenic liver abscess is sent for culture. That makes more sense, right? So when we are talking about liver abscesses and conditions, two things. Either they're dealing with a pyogenic liver abscess or you're dealing with an amoebic liver abscess. So amoebic liver abscess means parasite, entamoeba, parasite. Here when they say pyogenic liver abscess, bacteria. The colony morphology shows large mucoid colonies. When I say large mucoid colonies. Mucoid is something which I have studied, is organism that contains a capsule, and out of these the organism that contains a capsule is Klebsiella pneumoniae. The organism with a capsule is going to be Klebsiella pneumoniae. So this mucoid word and Klebsiella will always go hand in hand. Coming to the next question, PPA test is used for the diagnosis of. PPA test is used for the diagnosis of. So please remember P for P, PPA test is used for the diagnosis of Proteus, and that is the next organism that I am going to teach you. So first let me teach you and then I'll come back to this. What is Proteus? Everything about Proteus has been written over here. So P for, there are two P's that you have to know. So first, it has something known as the PPA test or the PPA reaction positive. You don't have to know the name of the entire reaction, but that is phenyl pyruvic acid. If you wish to know the name, remember Proteus is phenyl pyruvic acid going to be positive. Another P for Dine's phenomenon. What is Dine's phenomenon? Let me tell you that. So before I move on, can you see U and S? U for what enzyme does it produce? It is urease positive. And what motility does it show? It shows the swarming motility. It shows the swarming motility, and urease positive. Do you guys know what is swarming? This is swarming, that if you're dealing with the case of blood agar, can you see these concentric? So bacteria grows, then another circle of motility, another circle of motility. So this is referred to as the swarming motility. When you're dealing with microbiology, what are all the organisms that show you swarming motility? PVCs. PVCs show us swarming motility. What is all of that? Remember, P for Proteus, VP. VP means Vibrio parahemolyticus. That way you'll recall it's not Vibrio cholera, it's Vibrio parahemolyticus. C for Clostridium tetani and S for Serratia. Let's do a recap. We have P for Proteus, VP, so that is Vibrio parahemolyticus, Clostridium tetani and Serraceus. They show you the swarming motility. So I've understood the meaning of swarming motility. I've understood that it produces urease. I've understood it has a PPA reaction positive. It is something called Dine's phenomena. What is Dine's phenomena? It's the story of you meeting your best friend versus you meeting your enemy. Before I get on to it, very interesting. So whenever, for example, in the lockdown, you were not able to meet your best friend for 2 years, for example. Now after 2 years, when you meet your best friend means someone you vibe with, someone you relate to, someone whom you are very fond of, someone who's just like you for that matter. So when you meet your best friend after 2 years, you're going to just run towards him or her and hug her because you're meeting him or her after a lockdown after 2 years. So if you're same and if you vibe together, you'll hug each other. What if you meet your enemy after 2 years? You're going to maintain a distance, right? Because you're not same kind of people. You don't gel together. You don't vibe together. You don't like each other. So you won't hug each other. You'll stay away. You'll maintain a distance. Same happens with Proteus. Please remember, same happens with Proteus. So for example, this is a blood agar that I have over here. Over here, I put one Proteus, strain number 1. This is also a Proteus. So from patient 1 also I have taken Proteus. From patient 2 also I have taken Proteus. Now, if they are the same, if they are the same strains, means they are best friends, what will they do? This one will start swarming from here. This one will start swarming from here, and ultimately they'll all fuse into each other because best friends are supposed to gel. Best friends are supposed to hug each other, right? However, if from patient 1 and if from patient 2, they are different strains. If they are different strains, you'll say that is exactly what has happened here. Can you see this has stopped swarming over here? This has stopped swarming over here and this one has stopped swarming over here. There is a line present. I'll show you one more picture. See, very classical. This was Proteus number 1. This was Proteus number 2. It started swarming but it stopped over here. This started swarming but it stopped over here. So if you see that there is somewhere a line of demarcation present, means distance has been maintained. Line of demarcation is present and distance has been maintained. Are they same or are they different? Are they best friends or are they enemies? If there is a distance maintained, you will say they are enemies. They are different strains of Proteus that you are dealing with. They are different strains. So let me make you ask you one question. The phenomenon shown below is indicative of. So can you all see the phenomenon shown below is the Dines phenomenon for sure, right? This is the Dines phenomenon. And do we also know that I can see a line in between? So they are different strains. So if I ask you what is the answer, are they same strains of Proteus, different strains of Proteus, different strains of Parahemolyticus or Clostridium? I know this is Proteus, and if distance is present, they are enemies. They are different people. So what is the answer in that case? These are different strains of Proteus. So repeating once again, guys. Repeating once again. P for Proteus, P for PPA test positive, P for Dines phenomena positive, O for they result in a fishy odor. This organism has a fishy odor. It is urease enzyme positive and it shows us swarming motility. What are all the other organisms that show us a swarming motility? Swarming motility is also shown by PVCs, which means we have Proteus, Vibrio parahemolyticus, Clostridium tetani, and Serratia. All of these belong to the PVCs that you have to know, right? All of them show swarming motility. Well, I guess we can take a break on that note, and obviously the family that we are dealing with, that is the Enterobacteriaceae family, still has a really long way to go. So we will be taking that up in the next part of the microbiology crash course. So we've covered E. coli, Klebsiella. We've also covered Proteus. We would be covering Salmonella, Shigella. Along with that, we'll also be taking up Pseudomonas, Rickettsia, Chlamydia, and all the tiny bacteria that we are left with. So hope also Vibrio, one very important family. So these are the bacteria that are left, which we'll be taking up in a slightly lengthy session that we'll have tomorrow. But yes, tomorrow we would be wrapping up, or in the next session, we would be wrapping up the final bacteriology portion so that we can move on to virology and parasitology after that. Out of all, bacteriology always takes a little more time. It's a little more time consuming because there are so many bacteria. The problem is that I can also teach you all the bacteria in one day in a single session, but then it becomes a big mess in the mind. So it's always better to break it up into smaller portions and study bacteriology for a longer retention. Well guys, as I said, the PDF of this session, I would also be sharing it over Telegram and other social media platforms. All the links to the social media platforms are already given in the description below, so you can definitely join in and download the PDFs from those. Thank you so much for joining in, and I'll sign off for today for this session, and I'll be meeting you in the next session with all the other bacteria important as a crash course. Thank you so much guys. Study well. All the very best.